toggle all these on. Yeah, do it. Okay, we appear to have nice. Nice. Just need to turn the volume off in a few places. Yeah, okay. So we're now live on Facebook, sorry, on YouTube. And
don't have any feedback now?
A little bit.
Okay, I'm seeing it on Fem's YouTube. Are we on IMD?
Yeah, on YouTube it's already up.
Yeah, it just goes straight onto YouTube, so that's great. So if you're watching, hello, welcome to the IMD live stream preamble.
YouTube it's already. Sorry.
Yeah. So what I'll do in the meantime is we can share. Ooh, my laptop's having a little bit of a wobbly. Cool, and then we appear on the side. We are ready in Twitter. Almost, anyway. What's it not letting us do it? That's fine, that's fine, that's fine. Let's try it again. Cool. Let's try it again. Cool. So I don't have us yet on IMD Facebook. But I'll try and fix that now. Cool. Thanks, guys. Cool, okay, we're ready on all of them now. Okay.
Did the tweets go out for IMD and FEMS? Or
Not yet. I'm just going to put us live on Facebook.
Okay.
And then I'll do the tweets last. But we should be live on both Facebooks, both IMDs.
Great.
Yeah. I just need to embed the YouTube stream in the home page. So
Is it live on the?
let me just check. It might not be live. It might not be public. Yeah, there we go. Publico. And then we just check it's the same here. Yeah. Yeah, cool. Now let me see if I can embed this. Bum, bum, bum.
Do you want to tell me everyone is muted? Then the picture is.
Yeah, I will. Oh, it's not when you're muted, it's just when your camera's off. Yeah. Let's see if this works. Cool. Yeah, nice. Okay, so the live stream is now on the homepage. Do you guys mind checking that for me? If you can see it on the IMD homepage.
Yeah, it's there.
Cool. And should I tweet us out on the Twitter pages?
Yeah, I think we're ready to
IMD
go.
homepage.
Got 2 minutes. 2 minutes to go.
Nice. Managed to not be late. That's always good. Managed to not be late. That's always good. Okay, cool. So we should be live in every space. So if you're already watching, thanks for joining us. And soon we will begin our opening ceremony. Okay. Okay, we've got 1 minute. Just going to pin these to the profiles on the Twitter pages. Oh. Just just going to pin these to the profiles on the Twitter pages. Can you check on the IMD Twitter? Because I just want to see if has it come out properly. It just seemed to be a code rather than the actual stream.
Yeah, no, I don't see it there actually.
Let me try again.
On IMD or FEM's Twitter?
IMD
Twitter. Let's try it one more time. Other than the actual stream.
Yeah, no, I don't see it there actually.
So I just deleted it because it wouldn't let me do it twice without it being a duplicate.
I was just thinking, actually, we're starting like IMD a day early. I was wondering if it is actually International Microorganism Day anywhere in the world at the moment. Like it must be.
So it nearly is.
Australia.
Yeah. That's annoying. It won't let us do it on Twitter through the IMD account. So I'll have to just retweet.
I can play with that, and then we can get started with the opening ceremony.
Yeah. I'll just retweet the FEMS one.
Cool.
For the meantime.
It just comes up as a link.
Yeah. It might be we don't.
If you click it,
Yeah, okay. Right. Okay. Well, if you joined us today, welcome to the IMD opening ceremony. And I'm joined here today by the whole IMD team. We have Isabel Zakaria, Claudia Godino, Ben Liberton, myself, Joseph Shuttleworth, and Eleni Kusari. We're streaming from the Hague Humanity Hub in the Netherlands, but we're joined by Ben in Sweden and Claudia and Isabel from Portugal. Just want to welcome you to our live stream. For the next 24 hours, we're going to be bringing you a huge plethora of microbiology content. If you follow any of our social media accounts, or you're watching us there, or you want to navigate to our website, internationalmicroorganismday.org, you can find the full program as a PDF on the homepage, and you can find out the timings of every session. We're just going to give you a little bit of an introduction to IMD, the history of this initiative, and talk you through, a few bits about the day, a few initiatives we had running this year, and then we will commence with our opening sessions after this. But Claudia, do you want to just share the PowerPoint slides and we can run through some brief bits of information for you?
Hello, good morning from Portugal. I will then share some slides. Please let me know if you are seeing them correctly.
I can see it. Looks good.
Yeah, all good.
Okay, so here goes a bit on the International Microorganism Day. So as you may know already, the mission of this day is to disseminate and promote microbiology as an area of professional activity and careers, and also to raise awareness among young people, society in general, everyone in the public, about the role that a multitude of microorganisms play in our lives. The date, so today in Portugal is still 16 September, but maybe somewhere in the world already is 17 September. This date was chosen because it was the date of the first description of a single-celled organism. So this Dutch Anton van Leeuwenhoek, which was not a scientist, with no richness, but he was able to develop a system of lens and to observe the first single-celled organisms, and this was the day in which he reported this observation in a letter to the Royal Society of London. So here are our little helpers. So we have Saka, inspired on Saccharomyces cerevisiae, and Bach, friendly bacterium, and Gilles, inspired on the fungi Aspergillus. So just to give you a very short introduction on IMD, IMD was indeed started here in Portugal in 2017, from which it was expanded to be celebrated in Europe in 2018, and in 2019 it was already a very global event. So there were commemorations all over the world, and with the coming of the pandemics in 2020, we were forced to move online. So if you if you were there to watch us, we were already also in a 24-hour marathon, such as this year. So the launching was in Portugal, as I told you before. It was here in Pavilhão do Conhecimento, which is a home of it's a science center. And there were a lot of exhibitions. You can see here some pictures. Actually, Professor Isabel is there in some pictures with Bach and Saka. It was a great day. Then, while she was the president of the Portuguese Society of Microbiology, in 2018, Professor Isabel made an extreme effort to expand the word on International Microorganism Day and was able to reach several microbiological societies and other organisms with the help of FEMS, and we were able to make IMD truly a global event. So we started in 2018 our presence in the social media world with Twitter and the website, but then we invested a lot in social media, and this allowed us to become truly global. So now we have a Facebook account, an Instagram, a YouTube, all with a unified name, Int Micro Day. So if you don't follow us yet, please do. And also contributing to our dissemination through the world was the microbe art competition. So it's already its third edition. And this year, 2021, we also launched a mascot art competition to find our fourth mascot for IMD. So here I pass the word to Joe to tell us a bit about last year's marathon.
Yeah. You can all hear me okay, right?
Yeah.
Good. Yeah. So last year, as I'm sure everyone's aware, we weren't able to do any physical events due to the pandemic. And so to kind of fill this space, we decided to put on a marathon 24-hour long live stream, which we're repeating in a different form this year. But yeah, the live stream went on for 24 hours. There were 58 speakers. You can opening ceremony from last year. We had the mascots join us in their costumes. We had some live microscopy. We had scientists from all over the world, and all streamed from a central Zoom call to different social media channels. People hopped in and out from different time zones across the entirety of the stream. And it was great fun. And we had so many wonderful faces and wonderful talks come together to spread and celebrate the joy of microbes, which is, again, why we're here today and tomorrow. If you take me to the next slide, we have a few analytics from last year. So in the small period of time around the actual live stream on International Microorganism Day, 17th of September, 2020, viewers streamed over 1,000 hours of video during that time. And we had, 4 chunks of the live section, of the live stream, 4 sections. And it's quite hard to know exactly how many viewers because, people watched on YouTube, Twitter, Facebook, on the homepage. But we had somewhere between 1,000 and 7,700 viewers, section 2 being our most widely watched section. You can see here a little bit of data on the kind of like percentage where people watched it. Most people watched it in some form on the YouTube channel, at least in terms of the watched time. But we had far more individual viewers on Facebook and Twitter coming for sort of like micro content. And you can see, the average watch time on YouTube was much longer than on the other social media channels. People seem to settle in and watch it for the entirety of a session, whereas on Facebook and Twitter, people would catch little bits of the stream and a few segments of the talks. So how many viewers exactly? It's hard to know. All the different social media channels measure different metrics, but several thousand people for sure were there watching and enjoying our content. Hopefully many more this year. Do you want to take me to my next slide? So this is the hashtag data for International Microorganism Day, and this giant spike in the middle is actually the 17th of September, and you can see across the 6 months preceding, 3 months before and 3 months after IMD, we gathered around over 12 million impressions, over 4 million people saw the hashtag, 52,000 engagements, thousands of users actually using the hashtag, and nearly 6,000 individual posts with the International Microorganism Day hashtag. So if you're following today, use the hashtag in your social media posts, tweet about it, post on Facebook, Instagram posts, even TikTok posts if you're on that platform. So let's try and get as many impressions with the hashtag out over the next couple of days. We have the next slide. So here's some just social media stats. We have some small but growing social media accounts. Twitter, Facebook, YouTube, and also Instagram. We can see that in terms of the countries on our Instagram, it seems to be Brazil, Portugal, UK, US, and India. Huge following of women versus men. And thousands of impressions from all these accounts. And we really want to grow the YouTube channel, which is where all the recordings from the live stream end up. So we have a few, nearly 200 followers, but if you subscribe to the YouTube channel, you'll get the first notifications when all of these videos get put on the YouTube channel. So follow us on these accounts if you don't already, and find us there. And you can message us and post about us and talk to us there. So this is kind of some website statistics. So last year we built a new website, which you might be watching the live stream on, internationalmicroorganismday.org. So tons of wonderful blogs. We have education resources. There's a social media pack. There's a gallery of illustrations and pictures from previous events and some new content there we're going to talk about later. If you look at the countries where people are visiting our website from, you can see we have a spread of countries from every continent on the planet. A few really engaged countries like Japan, India, the UK, Portugal, Spain, Germany, Netherlands, Brazil, and US. But you can see that, every area of the world, some people are coming to the website, which is great. Good news is that even though 2021 is not over and we haven't even had International Microorganism Day yet, we've already had more visits and more visitors this year than the entirety of 2020. So lots of lovely traffic. We encourage you to go there, submit blogs to us, read blogs on the page, enjoy the content we put up there for you. It's a great place to get all of your IMD content and enjoyment out of the web. A few things to point out. If you're a real, microbe aficionado, or even a teacher or an educator, we have a really active blog full of articles and blogs about tons of different microbiology, tons of interesting stuff there. You can write to us if you want to submit blogs. We also post news about our developments of the day and about things like our live stream and different initiatives we have. But we also have an education resources section. And last year we posted 2 education resources for the use in schools by teachers or to do at home about making sourdough bread, and also building your own Winogradsky column to look at kind of microbial ecology in a small space. And this year we published 2 more ones. So they're just on the website as of today. And I think last week we've got making yogurt and making agar plates. So if you want to make your own yogurt out of milk, follow this one. There's a beautiful illustration there that takes you through all of the various steps you need to make yogurt at home. And then making agar plates, there's a really handy set of simple instructions for making your own agar plates to grow colonies of microbes from, inside your mouth, from your garden, from your home, to just kind of get an idea of what kind of microbes are living in your space. You can download these on both of the, all 4 of these education resources now, and bring them into school if you're a teacher, get the kids involved, playing with microbes in a nice, safe way. And that's it from me. I think, Claudia, do you want to talk about the mascot art competition?
Yeah, sure. So as I told you, this year we run our first mascot art competition so that we can find a 4th friend to our mascots. And it was a great success. It ran through 1 month, and we got actually 39 mascots competing to be the next IMD mascot. And these mascots come from 14 different countries, although this number might be high since not every contestant told us where he was submitting from. So a panel of 6 judges, so the IMD team, voted independently and elected our new mascot. I'll take this opportunity to show you our top 5 mascots. So in 5th place, you can find Beefy, inspired in Bifidobacterium, from Elisa Wolfson, who submitted from the UK. And then we have a COVID-inspired mascot, so it was Corona Terror, from Lee, from Singapore. Then our 3rd and 2nd runner-ups were submitted by the same author. So Jordan Bailey from Australia submitted 2 rust spores, the Rusty and the Tadeus. But our winner was indeed Rizu. So Rizu is inspired in Rhizobium bacteria, and it was also submitted by Elisa Wolfson. So congratulations to Elisa. It was a great design, and now we have our mascot family complete for 2021. So who knows if we are joining another mascot for next year. For now, this is the IMD mascot family. So this is it from me.
Great. Thanks, Claudia. And just for the remaining few minutes, Isabel, I believe you had some words to say.
Thank you, Joseph. And hello. Good morning, good afternoon, or good night to all those around the world who are following this opening session. This is a borderless online meeting on microbiology, and I'm extremely pleased to note that 5 years after the first edition of the International Microorganism Day, IMD, in Portugal, the day has become a celebration of microbiology at a global level. And well, I would like to acknowledge the essential role of FAMS, in particular, the very active and competent FAMS office and science communication sector in the internationalization of the day. And of course, well, my greetings to Claudia Godinho, who is an extremely important volunteer of IMD. And people may ask, well, why microorganisms have their International Day? We all recognize that microorganisms play a major role in life sciences, of course, but in everyday life and in a wide range of professional activities and career opportunities. Never forget this. Moreover, only the alphabetization and the literacy in microbiology of the society will allow all citizens, to understand and trust the science and reject unfounded and obscured theories. For this reason, all efforts, among them is the International Microorganism Day, put in the dissemination of microbiology will facilitate informed choices, for example, on vaccination, use of antibiotics, food quality control, food safety, advanced biofuels, and so on and so on. Well, governments also need to adequately inform their political decisions, as became crystal clear for the worst reasons I know during the new coronavirus pandemic. So remarkably, society is now giving value to specialists and disseminators who rigorously make the scientific knowledge understandable by common citizens, especially in mass media. Well, however, beyond COVID-19 pandemic, there are other areas of microbiology where essential and difficult individual political decisions also are needed. For example, due attention should be given to the exploitation of microbiological resources of interest in biotechnology to promote an increasingly sustainable bioeconomy, capable of generating benefits for the economy, the environment, and the society. And this is now a priority, as you know, because time to counteract climate changes is running out, and the associated economic and social costs could far exceed those of the pandemic. Also, a better knowledge of the control of the activity of harmful microorganisms allows an adequate response to the challenges they pose to human and animal health, to agriculture, food quality and safety, and so on and so on. Well, certainly, there is no lack of reasons to have an International Microorganism Day. Well, at last, I would like to thank all the speakers who gladly accepted to contribute to these 24 hours of microbiology online, and to thank you for participating in this borderless meeting to celebrate microbiology. Enjoy the meeting. Thank you.
Awesome. Thank you very much, Isabel. So yeah, enjoy the live stream. We've got lots lined up for you. Our first session is actually a video by Micropia. And we have our first guest who we'll bring into the room. And shall I pass over to you, Eleni, to introduce him?
Hello, everyone. Greetings from me as well. And thank you, Professor Isabel, Claudia, Ben, Joseph, and everyone that gave the talk of the opening ceremony. Hi, Jasper. Can you hear as well?
I'm muted. Sorry. Hi. Hello. I hear you. Good morning.
Hi. Welcome. So yeah, as Joe said, we are moving forward to our first session of the day, which is going to be about a tour in the Museum of Microbes. And I am referring to the museum called Micropia, directly from Amsterdam in the Netherlands. And I've been there a couple of weeks ago, and it was a fun and unique experience. So I'm very much looking forward for all of you guys to see what Micropia has to offer. So I have Jasper with me, who is the manager of the Micropia Museum, right? And he's going to give us a few words before actually going and having the tour. So yeah, Jasper, over to you.
Thank you, Eleni. Hello, everyone. My name is Jasper. I'm the head of Micropia, the world's first and still only microbe museum. And I'm really thrilled that you've asked us to be part of the program. Of course, the International Microbe Day, in our mind, of course, is a very important day to, well, get something more about microbes and their huge presence in our lives, even though a lot of people don't realize that, out in the open a bit more. So that's why I'm really thankful that we got this opportunity. But having said that, I'm presenting now from our lab, which is also part of the museum, which Eleni will know, of course. But of course, today we will show you a bit more about Micropia itself. And that's being done by Remco Kort, a professor from the Free University here in Amsterdam, who's also part-time the Micropia professor, because Micropia is more than a museum. We also want to be a platform where we not only tell our visitors something more about microbes, but also make connections between, well, for instance, the university and our public, but also different commercial partners, non-profits, other museums. And that's something that we're continuously working on. And that's why I'm also really thrilled to be here. Maybe we get to know some more of you guys here. If you're ever interested, just send us an email. We're always willing to hear you out. But like I said, I'd like to now give the floor to Remco Kort to give you a bit more information about the museum, how we got to build a Micropia, and then in between I'll be back here in the lab to tell you something more, and at the end, of course, for the Q&A.
Yeah, great. Thank you, Jasper. Just to mention to all of you guys that are watching that we are monitoring our social media channels. So if you do have any questions about Micropia or any other things that you want to know about IMD, do comment them below, and we're going to address them at the end of the session. So Ben, can we have the video from the tour, please?
Absolutely.
Great, thank you.
Hello, welcome. My name is Remco Kort, and I'm affiliated as a professor in microbiology with the Vrije Universiteit in Amsterdam and connected to Artis Micropia, the first and only microbe museum in the world. And what I will tell you about today is that this museum is actually more than only a museum, and let's dive into that. So I'm right here located in the museum at the center of Amsterdam, and the museum is actually part of Artis. It's the oldest zoo in Europe. And if you look here behind me, you see this map of Amsterdam, probably well known to many of you, with these typically horseshoe-shaped canals. And right here, you see where the microbe museum is located. So it's just next to the zoo. And in the development of the idea for Micropia, this was actually initiated by the former director called Haik Balian, and he was wondering what would be the role of Artis, what would be the role of zoo nowadays? Is this just a mausoleum to keep endangered species, or should a zoo be much more than that? Well, actually, it's good to know that Artis was founded as by the Zoological Society already in 1838 with a mission. And his mission was to reflect upon the position of mankind to nature. And considering this, it would be really great if we could, let's say, take a role again for a zoo. So it's much more just a place where you keep animals, but it's also a place of getting in contact to nature in the middle of the city, and actually also a way to stimulate our what is called biophilia. So that's the innate tendency to connect to nature, and Artis is really a beautiful place to do that. So it's actually not only only the zoo in terms of animals, but it's also a botanical garden, and you will find there a beautiful aquarium as well as a planetarium. And the former director was thinking, how can we extend that? Because he really liked innovation. So how to innovate a zoo? Well, you could think of, for example, a new animal. It's a bit complicated. But at one point, he considered that, in fact, this innovation is already there all around us. It's the majority of life forms which are invisible, but have never shown in a zoo. So this actually initiated the idea for Micropia now more than 15 years ago. And he figured out, well, how do we attract now young people to this zoo? And he was thinking of a kiss, because in the mouth, this is a very complex ecological system of microbes. And actually, during an intimate kiss, you transfer these microbes. So wouldn't be a kiss the way to also interest young people for the topic of microbes? So in fact, it all started with a kiss. So when I became involved in the development of Micropia as a museum, this was in 2006, and we were working in a small team with 8 people on the development of this museum. So we were there with an exhibition designer, with a science journalist, with a microscopist, and a content manager, and myself as a bacteriologist. And we were having meetings for a period of 8 years to talk about this development. And it was really complicated, because you can imagine this world of invisible microorganisms is enormous in terms of diversity, but also in terms of topics you can put in the museum. So we were brainstorming about how and where should we put the limits. And each time when we were considering a framework, there was another topic that was just falling outside of it, but definitely we wanted to show this as well. And this slide here gives you an example actually of such a framework that we considered in that time, by just dividing the microorganisms we wanted to show in different ecological systems. Like, for example, what do we find on our body? What do we find in space, on the subsurface, in our house, in the water? But as I told you, this really didn't work out to let's say, consider this framework as topics for the museum. So what we at one time decided is to just leave this for what it was, and just take a variety of themes, and as a central position is actually myself. So humans. So what do these microorganisms mean for me? And by working out a number of different themes, we were able to create a museum, and each time asking the questions, what is the impact of this microbial life for myself, to my own life? So, as I told you, we finally made it to a museum. Opened in 2014, actually by our Queen Maxima. And you see here leaving her microbes from her hand on a giant Petri dish. And you see she's doing that next to the former head of Micropia, Eveline Hensel. And this was, of course, a very unique way to open a museum. We had to promise that we were not storing any of the royal microbes in our collection. But apart from that, it was really great that the Queen of the Netherlands wanted to collaborate with this and open the museum. So what do you encounter now if you enter this museum? Well, first of all, we wanted to show the enormous microbial diversity, and show that actually microbial life is in a way much more extensive than all the visible life forms. So we designed this sculpture over here. And the interesting part about the sculpture is that it shows actually all life forms on this planet, but the microbial life forms, they lighten up. by these on this fluorescent sculpture, while the visible life forms do not. And by showing this tree, you can immediately see that there is so much more of invisible life on planet Earth than there is visible life forms like those we encounter in the rest of the zoo, like the elephants and the lions and everything. And we even needed a kind of magnifying glass, which you can see over here, to actually show the part of the animals that were present and on this present in the zoo. Two more things that I would like to tell you about this tree is that, as many microbiologists among you are aware of, is that all life on this planet can be divided in these three domains. So on the left, you will see the bacteria branching, and on the bottom you will find all the archaea. So all of the archaea are also invisible life forms. And then on the right you find the eukarya, and part of the eukarya are invisible. So it's the yeast and the fungi, but this also includes all the visible life forms, including the mammals and humans. And then there is this separate part here, the viruses. Well, and those are not really considered life forms because they are not able to grow independently without a host. So that's why we put them there under the tree. So such a museum, of course, well, it has never been done before. So there are a number of exciting challenges you're facing in the development. And one of them is, how do we show these microorganisms to the public? It was very important to us that we wanted to show them real life, because as in the normal zoo, you have all these animals. And we want to do exactly the same microbes. So we kept a living collection. We developed that of about over 250 of these microbes. And for each of them, we needed to develop the methodology in order to be able to show them for a long period of time under a microscope. And it was not only this was a challenge, but also the microscope itself, because, well, if you have people that are not trained, it's quite challenging actually to look under a microscope. So we developed our own microscopes in which we made them very robust, so they would not be damaged easily, and also easy to operate by, as you can see here, a joystick that allows you to navigate in this microbial world, so to speak, and an additional button that has a zoom function. So by doing this, really everybody is able to become a microbiologist and look into the microscope. And this is just an example. And that's also important, that we wanted to develop this museum with all kinds of separate exhibits. So in this particular case, you look at the medicine makers you will find in the soil. Very important because they actually provide antibiotics. One of the major achievements. So almost all of them are actually produced by soil microorganisms. And this is what we wanted to tell in this exhibit. So we had a microscope, the living microorganisms under the microscope. And here on the right, you find what we call a content screen. And this content screen can operate in several modi. So what you can see is the live image on that screen. So also other people will be able to watch. You'll find additional background information about the microbe. And also we gave an assignment to animators to make a short movie about actually what we wanted to show in this exhibit, explaining the major message and the importance of these microorganisms shown in the content to our daily life. So if we now continue our trip through the museum, you see that we took this quite literally. What does the microbes mean for us by including a body scan? And you can see over here with an interactive device, which allows you to navigate through all different sites in your body. And by doing so, you can get all kinds of information on, well, well-known term, I assume, to all of you, the human microbiome. So you can have a look at your intestinal microbiome, as well as your skin, the oral microbiome. And what's really cool about this feature is that it actually starts with a scan, and it scans your body and gives you an estimate for the total number of microbes that are inhabiting your body space. So let's continue our trip now through the museum. And over here, you see that it's actually a quite dark space. So we designed the museum in a way, it's kind of some kind of inverted laboratory where all the light is coming from the exhibits. So this also gives you the impression of a rather mysterious world in which you become submerged, so to speak. And to add to that effect, we created a specific sound design, which allows you to actually get an experience of these microbes. And even though, we know, of course, that they do not make any sound, but it adds up really to having this submerged experience in the museum. Here on the left is actually one of the greatest symbols of Micropia. It's a tardigrade. It's a small animal. You find it everywhere around the world, even in your own back garden. It's a bit smaller than even a 1 millimeter. And it's really known to be a very extreme survivor of all kinds of conditions. And it's not being active under these conditions, but it has this extreme characteristic that it can be completely dehydrated. And in this dehydrated form, survive all kinds of conditions, even travel through space. And once it finds a surrounding where where there is water and where there is food, it kind of inflates again and just starts living again in this new environment. So continuing our trip, we do see here that life of microorganisms has been extremely diverse, and there's actually no niche on this planet in which bacteria are not able to thrive. And that's what we wanted to show in this microbial theater, the extremophile theater. So over there, by turning this giant wheel right here, you're able to move to all kinds of different environments and get information about the microbes that are living in these environments. So like I already said, the thermophiles living under extreme conditions concerning very high temperatures. So this is in the deep sea. But then we have also, for example, at Chernobyl, the organism Radiococcus, Deinococcus radiodurans, which is able to survive very high doses of radiation, and it has a very unique mechanism to repair its DNA. So you see all these kind of fascinating adaptations that are developed over the evolution by microorganisms, allowing them to thrive under all these different conditions. And I think this is also a very important message, because we can make use of these properties for all kinds of different applications. So that brings me to the collection of microbes that we keep. I already told you more than 250 of these microorganisms are actually kept in the museum. And we set up the laboratory in a way that is actually integrated part of the museum. So there's this enormous window in the museum, and you can see the technicians here at work in the laboratory, keeping the collection. And also this is a very important role in education. So we have interns here that are doing their internship over a period of time. So that's an additional role here of the lab. And I would like to give the word now to my colleague Jasper. He's actually right there in the lab, and he's going to tell you some more about this.
Thanks, Remco. Hello, everyone. My name is Jasper Buijks. I'm head of Micropia, standing in front of our own Micropia lab. As you can see, this is part of the museum as well, but this is also the heart of the museum. Because like Remco just told you, Micropia is full of live microbes, over 250 different species of them, and they're all cultivated here in our lab. So if we want to show a tardigrade, a water bear, we also have to cultivate some algae to feed it. And that makes it a very unique location, because it's possibly also the most diverse collection that you would find in such a small location. And that makes it also interesting because we have all kinds of different lab technicians with different expertises, from marine biologists to virologists to bacteriologists, you name it. and we've got them. But the cool thing is, it's not just amongst themselves that they can learn each other something. They can also teach our audience, because aside from being, well, the home of our microbes, the lab is also part of the museum in our educative program, because 2 or 3 times a day we have our so-called lab talk, where our lab analysts are set free into the world. They come out into the museum and they talk to our audience. They tell a story about a certain subject, but they also interact with them, because that's often the first time that normal people, so our visitors, can interact with white lab coats and ask them questions. Because why are you wearing a white lab coat? What's your favorite microbe? And that makes it a unique place inside this unique museum. So maybe if you're in the neighborhood, come by and say hello to our lab technicians. Bye.
Okay, Jasper, thank you very much for your contribution. So getting back to the museum now, and we continue our trip here along the wall of petri dishes, beautiful forms of art in itself, and it also really gives you this insight that on all kinds of objects in our daily life, microorganisms are present. And I think what is important here to tell you is that our main mission is that those microorganisms are in general not harmful. So we know the microbial world in general very well from, for example, infectious diseases or from microbes that spoil our food. But what we want to convey here is a completely different message, that of course those microorganisms are there, but they are the exceptions. What we do see in our daily lives is that microorganisms, for example, in our body, are a very important and integrated part of human physiology, and they play an essential role in maintenance of our health. So while we have been focusing on these exceptions and giving the microorganisms, so to speak, a quite bad reputation, what we really need is to turn this around and consider them and also their enormous potential in order to improve our health and also the environmental challenges that we are facing on planet Earth. I think also there we have an enormous potential, and we have not been using that sufficiently to today. But of course, there are pathogens. Nobody can deny that. And we want to also give some information about them in the museum. So therefore, we also needed to find alternative ways to show them. And for example, we did this by glass sculptures of the viruses we obviously cannot keep in the museum. So over here, you see the HIV virus, and it's kind of difficult to see, but here on the right, you have the example of the Ebola virus, also a beautiful glass sculpture. Then we want to of course, also pay some attention to all these different applications and all the fun projects you can do with microorganisms. For example, here you do see an artist who was able to grow mycelium in all kinds of shapes. And in all kinds of different applications, like for example here, you see that he created some kind of vase, but there is even furniture. Really, the sky is the limit when it comes to all these different applications you can use with simply grown fungi in the form of mycelium. So we had a special exhibition here in the museum about the work of this very innovative work of this artist. Another aspect of the museum that we considered a lot is that the information needs to be layered. And what I mean by that is that it needs to be accessible to all kinds of target groups who have all kinds of different levels. So from the last classes of the primary school all the way to the level of students. So therefore, we also considered a way to collect your microorganisms in the museum by a stamp. And that's, of course, a lot of fun, but you can go around the museum and collect your favorite microbes. And then as a next step, you can actually go to a device here. It's actually right in front of me, but you see this the same level here behind me. And this allows you to upload the microorganisms you have collected in the museum. In general, this is, of course, all about education. So Micropia developed a large number of different education packages for pupils from the primary as well as high school. And by doing so, they are kind of prepared when they enter the museum and learn a lot more by the classes they took before entering the museum. You see them, for example, here at the Petri dish wall. Not only on the level of high school, but also on the academic level for students. We have a number of lectures here. So I'm giving here the annual lecture on the human microbiome and health and disease. And for this, we also have a beautiful lecture hall, which is right here in the center of Artis. And the students are attending the lectures here, and of course combine that with a visit to the museum. Well, in the beginning of my talk, I told you that Micropia is much more than a museum, and it's in fact also a platform. So we developed all kinds of different ways to bring microorganisms under the attention of a much larger audience. So one of the things that is also connected to that lecture series is the annual Microbiome Award. So all the students attending my lectures need to think of a project, an application in which they look for, for example, a medical or industrial application using what they have learned about the human microbiome. And each year, the student with the best idea wins the Microbiome Award of €1,000. And this is, for example, the first edition of this award in 2016, which was won by Sarah Botschuiver. And she developed this very interesting idea about a pacifier with microbes, which allows you to transfer microbes from mother to child in case of a cesarean section. So it's not only the annual Microbiome Award, but we organized a large number of events, which altogether form the outreach platform of Artis Micropia. And this already started with a research which was connected to the opening about intimate kissing. I told you that the idea of the kiss, so the microbial started with a kiss, so we thought it would be really cool to connect this to some research where we invited actually visitors from Artis, on a beautiful summer day to kiss each other, and we monitored the microbial transfer from partners from one to the other. And we determined the complete microbial composition that we found on the tongue and in the saliva before and after the kiss. And this is a so-called analysis where we looked at the similarities of all the data that we collected. And we did a very nice finding that we reported in the journal Microbiome, that actually the oral microbiota on the tongue of individuals that have a relationship is much more similar than unrelated individuals. And also we found a correlation with the number of kisses per week. So if couples kiss more frequently, we found that in the saliva, the microbiota is more similar. Well, of course, this research raised a lot of attention, and it went actually around the world, starting here with a very nice news feature on the BBC. We did not only use this for research, but we also implemented this result in the museum itself by the kissometer. So here you are invited in the museum to enter a stage, kiss your partner, and then the kissometer is able to evaluate your kiss, so to speak, and gives you an impression of the amount of microbes you transferred. So I can all invite you to come and have this analysis going on in the museum. So this research was followed up by another research of microbes from the laboratory I was working in. And it's a completely different topic, but another topic where we could really engage the public in the important work that microbes are doing. And that is actually the topic of fermented foods. Because we all know that for example, the production of wine, beer, bread, And we really wanted to raise additional enthusiasm for this topic and showing the good work of microbes. And therefore we developed in the laboratory a novel starter culture, which allowed you to grow specific bacteria in ingredients like milk. And I'm not sure if you're all aware of this, but this is, for example, an EM picture of yogurt. And you see this is a combination of two bacteria that work together to convert the milk into yogurt. So this is Streptococcus thermophilus and Lactobacillus bulgaricus. And what we did is we in fact constructed a new duo of bacteria that together is also able to make a yogurt. But now in this case, we replaced the Bulgarias by a probiotic bacterium with additional health benefits. So what is more fun to actually apply this starter culture to all kinds of products and organize a dinner. Actually, you see me standing here just on the same place where I'm standing right now, with all people enjoying their variety of fermented foods made with this starter. And it's not only that we could apply this in a restaurant, but we deliberately developed this starter for actually a program where we taught people to start up their own yogurt business in East Africa. And this worked quite well. This initiative that was started about 10 years ago. You see here an orphanage in Uganda where the kids are enjoying this yogurt. And actually over the period of this 10 years, we have now about 300 production units, small companies that all make this probiotic yogurt in 3 East African countries. The next, I just stay a bit with this topic of probiotics, is actually a guide that we developed with researchers and the museum together about probiotics. And we launched this a number of years ago. And the purpose really was to give a much larger audience, the public, an impression of what are actually the health claims related to probiotics. And we did a review where we looked at efficacy of probiotics after the treatment of antibiotics, because there was a lot of unclear about this. So we published that work in the journal BMC Gastroenterology, and allowing people to really have a look at, okay, these are the products that are available on the market, and this is the evidence that is presented in the scientific literature. Likewise, we organized a debate. So we do these, we use this microbiology platform in all kinds of different ways about the hygiene hypothesis. So we invited a lot of experts from the Netherlands and asked them about their opinion about this hypothesis. And then we used a number of statements and see if these experts agreed or disagree with these statements. And the two major, let's say, results from this debate I put here is that, in fact, if we consider this hygiene hypothesis, that is that it's actually not really a good term. The biodiversity hypothesis is a much better term, and there is no clear consensus about effective interventions. And just as an introduction to all of you, I guess most of you are familiar with that, but it's actually the point that exposure to microbes, and about this point all the expert agrees, is very important for the development of our immune systems in early life. So in view of the time, I just want to address one final topic, and that's a more recent research that we started here in last summer, and that's really exciting. It's about the gorillas, where we are looking at the activity of the gut microbes in gorillas after a diet intervention. And the whole point is that if we look at these gorillas, you see that their diet, of course, in the wild and in the zoo is quite different. And we wanted to rewild, so to speak, the gut microbiome of the gorilla by giving them additional branches, because that's what they are consuming a lot in nature, leaves and the bark of branches. And those very difficult and those components are very difficult to digest. So we figured that we would affect, that this definitely would affect the composition, but also the activity. And what we really like about this is that this is, of course, giving a beautiful connection of what's going on in the zoo and microbia. But even for humans, this is relevant, because this is really about introducing more fibers into our diet. And that's a very actual topic for humans as well, because we also see that going from, let's say, a lifestyle of hunter-gatherers to what we are now doing now in the city, completely different lifestyle devoid of microbes. It's important to really go back to some of the ancient habits in terms of diet and see if we can improve our health by that. Okay, I would like to thank you very much for your attention. I hope you enjoyed this talk, and I, of course, hope that I will be able to meet you very soon here in Micropia. Thanks a lot.
Is there life on your nose? Sure there is. Your nose is home to a lot of creatures. Microbes. They live on your body and in the rest of the world. On your phone, for example, but also 5 kilometers underground. There are many, many microbes. If all the people on Earth could fit into a teacup, you would need a container to fit all the microbes in the world. Microbes can do many amazing things. They can create whole entire families in under 1 minute. They can live in boiling water. And they eat all kinds of things like metal, and in some cases even oil. Microbes. They are the living creatures that digest your food. In case you thought you did this on your own. Without microbes, we wouldn't be here. In fact, we can't live without them. Without microbes, we wouldn't have anything to eat or any oxygen to breathe. So we're happy to have them around. Microbes are fantastic. Meet your microbes in Micropia, Amsterdam.
Okay, so we are back with Jasper. I thank Professor Remco Kort for giving us a great presentation. And we have 5 minutes for questions. And I've already written down 10 questions for you, Jasper, but we can't possibly address them all in this short 5 minutes. So I'll get a head start and ask you the first question, which is, what ages do you see visiting most at Micropia?
Well, we said Micropia should be available to anyone, and what we see now is that really everyone visits Micropia, from really early ages to really old people, but also different levels of interest. So people that have no clue of what microbes are, expecting, of course, some kind of horror show because of all those disgusting and dirty bacteria, and of course, well, really getting to know themselves differently when they leave Micropia, to lab teams of microbiologists that are really happy that they finally have a museum of their own.
Great. And we received lots of questions about your collection and how do you maintain all of this. But I think the most popular one was, how do you collect all the samples that you have in the museum? Do you actually go out and collect them from the Netherlands? Do you receive them from abroad? What happens there?
Well, in terms of our collection, our platform function is also very important. So a lot of what we have, we got donated by research groups, for instance, that have been doing research on the specific strain of bacteria or fungus or whatever for a long time and have donated the strain to Micropia as well. But in other cases, if we want to show more about the, the microbe life in a ditch, then of course we go out into a Dutch ditch to find whatever microbes we can find there. So it's a broad collection, but it also has a broad amount of origins. Yeah.
Okay, and I think let's do one last one. So which one is the number one question people ask you guys when they visit Micropia?
Well, I think it's two questions really. One of them is, are there so many microbes in me as well? And that really shows a lot of people really think of something like microbes, something far away from their day-to-day lives. And then the second question they often ask us is, when they've seen the water bear. So the tardigrade, does the tardigrade live in me as well? Because then, of course, they think, oh, the tardigrade looks pretty cool. Maybe that's one of my microbes, and then I can tell something, well, very interesting at the next, I don't know, holiday or whatever family meeting. So it really focuses on themselves, and that's also what Micropia is about. Like Remco said, we really want to show people that they are themselves really more microbe than man, which is important because you really have to love your microbes and take care of them to also take care of your own health and your own future.
Mm-hmm. Yeah, great. Thank you for that. I think we need to wrap up because we have, it's 1 o'clock, so we need to hand on to the next speakers. I just want to say thank you to you, Jasper, and to everyone in the team in Micropia that worked to create this nice video tour for us. And thank you to everyone that has submitted the questions. And if you happen to pass by Amsterdam, make sure you visit the Micropia Museum because it's going to be loads of new bacteria, viruses, fungi join your collection every week, I guess. So even if you have already visited, do make sure to go again because there's always new stuff to see.
Thank you so much.
Thank you,
and
Jasper.
Enjoy the rest of the day. Thanks so much.
Bye.
Bye-bye.
So I'll hand now the microphone to Joe, who's going to introduce us to the next speakers. And I believe we have several science talks by people from all over the world. So, Joe.
Thanks, Eleni. Yeah, I'll just admit our next speaker into the room. So we have 3 speakers for this session, and they're scientists from all across the world giving different talks about different aspects of microbiology. So we'll just wait for Rajira Balasuria to join us, who is our first speaker. And I believe she's just here. Yeah, wonderful. Let me just bring you on. Hey, can we hear you is the first question I have.
Hello, Joseph.
Yes, we can.
Happy International Microorganism Day.
Yeah, fantastic to have you here. You're a senior lecturer from the Department of Biotechnology at Yamba University of Sri Lanka. So thank you for joining us. And to avoid delaying anybody, I'm just going to let you get on with your talk, which I believe is on microalgae and the solutions they have, or the roles they play for sustainability and in climate change. Is that correct?
Yeah, thank you so much for joining me in here. And it's
we'll just check.
Yeah.
Amazing. We'll just check you can share your screen, and then as soon as that's up and active, I'll hop off out of the way.
Okay. I'm sharing my screen. Can you see that?
Just loading. Oh, yeah, we can. Okay, brilliant. So I'll get out of the way and you can commence with your talk. Thanks for joining us today.
Right. Okay. Good morning to all of you. Good afternoon. Good evening for all the participants for International Microorganism Day from all around the world. I wish you all a great Microorganism Day. I thank the Federation of European Microbiological Societies and the Portuguese Microbiological Society for giving me this opportunity to deliver a talk on today for the wider participants, including the school students. So I'm Vajira Balasuria from the Department of Biotechnology, Yamba University of Sri Lanka. If you don't know Sri Lanka, so you can see our tiny island next to my photograph here. Maybe you are interested in knowing about my academic background because I'm making a talk on microalgae today. I obtained my basic degree in agriculture and master degree in agriculture from a university in Sri Lanka, the leading university, University of Peradeniya. And then I obtained a master and my PhD in Ghent University of Belgium, which is in Europe. And then I have 2 short-term postdoctoral research periods at the Argonne National Laboratory and University of Winnipeg, Canada. My research focus, as you can see here, are microbiota in the environment and their applications in soil fertility management, environmental biotechnology, and others. Recently, I have developed a great interest on soil and aquatic microalgae as biofertilizers for paddy and for wastewater treatment. That's why I have selected this topic for today to give you awareness on this very important group of microorganisms. The content will be like this: what are algae and microalgae, diversity of microalgae, why we should appreciate microalgae, role of microalgae in mitigation of carbon dioxide, role of microalgae in the bioeconomy, role in wastewater treatment and agriculture, and then I will come to the question that whether algae could save the world in the energy crisis. So what are algae? Algae, the word originates from the Latin word alga alligata, meaning seaweed. So algae thrive in both saline water and environments, ocean and lagoons and freshwater environments such as lakes, ponds, and rivers. Usually people have bad experiences with algae. Therefore, algae are perceived as bad or nuisance. That's why they are named seaweed, pond scum, and algal bloom. Most of the people are not aware of the importance of algae unless they have studied or have taken a short course on phycology, which is the study of algae. But by my talk, I'll show you that we need to appreciate algae for the services they have done and are continuing to do. Let's see what are they. What are algae and microalgae? Algae are not identified as a consistent taxonomic group, but they constitute of several divisions or phyla which share common characteristics such as aquatic, photosynthetic, no vascular tissues in simple vegetative structures, and simple reproductive bodies. Based on the cellular morphology and size, algae are classified as microalgae and macroalgae. Multicellular larger algae, which are visible with the naked eye, are termed macroalgae, while microscopic, smaller, single-cell organisms are named microalgae. That are the focus for my talk today. The latter generally includes microalgae, includes prokaryotic cyanobacteria, blue-green algae, and eukaryotic microalgae, including green algae, red algae, and diatoms. Why we should appreciate microalgae? Around 3.5 billion ago, the earth was not like this. There was no adequate oxygen for the plants to grow. First, microbes on the earth were anaerobic. Later, blue-green algae or cyanobacteria have evolved. They were able to photosynthesize using carbon dioxide and light energy. Oxygen was released as a byproduct of this reaction. With time, Earth's atmosphere was filled with oxygen so that eukaryotic microbes and plants and other animals like the human could live on Earth. Therefore, we owe very much to these blue-green algae that they are the reason why we as humans are alive on Earth. At present, also microalgae in marine and aquatic environments are producing lots of oxygen, half of the oxygen on the Earth, while the total biomass of algae is only the 10th of the biomass of other plants, which are responsible for producing half of the oxygen on Earth. So we should appreciate microalgae. Not only this, let's see other ones. Here you can see the stromatolites. These are the earliest blue-green algae found on Earth. They are found at various locations as fossils and in few places as living systems at present. Stromatolites are layered biochemical structures formed in shallow water by the trapping, binding, and cementation of sedimentary grains of microbial biofilms, mainly cyanobacteria. They exhibit a variety of forms and structures like these ones you can see. Have different morphologies: conical form, straight form, domal, columnar, and branching types. So these are the earliest blue-green algae. Next to the oxygenation, microalgae are very much important as the primary producers. We are dependent on the primary producers. What are these primary producers? They are the organisms who will produce food for us. Microalgae as primary producers use carbon dioxide or dissolved carbon dioxide in aquatic environments for photosynthesis using light energy, and they become the key source of food and energy for other organisms in aquatic ecosystems. And we obtain food from aquatic ecosystems. In the ocean environment as well, the unicellular microalgae, including cyanobacteria, generally known as phytoplanktons, are the major primary producers in the food web. These are usually invisible, but contribute for half of the annual global primary production, which is a large amount of carbon. And this is an equal amount produced by all the vascular plants on the land environment, such as the forest. Here you can see a marine picture, which is we cannot see the phytoplanktons, and on the above picture you can see how the phytoplanktons are spread throughout the oceans on the earth. In this small video, I hope you can see the video. Maybe it will take time. So this animation indicates the diversity and abundance of phytoplanktons in the oceans, but most of them are not visible unless they form a bloom. So they are fascinating to see, but not visible to the naked eye. And what is the role of microalgae for mitigation of global warming? What is global warming? Global warming is one of the great issues on the planet due to rising greenhouse gases as carbon dioxide. The phytoplankton play a major role in the regulation of atmospheric carbon dioxide levels by sequestration of carbon dioxide. 1/5 of the net primary production by planktons is moved to the ocean interior as sinking particles or dissolved material. Large fraction, 99% of the organic matter which have been sinked are mineralized to form inorganic carbon, which enter deep ocean circulation that will take 100 to 1,000 years to recycle. Thus, the phytoplankton act as a biological pump to transport atmospheric carbon dioxide to the deep ocean, where it is sequestered for long times. And that mechanism is more efficient than the carbon dioxide capture by the terrestrial plants. What are these microalgae? Are there different types? Of course, there are many types. Algae are not identified as a consistent taxonomic group, but they constitute of several divisions or phyla which share common characteristics. These are such phyla: red algae, green algae, brown algae, diatoms, haptophyte algae, dinoflagellates, euglenoids, likewise including blue-green algae. But phycology is this study about algae which dates back to late 18th century by identifying and naming a kelp species, Fucus maximus, by Swedish explorer Per Osbeck. He is an associate of the father of taxonomy, Carolus Linnaeus. However, at present, there are agreements and disagreements among phycologists on the classification of different taxonomic groups as algae. The commonly recognized ones are the ones I have shown you, but there are some agreements and disagreements. This picture again shows you the great diversity of microalgae. I have not named them, but there are vast variety of microalgae according to their morphology and functions, and also based on their ecology or where they grow. There are There are an estimated several 100,000 of algae species. About 10,000 have been classified so far. Currently, about 20 microalgae are used for economic purposes. Here you can see major growth forms of cyanobacteria. They can be growing as unicellular ones, colonial ones, and unbranched filamentous ones, and branched filamentous ones. These are some of the pictures that I study from my studies about unicellular cyanobacteria from paddy fields in Sri Lanka. And these are the filamentous cyanobacteria we have found from our paddy fields. So by knowing the different types of microalgae, what are the roles of microalgae? I have already shown you two, and there's a big role for microalgae in the bioeconomy. Microalgae are used for extracting different food and feed for animal and pharmaceutical products for health sector, cosmetic products for cosmetic sector, biofuel for the energy sector, fertilizers for agriculture, and they are used for wastewater treatment. And not only the food and other chemicals that I have mentioned, they are taken for biofibers, chemical industry, lots of applications are there. Let's see the major ones. So, food ingredients and healthy foods. Microalgae possess high nutritional value containing high proteins, lipids, and antioxidants. They are used as whole organisms or they are used for extractions. Examples are green algae, blue-green algae, and some of the bioproducts are as you can see here. You can see two pictures of cultivating spirulina on the right side, and at the left side, cultivation of chlorella in photobioreactors at the left side. So there are different systems of microalgae growing. For growing microalgae, open pond systems, closed photobioreactor systems, and environmentally controlled systems as well. As you can see here, there are different products have been produced, value-added products in different shapes and forms, including chlorella chewing gum. And there are different cosmetics products as well, like you can see here, different species like Duonalia salina, Hematococcus pluvialis are used for producing different cosmetic products. And not only that, microalgae are used for producing bioplastics and carbon fibers too. And they're used for wastewater treatment, as I have mentioned. Globally, our economic and societal development has led to increased water demand along with water deficit. There's a big demand for having recycled the wastewater because we will have a water shortage in the future. Therefore, it is crucial to select wastewater treated technology that could serve the dual purpose of effective purification and resource recovery. So there are some drawbacks in existing systems, and we can use microalgae for wastewater treatment. Microalgae can utilize solar energy for their growth, and they can simultaneously remove pollutants from the wastewater. They can grow in the arid environments and high saline water as well. They are more efficient than the existing systems because they do not require energy as in the existing activated sludge wastewater treatment system. And the harvested products from the wastewater can be utilized for some other purposes like extraction of fish and animal feed, biofertilizers, likewise. And here you can see a wastewater treatment plant which are applying microalgae. And in the agriculture, microalgae are mainly used as biofertilizers. For example, free-living cyanobacteria and Azolla anabinae are used as biofertilizers. And I have also tried to produce biofertilizer for paddy cultivation in Sri Lanka with some success. And then my next major question, could microalgae save the world? Energy crisis is the greatest problem we are facing in the world. The major gas and oil deposits are so rapidly depleting because of our utilization. These deposits also came largely from algae deposits that we don't know. And now it is essential to find alternative energy sources for a sustainable future. Microalgae have potential to produce different kinds of bioenergy: biodiesel, bioethanol, methane, biohydrogen, and bioelectricity. I'm not going to describe you all these in detail. However, now there are places called microalgal biorefineries, which allow the transformation of biomass into the production of biofuel, food, feed, chemicals, polymers, and value-added ingredients. So the microbes will be cultivated in the biorefineries, and then harvest will be extracted in the refinery. Other products will be taken, including the bioenergy. Why microalgae are best as future fuel? They do not require cultivable land and fresh water for cultivation. They can be grown quickly with minimum requirements irrespective to seasonal conditions. Microalgae can feed on industrial waste, especially wastewater. They use CO2. They do not need energy. Biomass productivity of the algae is 10 times as compared to crops and other plants, and it could be cultivated in arid temperatures like deserts and in brackish or salt water. So bioenergy is another gift of the algae to humankind. Here you can see two large biorefineries in the world, and there are many algal biofuel producing companies in countries across the world. I think in Portuguese or Spain also is available, and you can see such companies using this link. With that, I will sum up my talk. So as a summary, there are multiple roles of microalgae for which we should appreciate them. They have oxygenated the earth, and they are continuing to do so. They are the key primary producers in aquatic environment. They support for mitigation of global warming to reduce carbon dioxide levels. They produce different bioproducts, and they are useful in wastewater treatment, and they are very much useful to save us to find a good bioenergy. With that, thank you very much, and I wish to especially thank Joseph Shalworth and the team who is organizing International Microorganism Day. And for the presentation, I especially thank my research students, including Ms. Tilini Dinushka, who supported me preparing these nice slides. Thank you very much.
Thank you for giving us a fantastic overview of all the wonderful things microalgae do. They seem to touch on every aspect of life and are thoroughly fascinating. Thank you for being our first live speaker on International Microorganism Day. I'm going to swiftly hand over to Ben, who's going to play the video of our next speaker. But I want to thank you, Ajira, for joining us. Thank you for a very interesting talk. You will be able to catch this as a recording later on. And yeah, thank you for being our first live speaker. It's really an honor.
Yeah, thank you so much. Have a nice International Microorganism Day.
We will, you too. And I hope you have some celebrations in Sri Lanka across today and tomorrow.
Yeah, I think so. And I'm from Sri Lanka.
Thank you very much. Have a good day.
Hello everyone, my name is Dr. Dipesh Kumar Nilam. I am Assistant Professor at Department of Microbiology at JECRC University, Jaipur. Today I will talk on that nothing is impossible for the extremophilic microorganisms. So before I'm going to discuss in detail, we have to know about what are the extremophiles. So extremophiles are the remarkable microorganisms that are thrive in the harsh environmental conditions which are present on the earth. The word extremophile is originated from the Greek word that means extremus plus philia, means those who love the extreme environment. Later on, the term is coined by the Microorganism in 1974, and it is also known that the most of the extremophiles are archaea, but can also include the bacteria and eukaryota. But there is a question is arises why we have need of these extremophiles. So according to the astrobiology, extremophiles are those microorganisms which are able to grow at the higher extreme conditions. So on the basis of these extremophiles considered as a model organisms when they're exploring the existence of the extraterrestrial life out of the earth. Second thing is only the limited research studies have been conducted on them, so we have to know about more for the unexplored diversity by using the various culturable or unculturable techniques. Then the extremophiles can be a valuable resources for the novel biotechnological and biomedical products due to their biosynthetic properties. For example, mesophilic enzymes are not well suited for the harsh reaction conditions that required in the industrial processes because the lack of enzyme stability at the various conditions like higher temperature, lower temperature, higher salinity, and high acidic conditions. So at this stage, the extremophiles bioactive products could be the potential source to fulfill such requirements. So here it is the question is arises, what kind of the extreme conditions are there where the extremophiles can be grow? So there is the some extremophilic conditions are present on there. The extremophiles can be grow. For example, like the higher temperature, lower temperature, high saline condition, high alkaline condition, acidic conditions, pressure, and the other some specific dry conditions like heavy metals and concentration and the UV radiations. So such kind of things are also found in the entire world. For example, the Yellowstone National Park or Thar Desert is especially known for their high temperature. And if you are talking about the lower temperature, there will be found in the glaciers and the oceans. Similarly, the saline and alkaline condition will be found in the Sambar Lake, Great Salt Lake, and Bonar Lake. So based on these extreme conditions, in this my presentation, I will discuss only the important classes of the extremophiles. For example, number one is the psychrophiles. Psychrophiles are those microorganisms which are cold-loving and growing optimally at the 15 degrees Celsius, and their growth range is -2 to 20 degrees Celsius. And there is some cyclotolerant like pseudomonas which are able to grow at 4 to 35 degrees Celsius. And if we are talking about their habitat, these psychrophiles grow at the polar oceans, glaciers, Antarctic soils, and the marine environment, etc. But here is how they can survive. There is just some mechanism behind that. On the basis of them, they can survive in this such kind of lower conditions, which are anti-freezing proteins, cold stalk proteins, membrane fluidity by the unsaturated fatty acid, and some exopolysaccharides. And if you are talking about some examples like the Cyclobacter aquaticus, Pseudomonas antarctica, and Locaria polaris, these are the examples. There is another class is acidophiles. Microbes, those are living and they grow at the very lower condition of the pH, like 1 to 5 pH. That is the very extreme acidic condition for the survival of any other microorganisms. So such kind of microbes are known as the acidophiles, which are able to grow at the acidic conditions. And where these conditions we can find out in the such like the acidic river, acidic hot springs, and the various mines, there is the sulfur compound common, etc. And if you are talking about their adaptation, the sugar coating and the higher content of the glycoproteins help to maintain their high relative internal pH during the survival in acidic environment. And if you are talking about the examples, Sulfolobus acidochlarideus and the Hydrogenobaculum acidophyllum are the some examples of these extremophiles. And the another type of the extremophile class is halophiles. Halophiles means salt-loving microorganisms which are able to grow at 0.2 to 5 molar NaCl concentration. That means 1 to 30% NaCl concentration. That is the very extreme conditions. There is the nothing is the another life is can grow. So such kind of habitat are found in the hypersaline conditions like the Great Salt Lake, Dead Sea, Sambar Lake, etc. And what mechanism they follows for their growth, that is the salt in and salt out process. And second one is the organic compatible solutes. So there is some these kind of conditions they maintain by the some organic solutes which are like glycol, pyamol, etc. These are the organic content which maintain their higher concentration of KCl ions by using the organic compounds. And if you are talking about some examples like the halobacterium, haloprex, volcani, chlorobium, imiricola, and disulfovibrio halophilus are the very important examples of the halophilic bacterial classes. There is the another class, that is the alkylophiles. Alkylophiles are those microorganisms which are able to grow at the higher pH. Means most of the alkylophiles are able to grow at the 9 pH, but there is some extreme alkylophiles also present there. These are generally grow at the 10 pH or above. And these microorganisms are found in the alkaline environment like the Mono Lake, Lonar Crater Soda Lake, and Lake of Magdigi, etc. And if you are talking about their adaptation, they can maintain their low cytoplasmic pH than the external pH value by the coordination of hydrogen ion and sodium ion pumps in their cell. So these are some unique characteristics of these halophilic classes and the other microorganisms which are able to survive in such kind of extreme conditions. And if you are talking about the example, the Rhodospira, Halochloris, Netormonas species, and the Thiohalospira and Alkylophylla are some examples of alkylophilic microorganisms. There is another classes of the extremophiles like barophiles. Barophiles means those microorganisms which are able to survive at the higher pressure. That stage, there is the normal microbial cells cannot be survived. They immediately rupture at that atmospheric pressure, that is the 100 to 700 atm. So such kind of habitat where we can find deep in the earth and deep sea hydrothermal vents. These are some specific habitats where we can find these barophilic microorganisms. And if you are talking about the adaptation, the lipid unsaturated fatty acids like PUFA, the protein coat at the outer cell membrane proteins, enzymes, and the pressure controlling genes are responsible for their survival at the high conditions. We are talking about their examples, the Proteobacteria gamma and Halomonas cellular are the examples. There is another important class is radioresistor. Radioresistors are those microorganisms which are able to survive or they can tolerate the higher radiation at the various environments. For example, if you are talking about the UV radiation, there is one important bacteria is present that is known as the Deinococcus radiodurans, which is also known as the hardest bacteria which can be survived at the higher UV radiation concentration or higher UV radiation. So here is the point is how this bacteria can be survived at the higher UV radiation condition. So there is some DNA repair enzymes are present like the REC-A protein and which are able to matches the broken pieces of the DNA and splices them back together in the few hours. So at this stage, I want to tell you that there is another important class is present, that is the thermophiles. Thermophiles are those microorganisms which are able to grow at the higher temperature, and such kind of thermophilic bacterial class is not only important for the research purposes, but also these are important for the industrial purposes in the various industries like in the food industry, dairy industry, and the leather industry, and the enzyme industries. So what are the specific habitats there thermophilic bacteria can be found? These are hot springs, deep sea thermal vents, desert, compost, manure, and oil wells, and the laundry, etc. These are the such habitats. There we can easily find out the thermophilic microorganisms. So here we have to know about that what was the first bacteria who was isolated in the first time. So Thomas Brock and their colleagues in the late 1960s, discovered the first extremophilic bacteria which is capable at survival at the 70 degrees Celsius, which is isolated from the Yellowstone volcanic hot spring that is known as the Thermos aquaticus. And naturally, it is a source of tag polymerase enzyme, which is used in the polymerase chain reaction for the amplification of DNA. Then the Kesafi and Lovely in 2003, they isolated the Archaeobacterial strand that is 1-to-1, that is 1-to-1, which is known as Geogamma grossi, and isolated from the Mothara hydrothermal vent. This strand is able to survive at the 121 degree Celsius. So these thermophilic bacteria further classified into the 3 types. Number 1 is the moderate thermophilic bacteria, extremophilic thermophilic bacteria, and the hyperthermophilic bacteria based on their survival temperature. So moderate thermophilic bacteria are able to survive at the 50 to 60 degrees Celsius, and the extremophilic bacteria are able to survive at the 65 to 80 degrees Celsius, and the hyperthermophilic bacteria are able to survive 100 to 122 degrees Celsius. And for if we are talking about the examples like the Bacillus coagulans, Geobacillus stereothermophilus, Clostridium thermoaceticum, and Thermus disulfovibrio, Thermoanaerobacter, and Sulfolovas, Thermovoccus. These are the very important examples of the thermophilic bacteria. So now here is the question arises, how these thermophilic bacterial different classes can survive in the higher temperature? Because this is the very important class, so there is the various molecules are responsible for their survival in the higher conditions of the temperature. For example, the mechanism, if we are talking about the mechanism which is responsible for their adaptation in the higher temperature, the membrane lipid have a ether linkage. That means more branched and more stable. So there is the higher molecular weight. So these characteristics increase the melting temperature of the membrane lipids at the higher temperature conditions. Second is the heat shock proteins. More hydrophobic interior prevent the unfolding or denaturation of DNA during the higher temperature. And another important point is the high guanine and cytosine content in the nucleic acid structure. And there is the another important point is the reverse gyrase enzymes. These enzymes help in the catalysis the positive supercoiling of the closed circular DNA, means the positive supercoiled DNA appear to the resistance degradation more than the negatively supercoiled DNA. So there is the very important function of the reverse gyrase enzymes to catalyze the supercoiling of the DNA at the stage of the supercoiling. So there is the another thing is very important by the side the reverse gyrase, that is the DNA binding histone proteins or histone-like proteins which are able to make the strong binding in the DNA molecules. And there is the another some potassium and magnesium ions we are working for the higher temperature in archaea protect the standard DNA from the phosphodiester bond degradation. So based on this adaptation or based on these conditions, we know that the extremophilic classes of the various extremophiles, thermophilic microorganisms are more diverse and they are more attractive in the field of research as well as in the industries. So here I will tell you about why the extremophilic microorganisms are more emphasized for their enzymes. Generally, extremogens are those enzymes which are produced by the extremophilic microorganisms. So here we can say that the discovery of new thermophilic microorganisms and their enzymes has a great impact on the field of biocatalysis, because the extremogens from the extremophilic microorganisms are more active and more stable at the higher and the various different stress conditions. And these extremogens are able to replace the uses of chemicals from the various industries of the environment, and they are the environment friendly and sustainable for the new generation of industries. In particularly in the extremogens of the various classes, thermophilic microorganisms have found the most practical commercial uses to date because their overall inherent stability at the higher temperature, as well as they have some another characteristics like they are also able to grow at the higher pH. So they have multiple characteristics to survive and able to produce some enzymes. Those are working at the not only for a single specific parameter, but also they are able to stable at the multiple parameters. So here is in this table, you can see that there is the various exophilic bacterial classes which are able to produce various enzymes. If we are talking about the psychophiles, acidophiles, alkalophiles, halophiles, and pyrophiles or piezophils, these are able to produce the various enzymes. But if you will look at the end, there is some thermophilic microorganisms. These are able to produce more than 4 or 5 enzymes. So this class is very, very important in the industrial purposes, in the various industries like the food industry, like the dairy industry, like in the detergent industry and the starch industry, and the pharmaceutical companies, or for the medical purposes and for the removal of toxic compounds. So here is the important thing is why the extremophilic microorganisms are more important for the extremogens, because thermophilic extremophiles have attracted the more attention, in particularly for the extremophilic enzymes such as protease, lipase, cellulase, chitinase, and amylase have found their way into the industrial applications, as I have told you in my earlier slide. Then there is another reason. These thermophilic bacteria are have some reasons to stability at the higher temperature, but they have some more reasons, like they have to avoid the contamination leading to understand the complications to reduce at the higher temperature. So these are very important class of the extremophiles, which is not only for able to produce the thermophilic enzymes, but also there is another microbial life cannot be exist, or they cannot be able to do some activity at the enzyme level. So the usage of such enzymes in the maximizing reaction accomplished in the food and paper industry, detergent industry, drug, toxic waste, and removal and trailing of the oil is being studied extensively. So based on these things, Akhil Agarwal sir, and during my study PhD research work, we found that various thermophilic microorganisms which are able to produce not only a single extremophilic enzyme, but also they are produced the maximum various industrial important enzymes. Those are capable to work at the industrial level. In this figure, you can in this slide, you can see there is the one strand that is able to produce the alpha amylase, which is used in the starch industry. And if you can see in the B figure, there is the protease enzyme producing thermophilic bacteria. Those are used in the detergent industries and the protein industries. And if you can see, there is the C in the C figure, there is, you can easily observe the hydrolysis zone surrounding the bacterial strain. That means these are able to produce the cellulase enzyme. And if you can see in the D figure, there is the esterase enzyme, which is produced by the thermophilic bacterial strain to segregate some content related by the twin AT. And in the figure D, you can see there is clearly the red color zone indicate the activity of L-asparginase enzyme. And at the end, there is in F figure, you can see there is the anurelinase enzyme production by the bacterial thermophilic strain. Here, I will not disclose the name of strains because this data is unpublished. So I am not able to represent the name of these thermophilic microorganisms. Now, these are some qualitative analysis of the extremophilic enzymes. But in the industries, I want to tell you that these are not required the 100% purification of enzymes. Generally, in the industries, they only want the semi-partial purification of the enzymes. But in some cases, if we are talking about for the pharmaceutical industries, or for example, in the medicines, or if we are talking about the particular any research, there is a need of the purification of enzymes, but industry doesn't want to waste their time for the complete purification of these enzymes. So on the basis of based on this research work, here I will close this presentation, and I'm very happy to tell you that you are now at this stage. You can know about what are the extremophiles and where they are live and how they can survive, and you know very well now about the extremo enzymes, how they are great economic potential in the many industrial processes, including the food, agriculture, and chemical and the pharmaceutical applications. But still, we have more need to understand about the extremophiles, because till to date, only the less than 1% of the studies has been done in the various extreme conditions for the extremophilic microorganisms. If we are talking about some metagenomic work, through that we can identify there is the various microorganisms are present in that particular natural sample. Those are able to show the genes regarding the extremogynes, but there is some lack of cultivation techniques present till to date. We have, on the basis of them, we cannot able to grow these microorganisms into the in vitro conditions. But now I can say only one thing: in the upcoming future time, we can able to grow these such kind of microorganisms into our lab. So with this slide, I will stop my content regarding the extremophilic microorganisms and their applications. So this is the Department of Microbiology, Dr. Varsha Gupta, Associate Professor and Head of the Department in our JCRC University, and Ms. Devki Sen and Dr. Ravi Kant Rai are my colleagues. And here is some references which I have added here to know from where I have used this content for my slides. Thank you very much for listening me patiently. If you have any query, please write in the text box. Thank you.
Thank you to Deepesh Neelam, Assistant Professor of the Department of Microbiology at JECRC University in India, for submitting that pre-recorded talk. Just as we wait for our next speakers, I just wanted to ask Wajira if you could come back on camera, because we have some questions for you from the YouTube stream, and we'd love to chat about microalgae again. So if we can bring you back online. But in the meantime, here we go. Wonderful. Let me just do one small thing here. Yeah, so I'll pass the microphone over to Eleni so she can ask the question from the YouTube.
So we had one question that says, do you foresee microalgae as a possible solution for the world feeding problem?
Yeah, the feeding problem, maybe we need some clarification. Is it animal feed or food or?
I guess for general food, the question, human food, yeah.
Yeah, microalgae cannot replace all the human food, in my understanding, but microalgae are the source of human food in aquatic systems. They are the primary producers. So then all the food coming out of the aquatic systems, oceans, rivers, lakes, the fish that we are eating coming, the source is microalgae. But if you are going to increase the production of aquatic life, then we can increase the growth of microalgae, and then by that way we can have a solution for the feeding problem in the world. But as I have shown you in my presentation, there are some nutrient-rich food, healthy food, which are coming directly out of microalgae like spirulina and chlorella. I hope it is sufficient.
Okay, so it is the source of all our food one way or another. But you showed us in your talk some delicious microalgae based snacks and food. I think you had a slide, and I just wanted, have you eaten any of those? Could you give us a bit of a breakdown of which of these are the tastiest and which ones you've tried and which one we should eat if we could get our hands on these microalgae food?
Unfortunately, I could not eat them actually. I also didn't have the experience yet, but I have seen spirulina tablets, which can be taken as pills, or even the spirulina mix in the snacks or the cornflakes in the morning. You can mix spirulina powder with cornflake in the morning. And the other ones, actually, I also didn't have experience, but I'm looking forward as you.
Yeah. And do you know what kind of nutritional value it adds? You know, is this a good protein supplement? Is it carbs? Is it just a big mixture of different macromolecules?
It's a good one for the proteins and the good lipids. And they are supplying some carbs as well, the beneficial carbs.
Okay, so it might be quite good for people trying to bulk up in the gym even, because it has protein in there as well. I had one more question for you, which was, we were talking about the role that microalgae plays in climate change, or maybe even in helping us with global warming. Are microalgae, you know, a bigger influence at capturing carbon than all the trees? Like, how do they compare to trees on land? Are they much more capable at, you know, fixing carbon than plant life? Or, you know, do you have any idea what the relative effectiveness of plant life versus microalgae is in carbon fixation?
As a total, they are doing 50-50. 50% of the carbon fixation is coming out of the marine system from the phytoplanktons or microalgae, and the rest 50% is coming out of the forest and or the land plants. However, when we consider the biomass, the phytoplankton biomass is lesser when we compare it with the biomass of the land plants. It's I think it's 1/5 when we compare, and so their mechanism is efficient. The phytoplankton in the oceans, they take dissolved carbon dioxide in water, and the land plants, they sequestrate atmospheric carbon dioxide. The mechanism is more efficient with the phytoplanktons.
Okay, so they're more efficient, they weigh a little bit less, but they capture just as much CO2. So they're pretty good then in terms of, fixing CO2. Are there ways that we could increase the rate at which they, sequester CO2, or ways that we could increase their population, or is it out of our control?
Yeah, we should find ways to increase the population. As I have mentioned, we can couple them with the wastewater treatment. We are producing a huge amount of wastewater, and we are leading to a water deficit as well. Then we should find ways to recycle the wastewater, and use of microalgae will be best way to do that. By that way, they can fix carbon dioxide in the water, because finally we can harvest the algal cells after treating wastewater, and use them in the soil as biofertilizers and other products like animal feed and all. So they will be sequestrated.
So I just wanted to know, in Sri Lanka, where you're based, obviously you've got, the ocean all around you. Do you have quite good access then to phytoplankton and microalgae for your institution? Is it one of the advantages of being based where you are?
Yeah, but although you have, as you have mentioned, we have quite a lot of ocean area in the Indian Ocean. But as far as I know, the studies on phytoplanktons are still developing and very minute. And my department, I have initiated some work on microalgae, but the inland ones, still I had to look for the oceanic phytoplanktons. I did study something about the brackish water in the lagoons, but not in oceans.
So what do you need to go study the phytoplankton in the oceans? Do you need like a big boat and a crew to go out and find them?
Of course, I think there's an aquatic resource research center in Sri Lanka, But I think if I have an interest, then I can join with them for such studies.
Okay, so yeah, you need just some ships and some helpful crew to get out there and take some samples. Okay.
Exciting.
Yeah, very. I mean, that would be a really good trip, just getting out into the ocean to go and get some nice samples of phytoplankton. I'm just going to hand us over in a couple of minutes to the next session, but I just want to say thank you so much for joining all the way from Sri Lanka to us here in The Hague, in the Netherlands. And yeah, that brings us to the end of our science talk session, but it's been fascinating learning about extremophiles and plankton. You know, organisms that do amazing things in so many different strange environments and things that probably people don't think about very often. So it's been brilliant to have you here. And I can only, yeah, and we'll hopefully maybe see you again next year, or drop us an email and perhaps we can try and exchange some samples of phytoplankton and maybe even get something under this microscope one year so we could have a look at them.
It's a nice setup there.
Yeah.
Okay,
well,
bye-bye.
Thanks for joining us in the studio, and enjoy the rest of International Microorganism Day tomorrow. Right, okay. Moving on to our next session, we have up and coming a session on yeasts in bio-based industries put together by the Portuguese Society of Microbiology. We're just going to start this one a little bit early, but that will give people time just to sort of introduce each other and make sure all the streams and presentations are working. So I'll just admit everyone for the next session. I'll be handing over to Claudia Godinho from the Portuguese Society as the host, who will be able to run you through an introduction of our upcoming speakers. But they have arrived in the Zoom room and we will soon be getting them on board. So Claudia, I'm just going to make you a co-host so you can do all the things you need to do to get this session rolling. Okay. Right, Claudia, can we hear you?
Yes, sure, Joe. I'm here.
Ah, brilliant. So we have everyone we need for the session. I'll just hop off the call myself and yeah, let you start giving us some wonderful introductions to yeast in bio-based industries.
Professor Isabel, you are muted.
Thank you, Joe. Oh, we are seeing Irina. Hello, Irina. Irina is arriving. Okay. Okay, hello. Hello. So welcome to this session. Well, it will be yeast in bio-based industries. And is arriving. Okay. Okay, hello. Hello. So okay. Welcome to this I'm sorry. I'm listening. Session. Well, I'm sorry. Okay. In bio-based industries.
You have to shut the window of the live stream of YouTube or Twitter or.
I'm listening. Well, I'm sorry. Okay.
You have to shut the window of the live stream of YouTube or Twitter or.
Yes. Okay.
Have to shut the window of the live stream of YouTube or Twitter.
Yes. Okay. Okay. Now we are in. Okay, so again, welcome to this session in bio-based industries. And you know that baker's yeast is the most popular cell factoring for biotechnology, but well, not only because it has been. Again, welcome to this session in bio-based I'm sorry. Industries. And there is some problem. The most popular cell factoring for biotechnology, but well, not only because it has been used for 1,000 years for producing bread, wine, and beer, but today it can be engineered to produce an array of other useful bioproducts, including biofuels, pharmaceuticals, agrochemicals, fragrances, vitamins, pigments, and so on. All the wastes are easy to manipulate and scale up and have a short generation and production time. Achieving optimal and economically viable production of these products can be difficult. Scientists have uncovered a way to control many genes and complex biochemical networks in metabolic engineered yeast cells using synthetic biology approaches. So this opened the door to more efficient and sustainable production of bio-based products. Among these scientists is Irina Borodina. Well, she is with us to talk on yeast as biotechnology's best friend. Irina is professor in yeast metabolic engineering at the Novo Nordisk Foundation Center for Biosustainability at Technical University of Denmark. Originally trained as a chemical engineer in Lithuania, she holds a PhD degree in biotechnology from DTU, co-inventor of patents and co-founder of a biotech startup company. She was selected in 2019 as an European Union Human Innovator. So, Irina, please. She holds a PhD degree in biotechnology from DTU, co-inventor of patents and co-founder of a biotech.
Thank you so much, Isabel, for the kind introduction, and also thank you very much for invitation to participate in this event. I welcome everybody, all the listeners, and I would like to talk to you today about the yeasts, which I, as biotechnologists, they consider our best friend. And I'll explain to you why. And as Isabel already mentioned, yeasts have been used for millennia for baking, brewing, and wine making well before we knew that actually it was yeast that helping us to do the job. And this is, of course, thanks to the yeast natural properties to convert to ferment sugars and to generate CO2 that helps the bread to rise, and also to produce ethanol and different aromatic compounds that help to give the beer and wine its unique properties. But we can teach yeasts to do completely new stuff thanks to their DNA manipulation technology that has developed in the last 40 years. So they can not longer only make ethanol, they can make a lot of other things such as drugs, materials, food and cosmetic ingredients. And I would like to give you a few examples just to give a taste of what actually yeast can do after it has been engineered. So one of the very interesting products is polylactide, which is a biodegradable plastic. And it's made out of lactic acid. But the interesting thing is that the lactic acid for production of this plastic cannot be made chemically because you need a very pure stereoisomer. So you need L-lactic acid, and it's only microbes that can produce pure L-lactic acid. So normally in nature, it's lactic acid bacteria that do the job, but they are very difficult to ferment at large scale because they require some very special expensive nutrients. So therefore, firstly, bacteria and then yeast have been engineered to produce lactic acid in large fermenters. Yeah, yeast have a huge advantage in comparison to some bacteria such as E. coli because they can tolerate very acidic pH. So that will mean that you can produce lactic acid in acid form and avoid the waste of gypsum, for example, that would be generated if you use E. coli and need to maintain a neutral pH. So nowadays, the polylactide that you can see in the supermarkets and that you can purchase, it's actually produced using yeast. Another example of what yeast can do is the engineering of production of jet fuels, such as farnezen, which was accomplished very successfully by Ameris, where you can actually fly the plane with having, not as bad conscience as usual because their jet fuel would be produced from renewable feedstocks. Another excellent example is production of resveratrol, which was developed by Volva, and this is a particular very strong antioxidant compound, which is normally found in wines, in wine in very small amounts, but you can make it in yeast in quite large quantities, and then you can use it as a food supplement for health, to promote your health. So why would you want to use biotechnology? Why would you want to use biotechnological processes for making chemicals? And there are two main reasons. One reason is because you can reduce the carbon dioxide emissions because you produce this different chemicals using renewable feedstocks. And here a great example is a production of succinic acid. Here is an example from BioAmber that accomplished this process again using a yeast cell. And here they could show that in comparison to petrochemical succinic acid, the yeast produced succinic acid led to 99% reduction of the carbon dioxide emissions. Another reason for using a biotechnological process could be when the compound that you want to produce is non-obtainable from nature or is simply too expensive to extract from nature. So one example is stevia glucosides. So probably many of you have seen on their, for example, bottles of soft drinks that you buy that they might be sweetened with stevia extract. But stevia leaf extract, it mainly contains some stevia glucosides that have a bitter aftertaste. And that's why only a very small fraction or a fraction of their sugar in the soft drink can be replaced with stevia glucoside. However, you can engineer yeast to produce stevia glucosides and only the specific forms that have the sweet taste, but that do not have the bitter aftertaste. And this is a product, one of these products is EverSweet, which is marketed by Cargill. And that's that contains mainly riboside M. So it's stevia glucoside without bitter aftertaste. And using that, you can replace the whole sugar in a soft drink or in another product. You can replace it with stevia glucoside. And so it becomes completely zero calorie product. So these are the two main reasons why you would want to use biotechnology, and that's why it gives you a great advantage. So I have here a little video that I will try to run now, and which explains how we actually can create such yeast cells. So what we do, we metabolically engineer them. So normally, if we look at the yeast cell, it takes up sugar. Here it's illustrated as a yellow compound, and it secretes ethanol. And that's what basically yeast can do naturally very, very excellently. However, we can change this. So we can manipulate its DNA and its enzymatic machinery, because it's the enzymes that are responsible for production of ethanol. So what we can do, we can look at its DNA and we can cut it in specific places, and we can insert new genes that encode for enzymes that are responsible for productions of totally new compounds. So here you can see the DNA is being cut and there is a new gene which is being inserted. And this gene will then get translated to the protein, which is an enzyme in this case. And this enzyme will catalyze a totally new reaction. So you can see the enzyme is appearing here in the cell, and now their precursors, the carbon, is being converted into a completely new molecule. It could be, for example, resveratrol or farnesene or anything else. Normally, when you're going to insert the new biosynthetic pathway, the production efficiency will be quite low. So you need to manipulate the cell further, such as, for example, by deleting the ethanol production so that most of your carbon can be redirected into your favorite product, and so that you create a very, what we call an efficient cell factory. So here you can see the ethanol formation was deleted, and now the cell primarily produces the product that you want. So this redirection of metabolic fluxes, this is what we call metabolic engineering. And this is what allows us to create these different cell factories. So I want to show to you two examples from my laboratory where we created the yeast for production of two high-value compounds. One of them is a nutraceutical compound and another is a potential pharmaceutical compound. So the first compound here is called egothioneine, and it's mainly found in mushrooms. So there are not many other dietary sources of this compound for humans. So why is egothioneine interesting? Well, let's have a look. There are some interesting studies that show that mushrooms and egothioneine, that they promote healthy aging. So the study here on the left, it was performed in Singapore, and it was done on 300 people, where the mushroom consumption was correlated with the odds ratio of developing a mild cognitive impairment. And what was found in the study was that people who consumed more than 5 portions of mushrooms per week, they actually have a significantly reduced odd ratios of developing mild cognitive impairment. So the authors hypothesized that this would be connected to the presence of agathionin in the mushrooms. Though, of course, there could be also other positive compounds with a positive health effects there. Another interesting study was conducted in Sweden, and this was done on 3,000 people, where the concentration of more than 100 metabolites in the blood serum was measured. And then there was a follow-up that was performed 20 years later to see if there was any correlation with the diseases that people have developed with their metabolite concentrations that they had in their blood 20 years ago. And there were several metabolites that showed a very strong correlation, and one of them was agathionin. So the more agathionin was present in the blood, the less was the risk of developing cardiovascular disease or of dying. So there seems to be a clear correlation there that agathionin presence in our body can have some significant health benefits. There was also a large meta review that was published earlier this year, which analyzed multiple articles and found that mushroom consumption was correlated with lower risk of cancer. And again, the authors hypothesized that one of the reasons could be the presence of this essential agathionin in the mushrooms. So if agathionin is really that beneficial, how could we get more of it? So the only source of it is fungi. So you need to eat up to about a kilogram of mushrooms per week in order to get sufficient amounts of agathionin. Another option is to take agathionin supplement, and currently agathionin can only be produced by chemical synthesis, so it is quite expensive. So I think a treatment like a supplement for 1 month costs about 30 euros. So what we can do instead, we can teach yeast to produce agathionin, and in this way we can obtain large amounts of it by fermentation in a rather cost-effective way, and then we can make agathionin much more available to anybody. What does it require? So it requires that we reconstitute their biosynthetic pathway towards agathionin in yeast, because naturally yeast cannot make agathionin. So here you can see the biochemical reactions that lead towards agathionin from their common amino acid precursors such as histidine, cysteine, and methionine. So it was a PhD student in my group, Steven van der Hoek, who has done the job, and he has reconstituted this multiple pathways towards agathionin in yeast. And then during the fermentation, we could obtain about 600 milligram per liter of agathionin, which would lead already to a significant cost reduction, where probably agathionin could be produced at scale at about $1,000 per kilogram instead of $500,000 per kilogram. And this would make it much more available for wider use. And now we have licensed this technology to a biotech company who will hopefully bring it to the market within a few years, so we all can enjoy the health benefits of this molecule. And so another example I want to show you is production of psilocybin, which is an active compound of the psychedelic mushrooms. However, the reason why we wanted to make this compound was not for the recreational use, but it was because it has a great potential for some neurological disorders, particularly, again, seems to be very active against depression, and also could be useful for treatment of addictions such as tobacco or alcohol addictions. So it was Nick Milner, a postdoc from my group, who developed this technology, and now he's a co-founder and CSO of Octarine Bio, who has actually licensed in this technology and continues working on it. So on the right-hand side, you can see their metabolic pathway towards psilocybin, which comprises multiple biosynthetic steps, which Nick has reconstructed in the yeast cell and could achieve their high-level production of psilocybin and also the psilocin. And again, as I said, this technology is now being further developed in Octarine Bio, who will bring it to the market for the benefit of people, of the patients, for treatment of depression and other disorders. So with that, I want to thank my team at the DTU at Denmark Technical University, and also our funding sources. And I will be happy if there are any questions. I will be very happy to answer them. Thank you.
Thank you, Irina, for this for your views on this challenging area. We do not have a question as far as I know, but I think I can ask you a question. Well, it's a difficult one. Well, how do you see the field of synthetic biology of yeast, of Saccharomyces cerevisiae, and well, the relation with not exactly concerning the problems that you brought to us, but concerning others, just the use of non-conventional yeasts.
So recently with the CRISPR particularly a revolution, the access to the other different non-conventional yeasts has become so easy. So if before we could mainly manipulate the Saccharomyces cerevisiae, now we very easily can manipulate a lot of other different yeasts. So I think one of the most popular yeasts now is oleaginous yeast Yarovia lipolytica, which has become, I think, I wouldn't call it anymore non-conventional yeast, because I think it has already made its way all the way to the manufacturing size fermentations. So it's already producing the products which are being marketed. So I think it's not a non-conventional yeast anymore. And I think several other yeasts are going to catch up as well. And more yeast species are going to be used. And I think if we look in general on the tendencies in their synthetic biology and in the metabolic engineering also, so we're moving towards doing more high throughput metabolic engineering, which is again enabled by this better access to their molecular biology tools. But also quicker learning from this metabolic engineering experiments with the help of machine learning, modeling, and other approaches. So I definitely see this tendency towards a much more accelerated development of the yeast cell factories, and this is a big. This will be a big. It's really a big thing in supporting the development of the new processes.
Thank you for your views on the topic. We have questions now. Well, the first question is, should we eat more mushrooms to live longer?
Absolutely. That's more or less the main message of my talk. Eat more mushrooms. I mean, we don't know for sure whether it's agathionine or some other compounds in the mushrooms that give this positive effect. And agathionine, hopefully you will soon be able to buy as a supplement. But what if it's something else in the mushrooms that's also very beneficial? And besides, they're super tasty. So please eat more mushrooms. Yes.
There is another one, but this one should be more difficult, I think. What mushrooms have the most ergothioneine?
Actually, they're ones with the highest
kind of mushroom
are oyster mushrooms. And I think shiitake mushrooms also have a high content. And the interesting thing about oyster mushrooms that they are easily cultivated, and you can cultivate them yourself at home. For example, on the spent coffee grains. So if you drink coffee, which also maybe has some health benefits, then you can use the spent coffee grains to cultivate your own oyster mushrooms. So 2 benefits.
Yes, we are learning a lot. Thank you for the tip. Well, I think that we can move on. No more questions. Thank you, Irina. It was a pleasure.
Thank you very much.
And and now we are going to continue this session. And you know that the genetics of beer and wine yeasts and the development of novel superior brewing and wine yeast is a really exciting field of today's research, but this is a quite old area of yeast technology. The domestication of industrial beer and wine yeast is obviously very important from the industrial point of view, and is scientifically challenging. It's a very challenging research topic. And to explain it, we have with us Kevin Verstrepen. He's full professor in genetics and genomics at Leuven University, KU Leuven, in Belgium. He's the director of the VIB Center for Microbiology and the director of the Leuven Institute for Beer Research. Verstrepen studied biological engineering at Leuven University, joined the lab of Gerald Fink at the MIT, well, a genetics pioneer, as some of you should know, where he studied the origins of instabilities in the yeast genome. He was lecturer of industrial microbiology at Harvard University, but he moved back to Leuven University. Kevin Verstrepen is known for his research into the genetics of beer yeasts and the development of novel superior brewing yeasts. But he also received awards for best teacher from Leuven bioengineering students. And well, I believe that you will soon understand why. Well, Kevin, please.
Thank you, Isabel, for this too kind introduction. Okay, let me try and share my screen with you and see where we get. So you should be able to see my slides now.
Yes.
Great, thank you so much. Okay, so my lecture is entitled Beer Yeasts Are Dogs, Wine Yeasts Are Cats, and Some Other Yeasts Are Mules. And hopefully that will become clear when I flip through these slides here. I'm going to start with the conclusions, because I know in these online lectures, at least for my students, they give me prizes, but after 5 minutes, most of them are in a deep coma. Maybe that's why they like it, because they get a good sleep during my lectures. So I think it's smart to start with the conclusions. I will show that the different industrial yeasts that are out there for beer, for wine, for bread, for biofuels, are very different from the wild yeasts that are out there, and they've all adapted to their individual industries over hundreds of years actually. This adaptation, we've investigated it of course happens in the DNA, in the genetic material of yeast, and we've identified a few cases where we can specifically see changes that make these yeasts adapt to their respective environments and make them better at what they need to do for humans. So this is really a case of domestication. I will show you that domestication is the strongest in beer yeast. That's why they're dogs, and wine yeast, it's less so. So that's why they're cats. They're less tame. And then actually, I'm going to link back to a little bit what Irina has been talking about. I will show you how we make better yeasts for food industry, but then also even for pharmaceutical industry or biofuels, for example. All right, let me start by saying that when we talk about beer or bread yeasts, we talk about Saccharomyces cerevisiae, but there's actually many different individuals or strains of this yeast. And every brewery has their own specific yeast. And usually they use one yeast to make all their different beers, or a few. The same goes for wine yeast. There's many different wine yeasts. The difference is maybe that wine yeasts are often sold by big companies, so they're not often owned by a winery. And the same goes for bread yeast. There's many different bread yeast also sold by companies, so bakers can choose a yeast that they like, that has properties that they like. The funny thing is that while there are so many different yeasts, and they've been around for a while, when you ask people where they come from, they didn't really know. And here at the University in Belgium, we are sitting on one of the largest collections, especially of beer yeast, as you might understand, but also bread and wine yeast. So when I moved the lab from the US to Belgium, one of the first things I thought about was, hey, let's investigate what we've inherited here, and let's start sequencing a few hundred of these yeast. And back in those days, this was still a big undertaking. So we sequenced around 200, 250 different industrial yeasts. And much of that research is published. So if you want to know the details, this is one of the key publications, Calone et al. But the key figure really is this tree. This is really the family tree of industrial yeast, if you will. It's a circular family tree. It starts here with the Adam or the Eve of all yeasts. And then you have the wild yeasts here on the upper right corner. And they're completely separated genetically from all the industrial yeast, which you see with the colored bands here and the rest of the circle. We have way more wild yeast, but I'm not putting them all on the tree because then you don't see the industrial yeast anymore. So, but the first conclusion was that genetically, there's really two big groups. You have the wild yeast and the industrial yeast, and they're clearly separated genetically. So that's already something. It's a bit like dogs and wolves. They were clearly once related, but these industrial yeast became genetically distinct through their domestication. They started growing in man-made environments and they adapted to those, and that is really seen in their DNA. Then you also see that the industrial yeast split out per industry. We have two families of beer yeast, which we've called beer 1 and beer 2, not very imaginative. There's wine yeast here in red, and there's a mixed clade, which has some of the bioethanol yeast, some of the baker's yeast. There's in blue Asian yeast, so sake yeast, for example. So they're really split according to geography and the industry they're linked to. We were able, through some various molecular dating mechanisms, to put dates on when these industrial yeast split from the wild yeast. And maybe to our own surprise, it only happened sort of in the 1500s, which was at first sight maybe later than we first expected, because as Isabel already said, we've been making beer and wine for hundreds of years, or thousands of years. So why would they only split out that late? The answer is probably that this is a time where this yeast-based industry became more or less professional. And you see in records of brewers that they start really catching yeast and propagating yeast and taking yeast from one fermentation to the other, which was really the key to getting a separate genetic lineage or family of beer yeast, and then later also wine yeast. There's many more things I can say about this tree. The only other thing I want to say is that you see that the arms of the genetic tree here for the wine yeasts are much shorter than, for example, for the beer yeast, which really shows you that the wine yeasts are genetically more closely related and they haven't adapted as much to their environment. And also when we look at the traits of these yeasts, so that their phenotypes, how they behave, we see that they're more related to the wild yeast. They have more stress resistance still, they can still sporulate, they can do all these things that wild yeast do, that the beer yeast cannot do. On the other hand, the beer yeast have gained functions that the wild yeast don't have. So this clearly shows that the wine yeasts are not as evolved as much or not as domesticated as much as the beer yeast. The reason for that is probably that you only make wine once a year, right? When the grapes are ready. So the time that these wine yeasts have to really grow in the industrial conditions in grape juice is only limited to once a year for a few weeks. They don't divide that much. And evolution is, of course, based on progeny, on getting children, right? And the number of generations for these wine yeast in their industry was much lower than in the beer yeast, because beer is made throughout the year, pretty much every week. So these cells have undergone way more replications in the breweries. Another thing we found is that some of the yeast that we found in wine yeast, in brewer's yeasts, and in baker's yeasts, although less so, are actually not a pure species. They're a cross between two different species. Just a bit like a mule is a cross between a horse and a donkey. And then there's some cooler examples. There's ligers that are crosses between lions and tigers, or zonkies, a cross between a zebra and a donkey. These are real, they're not photoshopped, but they only happen in zoos most of the time. The mules are, of course, more known. So we found that many of these yeasts are also crosses between different species. And it doesn't look as spectacular under the microscope because they look exactly the same, which is why the brewers that were using these didn't even realize. And when we first got the results, I actually thought my students screwed up, that they mixed DNA of two different samples or something. So it took me a little bit of convincing, or it took them a bit of convincing to really ensure me that we're really looking at mixed species here. I don't have time to go into the details, but we found really mixes between lots of different species. And what seems to be a constant there is that these hybrid yeast, these interspecific hybrids, have the fermentation capacity of the beer and baker's yeast, Saccharomyces cerevisiae, together with the cold tolerance of some of the non-typical, non-standard yeasts like Uvarum or Eubayanus or Cudiraxevii. That seems to be a constant, and we typically find them in, for example, Belgian Trappist beers or Geuze beers, or wines often in cooler climates where they need to grow at cooler temperatures. And of course, the best known example, which we didn't discover, are the lager yeast. So if you drink a lager beer like a Heineken or a Stella or a Budweiser, those beers are always made with interspecific hybrids because they are made at cooler temperatures. Lager is the German word for basement, for cellar, where it's cooler. And so we actually, and that's unpublished work. We're hopefully going to publish a paper soon where we can show that these lager yeast really originated in a specific brewery in Germany in a basement, where this first sort of sex between different species of yeast must have happened. And then from there on, we can follow how it spread. It's really a beautiful sort of history lesson almost on industrial yeast. Apart from doing the genetics, we also looked at the traits, the behavior of these yeast. And I don't have time to go into the details, but it suffices to say that these different yeasts are extremely different in the aromas they produce, the stresses they can withstand, the sugars they can ferment. And that's all sort of summarized in this heat map here. But again, you see patterns. For example, the beer yeast, you see a lot of blue here, but some red on top. For the wine yeast, you see more red here. Again, no time to go into the details, but we could show that there's really a link between the behavior of these yeast and the niche they grow in. For example, the beer yeast have adapted to growth on maltose and maltotriose. These are sugars that are found in beer or in beer wort, in the medium from which beer is made. It's not found in wine, so you see that the wine yeast don't grow on this, but all the beer yeast obviously do. And the wild yeast actually are all over the place, so they've not been selected for this. And there's others, like the wine yeast are more resistant to sulfite, which is often added to grape juice. So they have learned to grow in high concentrations of sulfite. The beer yeast grow better at lower temperatures, the wine yeast are better at higher temperatures, so we can really see how it matches their proper niches. Another thing that I need to stress is that these behaviors are not just different, they're extremely different. For example, this is the production of an aroma compounds. You have to know that all these yeasts, they make alcohol and CO2, which is why they're most known, but they also make important aroma compounds. And the difference in concentrations they can make is huge. We found a more than 30-fold difference in isoamyl acetate, which is one of the main aroma compounds that they make in wine and in beer. So that's a huge difference between the lowest and the highest here. So you can really now start picking out yeast that produce way more or less fruity aromas, which can be useful. All right, on to the second part of my talk, and that goes a bit closer to what Irina has been talking about. These yeasts have been domesticated in the previous hundreds of years, and they've clearly gotten better at growing in their industrial niche. But we figured we can do better. We can use modern technology to speed this process up, to take it a level further and to make even better yeast. And so the concept is very easy. We're just doing breeding like we've done for cattle and for crops for thousands of years. Farmers have taken the strongest animals and bred them. So we're now doing this for microbes. And the cool thing is it can go much quicker. So we can make millions of crosses in the lab. That's more difficult with cows. We can do this in a few weeks and then screen them and take the best ones and repeat this. And for this, we're using lots of robots to really scale this up. I used to do this with students, but they complain a lot and they make errors. So the robots have been fantastic. So literally we can make thousands of crosses now in only a week time, and also test these. To test them, that's also a challenge. We've been working with microchips together with a company next door here, IMEC, that can make sort of small chips in which there's no electricity flowing, but we have little droplets flowing. And in each droplet, we can put one yeast cell. So you get like mini fermenters, mini brewing vessels almost, so fermentation vessels. And in each of those, we can put one yeast and then select or test how well that yeast is doing for a certain property. So that increases the throughput, which is essential for what we're doing. And I just want to show you one example that was together with a company. They asked us to create a yeast that can grow in a certain medium that none of the natural yeasts of our whole collection, we select 2,000 or we tested 2,000 yeast, none of them could grow very well in this condition. And in a few years, we were able to breed yeasts by just crossing and recrossing and testing millions of crosses. We were able to get these red yeasts here that in a very short amount of time ferment this medium completely. So they reach the complete fermentation of all the sugars in this medium. And the important thing is that these are not genetically modified yeast. This is just breeding, like farmers have done for thousands of years. You take two kinds of apples, you cross them, and you might get a better one. And that is important for the food industry because in many cases, the food industry is not ready yet to use genetically modified strains. So it's a bit old school technology, but using also the modern techniques that we have to speed things up and to make this thing go quicker. We've also, for example, made yeast that make even more aroma compounds. I've shown you this curve before. So we've bred a few yeast that make more fruity flavors even than the most fruity natural yeast. And those can be immediately used to make beer that tastes very fruity. So this was for a brewery that wanted to make a Belgian strong beer, but not have the strong alcohol, just the strong yeast aroma, the typical fruity aromas. And so they can do this. They can make a 5% alcohol beer that almost has the same aroma as a typical 8-9% Belgian Trappist or Abbey ale. And then the last couple of slides, we are also going into synthetic biology, right? As Irina has shown, we're now at the stage where we can make synthetic DNA in factories. We can order pieces of DNA, put that in yeast to make them make things that they normally wouldn't do. So those things you cannot do by breeding. And so one of the things we are doing there is trying to find out which genes we need to modify. And we're using, again, our big yeast collection to try and link known characteristics of yeast to specific genes that we can then further engineer. And so one of the projects that we've recently completed is together with Dan Jaros at Stanford. Dan had made 1,100 crosses between two parents. And that's another example why yeast is so fantastic, right? It's a sexual organism, and you can make 1,100 children of the same parents. That's way more difficult with cows or any other higher organism, I would say. Anyway, the reason why we did this is that these two yeasts are very different in their traits, and by making 1,125 next generation crosses between these yeasts, and then using sequencing, so all of these were sequenced, that already tells you something that we can now sequence 1,000 yeast without much problem, whereas 10 years ago, 100 yeast was a lot. And we also did 1,100 fermentations and measured the aroma compounds and whatever. And putting that together with bioinformatics allows us to link variations in the genomes of the two parental strains with the differences in their traits, in their characteristics, and a pinpoint like, hey, a change in this position here in the genome is linked to a specific trait. And I'm sorry that I cannot show you all the examples, but it's about isobutanol production, temperature resistance, ethanol production, stress in general, aroma production, all the things that we could measure. We've identified, I think, 300 or so QTLs in one huge experiment. That we're now, of course, also looking into to test. That paper is under review. If one of the reviewers is listening here, please hurry up because it's been under review for quite a while. And so that allows us then to further improve existing yeasts and, for example, make better yeasts as a starting ground for synthetic biology. And one of the things we've been working on is an antioxidant called astaxanthin. Other labs have also used it or produced it. You need to introduce lots of different genes. But by then tuning these yeasts a bit better, we can make yeast that make more of this compound. So those are the things that we're currently doing in the lab. Last slide I want to show is that there's a free online course on the edX platform for those of you really interested in beer and also the microbiology of beer. You can take 10 free courses on an academic level on beer. Just go to the edX website and look for Beer, the Science of Brewing. Highly recommended, like 15,000 people already took it. So if you're interested in this, you can find way more material there. Okay, I'm going to leave it at that. I have to thank the people in my lab who do all the work, and of course some of the industrial collaborators that we have. Thank you.
Thank you, Kevin. So you have a lot of questions. 4 questions, 2 of them are similar, and we have 3 minutes. So I'm going to read one of the 2 questions because they are similar. Do any of the mixed yeast species have trouble reproducing and generating descendants? So this is one. The other one is, it is possible to build a super yeast by sequentially breeding the best, more interesting yeasts, What are the limitations? And the other, well, it's very, perhaps more scientific, even more scientific. How are you able to construct a family tree for such simple organism as yeast? So
okay, I will start with the third one. So yeah, it's a simple organism, yeast, but their genomes, their DNA is actually more diverse than the human genome. So humans differ like a few letters per thousand. And yeast, it's almost 1 letter per 100 different nucleotides if you take the most different yeast. So actually, so whereas yeast seem like under the microscope very simple, their genomes, their DNA, in a way is more diverse than the human genome. So in a way, it's easier to make a genetic tree. And don't forget that the genome is still millions of letters long. So we have enough differences to know exactly who are brothers and sisters and uncles and aunts and a bit further. So that was relatively easy. Then the second question was about combining the sequencing with the breeding and where the limitations are. So it is true, it's something that I haven't talked about, but we actually do use some of the information from the sequencing in the breeding. Because once you know which part of the DNA are responsible for a certain desirable property, you can actually keep track of that part of the DNA and make sure that when you do breeding, that you definitely have that part also in the offspring. You can select for that part through PCR. I don't know how much people who ask the question know about molecular technology, but so yes, we keep track of pieces of DNA that we know are important, and we make sure that those are in the offspring. That helps us a little bit to speed things up. In yeast, it's okay. If you have to breed, for example, apple trees, this is way more important. Why? Because it takes you several years before you can test whether a cross is making better apples. You know, you start with a very small tree that needs to grow before it makes apples. And so you can gain a lot of time by testing the DNA, because you can do that immediately, even on the small tree. With yeast, when you cross two yeasts, literally a week later, you have the hybrid and you can test it. So following the DNA is not always the best mechanism, but we do it quite often. What are the limitations? Well, we feel we can improve traits with maybe 1 order of magnitude. That's about the maximum. So 10-fold better at certain things. Sometimes it's only 50% better. So, 1.5-fold better. But it's very difficult to go to 100-fold or 1,000-fold better. For that, you need DNA technology, genetic modification. And you cannot let them do things that they don't do in nature, right? You cannot have them do photosynthesis or make a pharmaceutical compound. For that, you need genetic engineering. And by now, Isabel, I forgot the very first question, actually.
No, it was not. If the mixed species have troubles reproducing and generating descendants.
Right, absolutely. So it's a very good question. Mostly when you cross two different species, like a mule or a liger, they become sterile, right? They cannot produce offspring, which means they're evolutionary dead ends. The same is true for yeast. They become sterile, so they cannot have sex anymore. But yeast can also reproduce asexually. And so it can make clones of itself. And actually, that's usually how it multiplies. Sex is actually not so common in yeast. You have to really almost force them to have sexual reproduction. So that's the cool thing about these interspecific hybrids. They can still grow, multiply. And that's what they're doing in the industrial niche. And they don't need sexual interactions or crossing to really thrive. So it's a very good question. And that means that interspecific crosses in yeast are actually not an evolutionary dead end and can really be useful. Whereas, mules and ligers and wolfins are a different story.
Okay, thank you. Thank you, Kevin. Thank you a lot. So we're going to continue with the production of bioethanol from biomass by yeast. So you know that bioethanol is the most widely used biofuel as a blending agent with gasoline, especially in the United States. So it's the largest producer, followed by Brazil. So we have here a speaker from Brazil, which makes a lot of sense. So the first generation bioethanol industry is based on the fermentation of food crops with starch and sugar, so sugarcane, beet, corn, wheat, but and has demonstrated viability. In the case of bioethanol, second generation bioethanol from lignocellulosic materials, well, this has continued facing difficulties. So the next presentation is about yeasts used in bioethanol production. We have with us Andreas Gonberg, he's professor at University of Campinas, Unicamp in Brazil. Andreas also acts as a consultant for companies in the field of industrial biotechnology. So he is a chemical engineer graduate with a PhD by Universidade de São Paulo in Brazil. He worked as postdoc in different laboratories in the world, so even in Portugal, Technical University of Denmark, TU Delft in the Netherlands, and more recently at Harvard University. So Andreas has also been active in promoting science for the general public, especially for children and teenagers. So he is very interested, let's say, in meetings like this one for disseminating microbiology. Please, Andreas, thank you for coming, and it's your turn.
Thank you, Isabel. Can you hear me well?
Yes.
So it's a pleasure to be here with Irina and Kevin and you in this session. And I'm going to talk about fuel ethanol, mainly from sugarcane. Let's see if I can share my screen here. Where is that? Okay. Can you just confirm that you
yes,
can?
Yes, that's fine.
Yes,
that's fine.
Okay. So I work at the University of Campinas in Brazil, as Isabel said, and we are interested in the yeasts that are used to produce ethanol as a fuel. And I will start with something that has already been mentioned here, just to bring you a very familiar scene to all of us. These are all dogs, different breeds that humans made along hundreds of years. And they are very different in shape and in size and color, but they all belong to the same species, right? And if we go for yeast, and it has already been mentioned here that yeast can be used for wine production, beer production, sake production. It can also cause some diseases in humans, and there are also yeast in the wild. But if we look at them under the microscope, they are going to look pretty much the same. So in spite of this, we should not be fooled. They look pretty much the same, but they are genetically even more diverse than the dogs I have shown you before. And they all belong to the species Saccharomyces cerevisiae. And I'm going to focus today on those strains of Saccharomyces cerevisiae that are used to produce ethanol. And I think the main reason we are interested in ethanol is that it can be used as a fuel for light vehicles, as Isabel mentioned, combined with gasoline, but in Brazil, we can also use it as a standalone fuel. So I can, for example, use pure ethanol in my car if I like. And compared to gasoline, it releases 90% less greenhouse gases in the atmosphere. Another interesting point is that in the sugarcane biorefineries, you can see a typical example here in this picture. We have 420 such industrial units around in Brazil. Not only ethanol is produced, but also table sugar and electricity. In the red rectangle here, you can see the fermentation section of this biorefinery, which is where the yeast will convert sugarcane sugars into ethanol. Another hot topic at the moment is the possibility of developing new technologies to use ethanol to fuel electric motors. So the development of fuel cells to produce hydrogen in the car, which will then react with oxygen, produce electricity, and fuel an electric motor. And this is an opportunity that countries like Brazil have at the moment as an alternative to pure electric motors, as has been discussed in Europe and North America, for example. And the other thing is that we should remember that we are not anymore discussing the possibility that we have climate change or global warming. We are really living a climate emergency at the moment. So together with wind power and solar power and other renewable options, I think ethanol is something important for us to better manage our climate emergency. I'm going to talk mainly about first generation ethanol, which is the ethanol, as Isabel mentioned, produced from sugars, from sugarcane or sugar beet, or from starch, from corn, for example, because Although there has been a lot of interest in producing second generation ethanol, as you can see here at the bottom of the screen, today, unfortunately, only less than 1% of the worldwide production is lignocellulosic ethanol, which is second generation ethanol. So still, 99% of the global ethanol production is first generation. And as you can see here, there is a global production of around 110 million cubic meters per year. 55% of this volume comes from corn in the US, and 30% of this volume is produced in Brazil, mainly from sugarcane. Corn ethanol is already almost 10% in Brazil and increasing actually very fast. One interesting point is that corn ethanol is mainly produced using genetically engineered yeast, as Irina already explained and Kevin mentioned. Yeast that have been subjected to genetic modifications using recombinant DNA technology. So these yeasts are mainly employed in the US to produce ethanol from corn. Whereas in Brazil, it is just starting now, we are just starting to use genetically modified yeasts to produce ethanol from sugarcane, but still more than 90% of the production is still based on non-genetically modified yeasts. So the production is basically carried out with selected yeasts, as I will describe. So I'm going to talk mainly about the sugarcane biorefinery, in which we have sucrose from sugarcane being used to produce ethanol, and also table sugar and electricity. So these three products are actually crucial to keep this business running. It's not possible to think about producing only ethanol, for example. We need the three products to make this a profitable business. So how does the yeast look like in this process? So differently from beer, as Kevin described, we don't have hundreds of years of experience in this. It's quite recent. So in the 1970s, that we started producing fuel ethanol massively in Brazil. And the process looks a little bit like this. We have a fermentation in a big vessel, which lasts about 8 hours. When the fermentation is finished, the broth is centrifuged, the supernatant is used to distill ethanol, and the very concentrated yeast cream is then subject to a sulfuric acid treatment at very low pH to fight contamination. I mean, this is a non-aseptic process, so contamination is actually inherent here. And then after this acid treatment, the yeast cells are recycled to a new fermentation round. So basically we have 3 fermentations every day in one vessel, and this process runs 8 months a year, so almost continuously. So there is opportunity for evolution to happen. So we have some similarities to what happens in the beer industry, but again, we started in the 1970s, and also the number of generations, as Kevin mentioned, which is crucial for evolution to happen, is very low in this process. So the process is run in such a way that we almost have sugar being converted into ethanol with very, very little yeast growth. And obviously we need then to drain a little bit of yeast from the process so that there's no accumulation of biomass here. But basically in the 1970s, people started purchasing baker's yeast, as you can see here, and throwing tons of baker's yeast into the big fermenters. And we are talking about 1 million liter fermenters, so they are really huge. And in the beginning of the century, some research groups in Brazil started following the population dynamics using molecular methods. So here we have an example of a PCR fingerprinting method that generates electrophoretic profiles like this, which basically are like fingerprints. So each strain, each yeast strain would present a different profile here. So they realized immediately that the baker's strains that were used to start the process were substituted by other Saccharomyces cerevisiae strains. So you can imagine here that each color is a different yeast strain. So baker's yeast wouldn't survive in the process for many weeks. It would be naturally substituted by other Saccharomyces cerevisiae strains. And we don't know exactly, and we didn't know, we knew even less at that time, where this other strains that invaded the process come from. So these works also showed that it's possible to have the coexistence of at least two Saccharomyces cerevisiae strains in the fermenters together. And they started also suggesting that ethanol producers should start the process every year with strains that they had isolated from the process itself, instead of purchasing baker's yeast in big amounts, which represents a high cost actually. This is a work that was published in 2008, and it's a sort of landmark paper in this field, because these people, they followed several biorefineries along 12 years. So this work reports the results of at least 12 years of studies. And they isolated many different strains from different biorefineries. And here they were using chromosomal karyotyping as the method to discriminate between one strain and the other. So you can see here that from 1993 up to 2005, they sampled from many companies. They made many, they produced many of these karyotypes, and they started characterizing many different strains that were naturally present in these biorefineries. So this is the kind of result we have from this chromosomal karyotyping analysis. And then you really need to be an expert to be able to say, oh, this profile is one strain and this other profile is another strain. This is actually also somehow subject to debate. Researchers sometimes will not agree on what is different and what is not different. And these people also brought these strains to the lab, and they created a miniaturized biorefinery to be able to look whether these strains produce, for example, foam, or whether they flocculate, because these are undesirable characteristics in industry. We do not want to have strains that flocculate. We do not want to have strains that produce foam. So they selected strains that would behave like this here. So these would be well-behaved strains that we could think about reintroducing in the biorefineries in the subsequent industrial periods. They also compared the industrial strains, and I'm showing here one which is called PE2, which is maybe the most popular industrial bioethanol strain used in sugarcane biorefineries. They compared the strain with baker's yeast, and they showed that they indeed present a higher ethanol yield, a lower glycerol yield, and high cell viability under conditions that resemble those conditions in the sugarcane biorefineries. Here on the right, you can see how the ethanol yield developed along the decades. So you can see that in the 1970s, 1980s, we were not very good at having high yields, but then in the 1990s, we started achieving more than 90% of the maximum theoretical yield. And since then, this yield has been quite stable. So now, if we want to increase this yield further, we really need breakthrough technologies, for example, using synthetic biology, but we basically also need to better understand this process. Contrary to wine and beer and bread, there is not much science behind bioethanol production, unfortunately. Here again on the left, you can see this natural strain substitution that occurs along the 8 months of a production period. So every different color is a different strain. And as you can see, baker's yeast is here naturally substituted by other strains. So in this study, they also mapped permanence and prevalence of these strains in the process. Permanence means the capacity to stay in the process from the beginning to the end of the season, and prevalence means the percentage or the frequency with which this particular strain is in the process at a given moment. So this was a breakthrough study, but the process has changed a lot in the last years. And one main thing is that we introduced mechanical harvesting of sugarcane in the 2010, around 2010. And this produced a great change in the microbiota of the process. So we do really need to invest more. 2 minutes, Isabel. Okay, we do need to invest more in understanding what's going on. This is just a slide. It was a research from our group showing that one of the hallmarks of these industrial yeasts is that it can it has a very high tolerance to low pH. So it can survive, you see the black dots, at low pH better than, for example, baker's yeast or laboratory yeast strains. Here you can see in this table that the producers of ethanol, they just have a handful of strains they can purchase in the market to start the ethanol production season. So it's about 5 or 6 strains they can buy to start the process. But still, we don't know what is the molecular basis for the outstanding traits of this of these strains. Why do they dominate and persist? We have some genome sequences that started being released in 2009. Some studies also show that these strains are quite well behaved. They are diploid, they sporulate well. They are not as complicated as beer strains, for example, as Kevin described. And just to give you two examples of traits that have been mapped, they have an increased copy number of vitamin synthesis related genes, which is an adaptation to this environment. They also have inactivation of aquaporins, which is related to the movement of water, which makes them more tolerant to osmotic and high osmotic environments. And a recent study, and this is 2021, proposes that these strains come from cachaça strains that in turn came from the European wine strains. So this is the first proposer of an evolutionary path for the bioethanol strains. And this is the study we are currently working on. This is 4 different industries and 4 different scenarios, just to show you that the population dynamics is completely different in every year and in every industrial unit. So we do need to better understand why these things happen to be able to design a better process. And I will leave you with this. Thanks for your attention. Thanks, Isabel, and I will be happy to answer questions and discuss.
No time for questions. Thank you. Thanks to all the 3 speakers. It was a pleasure to have you with us. We will be killed by the next session speakers and host if we stay in the room. So bye-bye. It was a pleasure and very nice one East hour. Thank you.
Thank you.
Thanks.
Bye.
Well, thanks everyone for our yeast hour. Fascinating set of talks there about products and organisms that, you know, impact on everyone's lives. I'm going to pass the microphone over to Eleni next, because she's going to introduce the next session, which is our science/art session. So we'll hear more from her.
Okay, guys. So science and art session. In this session, we are going to dive deeper into the concept of merging science with art. Art can not only make a concept easier to understand, but it can also make complex ideas creatively easier for people to understand. In fact, our ability to visualize and imagine processes go all the way from learning till creation to processing ideas and presenting science and microbiology. So all of these things we're going to be addressing in these 2 hours of science and art. And we have a fantastic array of 6 speakers from 5 different countries around the world. So I'm just going to welcome into the Zoom call Anna, who is a freelance artist. She has 20 years of experience in exploring infectious diseases and synthetic biology. And yeah, I'll just wait for her to join and see if she can hear us and check the microphone. Okay.
Hi.
Hello, Anna. How are you?
Yeah, good, thanks.
Cool. All right. So I just gave an introduction to the session, and I told everybody that you have over 20 years of experience with exploring science and art, and we are just so excited to have you here. Mm-hmm. Just a little background about you, I guess, before starting, that you were our Microbe Art 2020 Grant Prize winner with all your amazing creations. So yeah, can you just tell us a little bit about what topics are you gonna be discussing in your talk?
Well, I'm going to talk about my new work mostly and what I've been up to in the last year. So some shows, some work that's already existing but is already out in different places around the world that you can go and see physically at the moment, and also projects that I'm working on at the moment.
Okay, so I guess you have around 40 minutes to do your presentation, and then at the end we'll do a short Q&A. So everyone that you're watching the live, please do comment below the chats all your questions, and we're going to address them at the end. So yeah, I'll just hand the floor to you, and good luck.
Thank you. Just going to start my slides. Right, so I was going to talk about an artistic exploration of microbiology from my perspective, because I'm an artist who's kind of dedicated my life to microbes, I guess, and this is since winning the Microbe Art 2020 award, this is the kind of stuff I've been up to since. I've got quite a lot of projects on, so I'm just going to start talking about them, and then at the end I've got a short film to show you of our project, which is with Boku University, but I'll come to that. So early in 2020, I started working. I was already collaborating with the University of Brighton on a project about data around domestic violence, and it's not a microbiological theme, but I thought it was something interesting that I could to do with healthcare that I could turn my my creativity towards, but as I was just about to start the project, the lockdown happened in the UK, and my workshops with the charity Rise, who I was going to collaborate with, were all cancelled. So I ended up looking at the data around the impact of the COVID pandemic and the lockdowns on women who were survivors, victims of domestic violence. And the figure 30%, or even 3 times, kind of came up a lot. So I made these 30 little tiny beds, which you might recognize because they were from some very striking images of the first temporary hospitals in Wuhan of the COVID-19 pandemic. But in this case, I made the bedding by hand, and it's based on that. And I think they got the bedding from like various factories and whatever in an emergency, because they were all like brightly coloured bedding and things. And I was really interested in this sort of feminine art link. Sometimes there's a lot of embroidery in my work and telling the stories of science through things like embroidery. So the idea of the work in the future is that I would like to do workshops with women who have been subject to domestic violence, but these were ones made by me referencing various things and thinking about sort of Virginia Woolf's room of one's own and this idea of the emergency happening, but the women were locked in with the people who were kind of committing the domestic violence. So rather than having the home as a safe space, it became a kind of contested space. So this is what the work was about, and it's called shielding, which is a term that was used in the UK to describe shielding of the vulnerable. But it's not something. I think in the USA they call it sheltering in place, meaning a similar thing. So if you had if you were like over 70, or you had immune compromised issues, then they told them to shield during the pandemic. So we were playing with this idea of shielding, but kind of talking about how the idea of shielding was actually a negative in this case. So it was like playing with those things. And each of the beds is impregnated with the extracted RNA of COVID-19 or SARS-CoV-2 from a plasmid construct that I collaborated with Dr. Inez Mora and Dr. Jane Freeman at the University of Leeds to do this. So it's a thing that happens a lot in my work, that you kind of see there are these invisible elements that are the real biological elements in it. So they're very tiny, and you can sort of see me kind of setting up the installation there. It's never been open to the public yet, and that's something I would like to do. I was able to set it up in the gallery that we had got booked, but I couldn't do that. Something that is on show to the public now and also is impregnated with DNA, in this case, impregnated with Yersinia pestis DNA, is a collaboration I did with the National Collection of Type Cultures in London at Public Health England, and I'm their artist in residence. So it's like the dream job for me to be able to access this historic bacterial collection and make work with the stories that are there. And they've got some amazing things there. They've got letters from Alexander Fleming. They've got cholera, the oldest living cholera from 1915 from Alexandria, from a soldier, which I'm doing a project with now. But this one was with Yersinia pestis, and it's called Plague Dress, and it's currently on show at the Boerhaave Museum in Leiden in the Netherlands as part of a show called Contagious, which they planned a long time before the pandemic, but then got delayed due to the pandemic. They were talking about the forthcoming kind of disease X, and lo and behold, it beat them to it, kind of being the situation there. And this dress is a 1665 dress design, which is the date of the Great Plague of London. It's made of silk to reference the fact that the Silk Road was kind of a vector for the plague to travel from the sort of the steppe region through kind of the maritime port canton, I guess, as it used to be called. Now it's called Guangzhou, and through to the Netherlands and then on to the UK. So it's great that it's now on show in the Netherlands, and before that it was on show in Guangzhou, in the Guangzhou Triennial. So it's kind of followed the route of the plague to the UK as well, to London, and it's stuffed full of spices. There's a roll under the dress which is stuffed full of spices that we use to treat the plague, and the lavender references this, the posies and things that people would carry, and the beaks of the plague doctors and things like that. Not that in London there's no records of plague doctors dressing like that, but there are in the Netherlands. So sort of referencing those things. And this embroidery on it is an actual period embroidery that I managed to acquire. I use a lot of kind of found historical artifacts that are maybe not the most valuable artifact in the world, but they're from the time, so they're kind of conceptually they're important to include. So this embroidery on the dress is from that time, and I've impregnated the embroidery part with the extracted DNA of Yersinia pestis, which I extracted in the lab with the scientists at PHE in the National Collection of Type Cultures. And I have previously handled live plague. This was DNA from kill plague, but I have previously handled live plague in a category 3 lab at PHE Porton in the UK as well. And I'm the first artist, as I understand it, been able to do such things. I'm fascinated, and I deeply go into it, and I deeply go into all the health and safety as well. And I work with the leading experts in this. So there's some close-ups there. And that's how it looks in the Boerhaave Museum at the moment. And that is when they had the opening last July, and that's the King of the Netherlands actually looking at the dress. So it was quite exciting, considering we were still, just coming out of one of the lockdowns at that point, to see this happening. And the show's, it's locked down several times in the Netherlands, and so it has to close and then it opens up. But luckily it's a very long-running show, and they might even be extending that. So I'm hoping that I'll get to go there and see the show before it finishes, because it looks absolutely amazing. A piece that I also worked on with Alex May during the lockdown was a piece called Susceptible, which is through my collaboration with the University of Oxford and the Cryptic Project and Modernising Medical Microbiology there, who have been doing for a very long time. I've been their artist in residence since about 2010. So they've been doing this for a very long time, looking at how you could implement genomics technology and microbiology in the UK health service and beyond. And so quite a few years ago, Modernising Medical Microbiology actually had it. So it was implemented in the UK that everyone who had a TB, their TB was whole genome sequenced routinely and went into a database and could be understood and things. And the Cryptic Project has been looking at how you can really work internationally with this. And they've got partners all around the world. And what they did was they made these multiple well plates with different antibiotics in it, and they put different patients' TBs in these plates and sequenced the TB as well. So they looked at the different antibiotic growth, and from that huge data set, they were able to understand which of the first 4 frontline antibiotics will treat the disease just from the genomic data. So from the whole genome of the TB, they can say this patient needs this antibiotic. So it was absolutely groundbreaking and could be a protocol that can be applied to lots of different organisms in the future, so that we can actually have really targeted treatment, which is really important for resistance and avoiding drug resistance. And this installation we made from their data. So this is a visualization of it, but we do have the full working exhibit, but we haven't been able to show it yet, again, because still galleries are only just kind of opening up for their big, larger scale shows, and that's in only some countries. So what it is you're kind of entering inside these lungs, these sort of bronchi and alveoli and things, and the 4 different coloured clouds are the grey, the red, the yellow, and the white. They're different antibiotic drugs, and each of the little bacteria that you see floating around, they're actual data from a patient in the system, and so they're susceptible to one of the drugs, and they will cluster around you as you go to the installation, and you kind of need to move with them slowly towards the right antibiotic, and when they reach the right antibiotic, they kind of disappear and they get sucked in away from you. And so it's kind of about teaching people that antibiotic drugs need to be targeted. You can't just take any old antibiotic and hope for the best. And sometimes that bacteria are resistant to certain antibiotics. So the idea is that we'll be showing this as much as we can all around the world. And it's a very beautiful kind of magical thing. And it was shown with a reflective floor as well. So that's something that's up and coming. And these are close-ups of it. So this was funded through the Arts Driver program at the University of Brighton, and there was a requirement for this piece that you had to include Gatwick Airport data, London Gatwick Airport data, and so these kind of lines that are going through it, these sort of little dots that you can possibly see, hopefully, are based on, and I think there's another one here. Here you can see it more clearly, these little dots alongside the bacteria. They're the Gatwick flight data to and from destinations that are partners in the study. And so we had this ability to download this Gatwick flight data. And we were just expecting normal flight data and things like that. But then the lockdown happened. And then, so what we have the story of the impact of disease incorporated in the piece as this data just trails off and then picks up a bit. And you can sort of see lockdowns easing and lockdowns coming back again and things like that, all happening in the flight data that's overlaid within the installation. And there's another example of it. So it's all kind of moving around you and you're moving within it. And then another piece that I was actually able to show during the past year was my piece Ex Voto, which is each of the ribbons in the installation tell a story in their own right. They're dyed with different antimicrobial drugs, different antibiotics. The Staphylococcus aureus from my body, genetically modified Streptomyces silicola. There's different other bugs that we've kind of worked with. Woad, which is an antimicrobial dye, Madder, which is an antimicrobial dye, and all sorts of things like that. And so this is one version of it, and each of the little votives in it, these are kind of secular votives, like you might see in like a Greek Orthodox church, but these are kind of secular and about storytelling. Although somebody said that, it could be that if we don't deal with antibiotic resistance, then maybe prayers and votives will be all that we kind of have to go back to as well. So it's got that link there. So these votives are handmade, some by me and some by lots of different people, some by scientists, some by children, some by visitors to exhibitions, and all in different countries and all around the world. So I've had people work on it in Israel, in Egypt, things like that, and also There's one of the scientists who was a creator of the antibiotic augmentin, actually participated in a workshop once and made one. So that's in there too. And I showed it in the Valkhof Museum in the Netherlands this year, and they had a show called The Plague. And so in the case of this one, it incorporates both votives from the lockdown. So I made a series during the first UK lockdown. Every day I made a votive to tell the story of what was happening in the news that day. So there are things like toilet rolls, shopping bags, Captain Tom Moore, who became like a famous hero in the UK for he's 100 years old and he was doing laps of his garden to raise money for the NHS. So all these things are kind of in there in the story, so like a timeline. And then the Valkhof commissioned me to make a series of plague votives. So we've got the lockdown votives, which are on turmeric dyed ribbon with the SARS-CoV-2 RNA impregnated in it. And then the plague votives are on walnut dyed silk. And they're all made by me based on the history of the plague in the Netherlands and the UK, and the more contemporary like Madagascan outbreaks and things like that are all kind of incorporated in it. And it's impregnated, the ribbon's impregnated with the Yersinia pestis DNA that I extracted. And so you can kind of walk under it as well. And that's the installation view in this. This was a fantastic show with exhibits by sort of manuscripts by Albert Camus and paintings by Dürer and all sorts of things in this fantastic show that was there, but that's finished unfortunately. So they've just returned the piece to me and it's looking for another venue. And I've been working very intensively during the lockdown on this piece called Hypersymbiotics, and you can maybe make out on there that it says post-pandemic edition. So I've made a few versions of this work, which I came up with Professor John Paul, who's my longest term collaborator. I've been working with him for over 15 years, maybe more now. And we were talking about could it be possible to have like bacteria that could like improve you, kind of like actually improve you and things. And this was a commission that I kind of pitched to the Wellcome collection quite a while ago, and they commissioned it as a performance piece. And then I made a kind of an object based on an apothecary box for the Science Gallery Detroit, which was talking also about advertising and snake oil cures and things like that. And so now I've gone a step further with it. And this is a new commission, which is going to be as part of an exhibition called The World is in You, which is organised by Medical Museum in Copenhagen, but will be exhibited at the Kunsthal Charlottenborg in Copenhagen. And each of these bottles contains some kind of story, very complex story in the case of most of these. And so there's a link to it. I can't go into huge amounts of detail, but I've got, for instance, a bottle of psychobiotics in the middle there at the front, which I did a collaboration quite a few years ago, 2015, with the UCL iGEM team, who were working with, could you make a genetically modified E. coli that produced serotonin that could kind of make you happy. So it's kind of probiotic E. coli that would make you happy. Obviously, it hasn't been tested on people and things. You don't know what the overgrowth might be or things like that. So none of these things are kind of like definitely there. Some of them are more there than others, and that's part of the discussion with it. But it physically contains the DNA, the plasmid, inside these blue tablets in the psychobiotics, and the company GenScript sponsored this part of the artwork and they supplied that. There's also there's chicory root. I'm doing a project with that, which chicory as a superfood, genetically modifying chicory to make a probiotic that will improve your gut microbiota. And jumping genes, we've got one called Dr. Clara's Jumping Genes, and that's a collaboration with the Helmholtz Institute, with the Institute for Epigenetics and Stem Cells there, and looking at this idea of transposable elements in DNA. I can't go into the details of this because it's very complicated, but you can see some more close-ups there and there, some of the things. Oh yeah, I've got the tooth repair pills, which are containing a biomineralizing bacteria, a kind of Serratia N14, I believe it is. And there's also the new normal pills, which contain the SARS-CoV-2 RNA. So it's kind of bringing together a lot of different projects and telling the story in a different way. Another piece that's been exhibited quite a lot during the pandemic has been the Archaea bot, which is a different kind of, based on a different kind of microorganism, but based on Archaea this time. And it's, so Archaea, as you know, are another branch on the tree of life. They're more genetically similar in the way that they replicate their DNA to humans than they are to bacteria, but they look very much like bacteria. This Archaea that was based on, this is an underwater robot that I made with Alex May through the EMAP programme and funding from Arts Council England. It's a 3D printed robot. We worked with the MARA project at Imperial College, and I worked with the late Amanda Wilson, who was an amazing microscopy expert working with electron cryomicroscopy. And they were studying these archaea called Sophilobus acidocoldarius, which are incredibly acid tolerant and incredibly heat tolerant. So we they were studying them. Could you understand how the Archella motors, these little cogwheel-like motors on the surface of the Archaea, which spin their kind of tails around and help them tumble to better situations. And they were studying, could you find out how they exactly look and make them from DNA origami with a view to using them like robots, like nano robots that you could attach to something like bacteria and send off into a cell, into the body to attack bad cells or something like that. So we made this idea of the robot really struck us, and we made this underwater robot, and we were kind of thinking about how the Archaea are like in a perfect situation for, well, these Archaea anyway, are in a perfect situation for a potential post-climate change world. They love acid rain pollution. They love hot temperatures. It grows very happily in 80 degrees water or solution, growth medium anyway. And so they would be very happy with this. And then we were playing with the idea of the notion of the singularity and this fear that we have of AI and like the future of the human. So it's kind of a dystopian solution for climate change, that we could maybe upload our consciousnesses to the neural networks inside these. It does have a neural network inside it, controlling the movement of the motors and how it swims about. And we could live on post-climate change, post-singularity in this very dystopian world. But it sort of alludes to the idea that sometimes, you know, these sort of techno-progressive kind of billionaires and stuff are kind of coming up with these kind of solutions sometimes. You think you could just have a few wrong steps and you could end up with something like this as the best we could do. I've done a lot of work with seaweed during the lockdown. This is some microscopy of seaweed, and I started to get very fascinated. I did a project with Fabrica Gallery in Brighton, and I managed to find this old book, which was a study of certain British seaweeds and their utilisation in the preparation of agar. During the Second World War, the agar supplies, there's been shortages recently as well, but the agar supplies were obviously being blocked by the war, and we needed a lot for things like the development of penicillin. So there was a move to try and investigate which British seaweeds might work for this, and it's quite interesting because things have changed as well. We've got a lot more kind of invasive species as the temperatures of the water change and stuff. So I did a lot of study on that. And there's just a picture of me kind of in the lab doing some other work. That's with my Fermenting Futures project. And so another thing that I was working on in the past few months is a collaboration with the University of Western Attica in Athens, looking in the Department of Conservation of Works of Art and Antiquities, and I've been exploring the impact of microbes on antiquities, and they're researching, it's really fascinating, the microbiomes of the antiquities and trying to work out whether you could do things to those microbiomes or adjust those microbiomes or encourage different growth or even consider using synthetic biology to do this. So you could actually have microorganisms growing on them that are actually cleaning them. And so I did a collaboration with the scientists and the art historians there and the conservators there, and learned about all the issues that they're dealing with, and then made these kind of, I call them 3 sketches, which each kind of are impacted by these issues. So I used seaworn marble that I managed to get from eBay really, really cheaply. So it's not a great kind of valuable antiquity until, maybe in the future. And then I drilled holes in it, because holes are apparently the worst thing, and plants are even worse. So plants growing into these things. And I painted them with agar, and I painted them with the mud from a Vinogradsky column, and they started to kind of turn purple in patches, which I guess is Chromobacterium violaceum and things like that on there. And there's paintings on there because there's paint on there which is referencing the microbiomes as well, but the paint also references the fact that all the Greek antiquities were painted, and that's kind of very much not in our consciousness. We kind of think of them as these sort of pure white things, but they were actually very gaudily painted in some cases. So kind of playing with this issue as well, and how do you conserve this? And there's some issues around like the inclusion of bioartworks in like museum settings. So I've shown a few pieces where I have managed to do this, but in a lot of cases, especially with the living works, people are very wary to include those works in museum collections. So part of our exploration is looking at how could these works be conserved. So one of these objects, the larger one, has gone to Athens and is now in the hands of the conservators there, and is being looked at under electron microscopes and things like that. And some of them have these carvings on, which are based on the electron microscopy of the microbiota on antiquities. And one of them is, just trying to move along. One of them's been put into, that's not the right one. Oh, here it is. I've gone too far there. One of them's been put into a Vinogradsky column to see what the effect of the microbes are. One of them's staying with me at the moment damp, and when I feel it's reached the correct patina, I will try and preserve it myself. And one of them's with the conservators. And we've studied like what documentation you need to keep and things like that in order to be able to keep such works. So, and there's also, they've got RFID tags in because all the documentation is incorporated in there. And we're exploring things about how to use blockchain technology in order to keep the provenance and conservation data safe for the artwork. So rather than a sort of commercial use as these crypto art works, if you've come across this at all, and using blockchain technology, this is using it in a different way through the smart contract stuff. And I'm also working on this project called Collateral Effects. This is just a work in progress and looking at the collateral effects of the pandemic. So things like TB diagnosis went right down during the pandemic because the symptoms were so similar, and that's going to cause a huge problem. A lot of antibiotics were used much more. So somebody said to me, I mean, my collaborator, one of my collaborators on the project said to me, it's like throwing a bomb at the issue of antibiotic resistance. But then we've had other situations where people just not going out as much has kind of reduced it on the other side. So studies which are based on the fact that people are catching things are also delayed. And then there's this huge knock-on effect of the pandemic on basically stopping all the other research for a long time, and people are just getting back into it now. So just to conclude, I mean, we're still facing a lot of existential threats at the moment. I think this is true. We've got the issue of antibiotic resistance. We've got the pandemic still going on and needing a lot of attention. And we're also facing this huge issue of climate change. And we started about 2 years ago, we started to collaborate with Professor Ditard Matanovic at Boku University in Vienna, the University of Life Sciences, in the Institute of Microbiology and Microbial Biotechnology, looking at yeast. So another different microbe here. And through this project, we developed a series of 3 works. So I collaborated with Alex May again, and we developed this series of works. And And we've developed a work that points towards a possible solution to climate change. It needs a lot of work, obviously, but it's something that biotechnology could be useful. So we're very much involved with investigating synthetic biology now, as well as a solution, and also in terms of being aware of potential future risks. So I'm just going to now show you a short film. Just let me turn over to it. Oh, what do we can't access the optimized video for video clip file thing. So we're going to try and share it, but if you find it's not working, maybe you can stop it and readjust this, because it doesn't come up like it normally does. So we're just going to show this, but I hope it works, because we're unable to access the optimized for video clip. For some reason the video is not showing the subtitles, so I'll explain what you're looking at here. This is a piece that we made that physically contains and explores a yeast that is modified to capture carbon. So it's a Pichia-Pastoris yeast that captures carbon from the atmosphere. And the scientists at Boku are also working with another yeast that could, a Saccharomyces cerevisiae, that could capture, that could take food that yeast eats and output lactic acid as a result. And they were slowly evolving it to become more tolerant of the yeast, of the lactic acid. And what we thought would be interesting is if we could put the two together, and we could have a yeast that would capture carbon from the atmosphere and output lactic acid, which you can polymerize to make PNA. And so this blob that you can see here is the important one, because this actually contains the lactic acid producing Pichia-Pastoris that basically eats carbon and outputs lactic acid, which you can polymerize to make this 3D printing filament. So you can make kind of as much plastic as you like, and it would, in theory, kind of drop back down, because it biodegrades quite quickly and then recaptures the carbon. Another piece that we worked on is called the bioarchaeology of yeast, and it kind of alludes to this this thing of appreciating the microbial life on antiquities. And so there were scientists at Boku who were studying this side of things and looking at these what they call black yeasts that contaminate kind of marble statues and things. And they're absolutely stunning, these yeasts. And so, working with Alex May, we photogrammetry scanned these objects, these tiny, tiny little yeast colonies. And these objects that we've made, these sculptures, are casts from, we kind of made a 3D model from the photogrammetry scans and 3D printed it, and then cast them in the materials that would be their ideal substrate. And so these are now going to be grown with the yeast in culture. So the yeast will kind of go into every nook and cranny and actually pattern it. So it's sort of that moment when a microbiologist in the lab, and they're just like, this is the most beautiful thing I've seen, this blob in a petri dish, which they are completely gorgeous and magical. And we wanted to kind of take that out of the lab and share it with the audiences, the arts audiences, and talk about like maybe seeing the world from a microbial perspective, sort of the bacteriological or yeastological point of view, if you like. And another work in the series is called Cult, with Alex May, who works a lot on this And this is based on the idea that culture evolved through yeast. This is a kind of sort of an idea, the drunken monkey hypothesis, that basically the yeast that produced alcohol managed to proliferate around the world because they got us to help stopped being hunter and gatherers, and we started to form cities, villages, and then cities and settlements, and build and build and build on this. And so this is, I think, based on the golden section. So it's talking about the crowding and the growing up of cities and things getting smaller and smaller and more compacted. And the thing which is based on the kind of cityscape, which is based on architectural models, is covered with breadcrumbs, but they're special breadcrumbs because they're from the lab at Boku, cooked, and then the yeast was killed, and then we kind of ground them down and applied them to the artwork. They are a Pichia pastoris yeast that's capable of making bread rise and fermenting beer and wine. Because Oscar Attar, one of our collaborators there, has understood the mechanics behind this and been able to kind of produce this yeast, this non-fermenting yeast that can now ferment. And they're actually exploring ways to develop this, and maybe it's got different flavors, maybe it has, you know, it's interesting in other ways to us. So and just coming to the end of this. And so this project, we're going to be showing it next year at various venues. So keep a look on my website and keep a look on my social media, and you'll see more. Just come back to my social media page.
Yay!
Thank you.
Thank you, Anna.
So if you want to follow me on social media, these are how you can follow me, or if you want to get in touch, there's my email, my website if you want to see more.
Great. Such an amazing talk. I mean, there's so many dynamics, so many layers, so many techniques involved, and just so many collaborations that you managed to talk about in this kind of 40 minutes. So thank you for that. I guess we can answer 2 questions really quickly before going into our next speakers. And one is, where do you get the inspiration behind all these creative pieces that you do?
From the scientists and from research, really. I'm obsessed with like learning everything from the scientists. So I always say, like, to Dr. John Paul that I mentioned there, I always told him he was my muse, because it's like everything he would tell me, and he'd say, well, we had a conversation a few weeks ago and now it's an artwork, you know, because that would inspire me. And then on the other side of it is looking at the history and looking at thinking about the future. So I always do lots of research into the historical background to these things. So I find the history of microbiology absolutely fascinating. So there's that like link and that thread across time from the past right up to the most contemporary, cutting edge and future thinking kind of research. So all those things are kind of my inspiration. And I just love microbiology, really.
Cool. And then, like, how much time does it usually take you, from coming to the idea to actually presenting a piece into a museum, or how much time, or what's the time frame involved in this?
Well, it varies. So I kind of look at my works as never finished. So I'm always up for kind of collaborating and taking that next step. But a lot of it's controlled by the deadlines that the museums and the kind of the funders have as well. And then you have things like pandemics in the way, which affects things again. So originally the yeast project was supposed to be a sort of 9-month project, but it became over 2 years because of the pandemic, and we weren't able to go there and things like that. The sort of some projects have a short time frame of 3 months, and then some projects I'm working on for years and years and years. Like I've been working with Modernizing Medical Microbiology, who also oversee the Cryptic project for like 11 years now. So it can vary.
Yeah. All right, I guess we'll wrap up the session. Again, thank you very much, and I hope we'll be able to visit live one of your collections. I guess a lot of them are here in the Netherlands, so that's a good time for us, Joe. But yeah, thank you very much, and good luck to all your creative projects.
Thank you. Bye.
Bye. So we have in the waiting room 3 of us speakers, which are going to be joining us shortly, and we are going to talk about 2 different projects that we've been working as part of International Microorganism Day, which is Women in Science, Women in Microbiology, illustrations, showcasing all the hard work that it's been done by women in microbiologists, that sometimes it's difficult they don't really come out as credible pieces. So I'm in the very happy position to welcome with me Mitra, who has worked on our blog last year, 10 women microbiologists you don't know about but should. We also have with us Veronica. So thank you very much for the last minute joining. Who she actually worked on the illustration part of the Women in Science and Women in Microbiology. And we also have Ben, who orchestrated all of this collaboration and helped finalize the project. So welcome you all. We're so happy to have you here.
Thank you. This is great. This is the first time we've actually all met. At the same time, so this is kind of maybe a symptom of COVID times, but it's an absolute pleasure.
Yeah, I mean, there's just so much to talk about in this session. I don't even know where to start, but I guess we could just maybe take a chronological view now of how things started. And I guess the first thing was the blog on our website, which you can find. So I'll maybe hand out first to Mitra and Ben to just give us an overview of your inspiration, how you actually wrote the blog, the illustrations.
Yeah, so super happy to be here. Thank you guys for inviting me, and awesome to see both you, Ben, and Veronica. Veronica, awesome job on the illustrations. It was so good. So I got the inspiration for this blog, I guess, last year's International Microorganism Day. I got in touch with Joe, and we were bouncing off blog ideas, and one of them was to talk about some lesser known microbiologists. And I was like, okay, I know Leeuwenhoek and I know, Louis Pasteur, who else do I not know? And I was like, wait, I've never learned really about any, marginalized, especially any female microbiologists in any of my textbooks or school curriculum. So that was pretty much an easy start for me to start researching and to find out, who are some of those unsung heroes, heroines, I guess, of microbiology that oftentimes worked in the shadows of these really revered male scientists. So there's a decent amount of research on women microbiologists out there, although it is a little bit hard to come by. So the ones that I found, I highlighted in my blog that I think Joe is scrolling through right now. But I always like to mention that it is in no way an all-encompassing list. There is still so much out there. And there's still so many, marginalized scientists that remain to be highlighted, in this discussion. Microbiologists that really set the groundwork for the work that so many scientists are doing today. So yeah, I'll leave it there.
Mm-hmm. And I guess should we pick one and just say a little bit of history behind it, as we're just, which one may be the most striking or example of a woman in microbiology, which we should mention?
Yeah, I have one up here. It's Elizabeth Bougie. And the reason I like to highlight her, I don't know if I want to pull her up on the blog, but she was an American microbiologist and biochemist, and she helped develop or discover the antibiotic streptomycin, which is used against Mycobacterium tuberculosis. So she discovered this in a soil culture, and she was helping these male scientists, and I have his name up, Professor Selman Watson. So Professor Watson ended up winning the Nobel Prize for Medicine for this discovery, but they kept her name off the patent, and they didn't give her the recognition when they won the Nobel Prize, because they assumed that she's going to, one day get married and have a family, and so that recognition wouldn't really be important for her, and closed that book. So I like highlighting her because this basically sets the tone for what a lot of these women microbiologists, women scientists, and historically have gone through. It's just doing a lot of the legwork and the heavy lifting, and making these really amazing scientific breakthroughs, but just not getting the light of day for the work that they've done. And similarly, I could also highlight, I think, one second, Esther Letterberg too. She helped discover the lambda phage, which is the phage that is the virus that is specific to E. coli. But similarly, she worked in the shadows of her husband. They worked in the same lab together, and also didn't get a lot of recognition for the work she was doing. And so this theme plays out across a lot of these women scientists that I've highlighted in this blog, and surely so many more that I haven't been able to highlight yet.
Mm-hmm. Yeah, I mean, thank you for all these stories. Maybe let's pass to Ben, just giving a voice to everyone of you guys in this call. So, how was your involvement in creating this, the blogs and the illustrations, and can you identify with what Mitra said before?
Absolutely. I mean, so my involvement in this was absolutely minimal. I mean, it was Mitra who kind of got everything together and produced this amazing blog post that I think everyone who saw it kind of was really amazed by. And my involvement was basically kind of finding Veronica and asking her to kind of put some color on these amazing stories. And I think during the whole process, I definitely can't take credit for it, but I'm happy that it went ahead, because I think it's taught me a lot about the history of the subject that I'm involved in, and really taught me how little I know about microbiology and history, the history of microbiology in general, but then especially the stories of these amazing women who have contributed so much to the field. And like, as you said, Mitra, the story really does repeat itself, that they kind of get zero credit, despite having contributed so much under very, very difficult circumstances. And the project kind of that we did actually involved us kind of revisiting the list and kind of finding more. And I think that was also a great experience, kind of really looking at the list and really trying to find these amazing scientists who have been buried by this, the kind of circumstance that they found themselves in. It kind of made me really want to kind of do more and continue.
Mm-hmm. And Veronica, I guess we could get you involved in this question. So yeah, can you just tell us a little bit about what you did, and maybe we can also show the illustration that you did on the screen as you are walking us through it.
Yeah. Hi, everybody. Thank you for having me here. I think it's really interesting for me because I'm completely outside of the science field. I'm an illustrator and designer. So learning about these women was really interesting for me. And it's almost like the way that I composed illustrations was on the basis of discovery about what jumped out at me about each one. So I guess I'll use this analogy. When you're putting IKEA furniture together and they tell you, don't tighten all the screws until the very end. It feels like this to me, because I had to put them together, and we have this color palette that was pulled out of the event, and it's a limited color palette, but having to make each one unique and different with all of their environments and components. It was a lot of composing and then coming back to it. I'd do 2 more ladies, and then I would come back and then say like, oh, this one they all need to feel equally special, to me. Mm-hmm. And so I needed to add more to the environment, and it was a lot of revisiting them And even like going back to the very first one after I finished the last one. But it was really incredible to learn about all these women and their contributions, especially I think for me, it's always surprising when I see all these also women of color that are brought in, like from a very long time ago when they must have had such an uphill battle. So it was definitely an honor to honor them in this way.
Yeah, and I just want to say these illustrations are up on the website right now. So yeah, you can go and check them out and share them, and make sure you hashtag International Microorganism Day and Veronica as well, and make sure you're using the blog. But yeah, Joe, should we maybe talk about like the science behind it, or the color palette of the IMD? Yeah. Okay. Questions.
Hey, yeah. Hope you can hear me okay. Yeah, Veronica, I just wanted to ask some questions about like the process behind making these images. So,
Yeah.
I mean, is it all digitally created? I mean, did you have to go out and find physical because lots of these images have such good texture in all of the different colors. And I wondered, how did you get all of these kind of effects into your designs?
Yeah, so the way that I compose them is I had a lot of help from Ben in sourcing images, especially, if you're talking about somebody that lived 100 years ago, the quality of the image is going to be very different than somebody that lived 20 years ago when we had photography. So sourcing these images, and then I would recreate the elements of them in Illustrator, which is a vector-based program. I would bring the shapes into Photoshop and I would colorize them there. And then I have a set of brushes that have a lot of grain in them. And it's like painting almost. I don't know if you're like, it was like literally like painting. You would fill in, you'd create shadows and textures that way.
And do you have a touchpad and pen, or were you just using a mouse very carefully?
I'm very old school this way. I'm using a mouse. I've really tried to adapt using some sort of I have an iPad, I have a Wacom at my disposal, and I just always come back to the mouse.
Yeah. Well, if it works, it works. The other question I had was, you have these lovely microbe backgrounds. So you've got some diatoms and some red blood cells, perhaps, and some chromosomes. Did you create all of these individual images by hand, or did you go and find them in other sources, or bring in images from photographs or something?
Yeah, so I created them by hand by referencing photographs. And I tried to do that so that each one was different and it wasn't just like a step and repeat pattern.
Cool. And yeah, I mean, they really are like a lovely quality. And I think, we've got to this point now where we have the blog and we have these wonderful illustrations. I think the next step is basically trying to sort of create a nice biography to accompany each picture somewhere or another. So I think in the next year we can perhaps, maybe with Mitra's help, perhaps Veronica, if you want to help us find some volunteers as well, we can get together a team of people to write little biographies behind each scientist, and then we can complete this page, because I love seeing all of these faces and I just want to sort of find out more information about each one as they lie here on this webpage. But it's great to get to this stage and have all these beautiful illustrations. And I think, Ben, we were thinking, these are smaller resolution versions, but we were also going to put up perhaps the full resolution versions as well, or link these tiles to them so you can download the full high-res version, and even ask for prints to get made or something. Yeah.
That would be great.
So
I think
you can see.
Oh, sorry.
Oh, sorry.
Oh, go
I was going to say that you can really appreciate the texture because when I see these pictures, I'm seeing them so small, but when I'm working on them, I'm seeing them at a full screen resolution on my computer, and you really appreciated them. Sorry, Ben.
No, I was just going to say, I really want to print them out. I think one of the really exciting things for me about this project, it kind of reminded me of a poster that we had when I was kind of studying, that was basically kind of microbiologists, famous microbiologists. And in terms of diversity and representation, it left a lot to be desired, and it was a little bit depressing. And so having something like this now and having kind of made it look so nice, you know, it's something that you can definitely print out and kind of put on your wall and kind of, realize that that microbiology is for everyone.
Nice. And I think that your blog, Mitra, is still the most well-read blog. And I think it's had twice as many views since we last chatted. So I think it's, beyond the 10,000 mark now.
That's wild.
Which is wild. I think it's even more, it brings more people to the website than the homepage, which is, amazing because it's going to be bringing in so many different kinds of people as well. I'm just going to pass the microphone back to Eleni because I think we've got 2 minutes right before the next section begins. Okay, nice. Cool. Okay, then we've got a bit more time. So, Veronica, I know that this isn't the only Women in Science illustration project you've done. So I think there was another one that you might have done, and I wanted to ask you some questions about that. How did that one get brought to you? And could you tell us a bit about it?
Yeah. So I was, I was working at Google at the time, and I was working specifically for like a section of Google Earth where they have a blog, and they did a section on scientists, specifically like astronauts. It was women in space. Actually, it was women also.
Okay, so
I started doing portraits of science. It's a very niche art field.
It's a good niche. But you've now done, women in the largest possible space and also women looking at the smallest organisms. So you've got both ends of the scale, I suppose.
Yeah, I love that. I didn't realize that was the connection, but yeah. These are slightly different though. The other ones, I feel like I challenged myself more in this project because in the previous one, I didn't give anybody any facial features. If you ever go back and look at that project. But for this one, their expressions are so endearing. I felt like it would be a shame to not include it. Gives them so much more personality.
Yeah, there's such life in each of these pairs of eyes, kind of like staring at you with this scientific intelligence, which is a really nice feature because they're quite simple images as well, but they have that kind of extra depth, even though they don't contain too much kind of like heavy detailing, but you capture all the kind of facial characteristics very simply, which I really liked.
Thank you. It was really important for me also to not have compositions where they're all with microscopes,
Yeah. I mean, we have our cliched microscope here, but I see what you mean. Yeah. Because that is the kind of like stereotypical image of a microbiologist.
Right.
Yeah, I'll add that, the pictures I used for the blog are you kind of feel like you're reading a textbook at some point because they're just these old black and white images, and seeing these just brings so much character and life to all of their stories. So, more science and art is where I'm standing right now.
Yeah.
Or more art and science.
More art and science, but wonderful to hark to all the scientists here. But yeah, so I think as we go forward and we try and build the biographies around each of these scientists and link in all the high-def images, Ben, you were mentioning that there might be a way in which we can add in a thing that would allow you to print off any version of these you wanted and post it to your house. I think that was your mad scheme for the next step.
Yeah, exactly. I'd like to kind of set up like a print on demand or something that maybe we could have on the website, so we can design some of them as individuals or as posters so that people can just print them off as they want. So when we do that, will definitely be added to the website. I was also kind of thinking before we finished, like talking to everyone listening, like so the blog kind of developed now into the series of 10 illustrations that kind of includes a few different names. And I kind of really want to know who we missed, like who should we have on a list, because I don't feel like stopping. I feel like I want to learn more, and I want people to kind of, shout out their favorite stories, their favorite heroines of microbiology, like today and in the past, because it's so important to learn, and it's fascinating.
So is anyone, Mitra, on the list that you provided that we didn't cover with an illustration who you think should be next in line?
So there are the few that are on the blog that are in this illustration series. But as Ben said, I mean, even beyond like this handful that we've covered both in the illustration series and in the blog, there I'm sure are many more untold stories. And I particularly am now interested in digging up as much as I can on more both women of color microbiologists or that contributed to microbiology, but also a lot from the LGBTQ community. I'm sure both of these groups have had hurdles upon hurdles in addition to being either women or in any other marginalized group trying to do the science that they're doing. So I expect the research to be a little bit difficult and hard to come by, but I'm excited to look into it. And as Ben highlighted too, if there's some avenue of maybe it's on Twitter, social media, or through this this live stream, if people can shout out like other microbiologists that have not gotten the recognition they deserve, if they can add that to our list and spotlight them, that would be very helpful.
I think that's a great idea. I think we also have next year, before IMD, but in a few months, there's LGBT and STEM Day. So maybe we could aim to get the blog ready in time for that day. That would be a nice kind of like timeline. Yeah. I'm just trying to find which day it was, because I remember it was earlier this year. I think it might be in March or sometimes in April. But anyway, we'll find the date and we will put that down as a good release date for the next blog.
Awesome. Good stuff.
Cool. Right, Eleni, you got 5 minutes left. Anything you want to wrap up with?
I guess we can highlight a few more microbiologists, I can say, because we do have another 5 minutes. So I guess that would be a good time to highlight maybe two more women microbiologists that you covered in the blog, Mitra.
Sure thing. Yeah. We can do Jessie Isabel Price. I thought she was really cool. So she's the one that's holding the waterfowl, the duck, right now. I don't know if we want to go to the blog too, but I can just speak briefly about her. She was a veterinary microbiologist. She was an African-American veterinary microbiologist. And she helped actually develop vaccines that fight against bacterial diseases in waterfowl and commercial ducks, and received her PhD from Cornell. And then she went on to develop, I have the name here, a vaccine against Pasteurella antipestifer. I totally didn't butcher that name, virus. And that's again found in ducks and waterfowl. So it was a really niche field that she was in microbiology and in veterinary sciences. Definitely kind of unique work that she was doing, and especially for her time, again, she's one of those scientists that would not only have been dealing with being a woman microbiologist, being a woman scientist, but also being an African-American woman during the historical context that she was in. So kind of had like that multifaceted double hurdles to overcome, and yet she did really groundbreaking work in her field. Another scientist I can highlight is June Almeida, and I don't know if she wasn't in our illustration project, but I think she's somewhere. She's going to be pretty relevant to our times. I think she's probably closer to the bottom, maybe. There she is. So she actually never had, June Almeida was a Scottish virologist, and she had no formal education, like no formal, science degree or, PhD, let alone like any formal education. But she was a skilled technician, and she was actually the first person to visualize under a microscope a coronavirus, among other viral structures. And so she did that at the Ontario Cancer Institute in Toronto, Canada. And then she went on to identify and image and diagnose many other viral structures. But whoever, or if you all are wondering who found the first little spiked, virus, coronavirus, it was June Almeida.
Okay, thank you for that. I guess we can wrap up the session and just say thank you for the three of you that have joined. Or maybe I can say, do you have any last things that we could say or mention in this last 3 minutes that we have? No? Okay.
All I would say is thank you, to Mitra and Veronica. It's been a pleasure, and I hope that we can kind of continue exploring this. It feels like there's a lot of good threads to pull at now, so we can hopefully keep going.
Yeah, and it's been great that you can all of you guys, this project brought you together, just combining the blog, the illustrations, the managing of all of this. It's just so nice that you were able to come together and maybe develop some new projects in the future. So yeah, keep an eye on our website or on your social media accounts. And yeah, thank you so much. Have a good International Microorganism Day wherever you are in the world. And yeah.
Thank you.
Thank you,
Yeah,
thank
thank
you guys
you.
so much. It was
Cool.
so fun working on this. Thank you.
Amazing. So we'll say goodbye to the three of you guys, and then we'll bring up to stage. Bye, guys.
Bye. Thank you.
Bye.
We bring to stage Jeff Hunt, which is part of ASM, the American Society of Microbiology. And also we have with us Danielle Snowflake, which they're gonna tell us some cool facts about agar art. And also we are hoping to get a live agar art demonstration. So stay with us. It's gonna be a very interesting and unique session. Hello, Jeff.
Hey, Lainey. Hey, Joe.
Hey. How are you both today? I guess it's really early for you in the US.
Maybe for Jeff. It's not that early for me. I have 2 kids.
Great. I mean, we have half an hour to our to discuss about the agar art competition, about ASM. Doing a live demonstration as I was hearing. So maybe I'll give the stage to you guys, and maybe you can just introduce yourselves and then give your presentation or the demonstration.
Absolutely. I first have to say I'm really jealous of your guys' seating arrangements there. It looks way more comfortable than where I am. So bravo to that. Great. So hi, everybody. My name is Jeff Hunt. I manage public outreach for the American Society for Microbiology here in the United States. I'm based in Washington, D.C., and I'll be doing just the introduction, but the main presentation here will be my friend and colleague, Danielle. So Danielle, you want to introduce yourself?
Hi, I'm Danielle Snowflake. I am the Senior Director of Education at Edvotech. We are a biotech education company that works to bring biotechnology and microbiology into classrooms. We want to make it safe, we want to make it reproducible, and agar is a great way to bring microbiology into classrooms and into outreach events. So thank you so much for inviting me. I'm really excited to be here.
Cool. So I am just going to start with a couple slides just to talk about sort of agar arts in general, and then we'll get to Danielle with the demo. So I know Anna talked a little bit about agar and its use in artwork. So from our perspective, when we talk about agar arts, we are talking about the practice of essentially drawing microorganisms onto agar petri dishes and creating works of art. And this has been going on for centuries, sort of unintentionally. The first sort of recognized agar artist has actually been Alexander Fleming, who obviously discovered penicillin, but was also sort of an artist in his spare time. And he was one who really sort of first popularized this practice of really drawing on agar pieces. And there's a whole history of Fleming and his works that you can really dig into. So that was sort of the initial inspiration for what the American Society for Microbiology has run, which is a contest for creating works of agar arts. And this is run since 2015. It is an international contest, and we are currently running it right now. But as you can see, we get some really amazing works of arts that people create simply using microbes, different colored microbes, differential media, and different agar plates. And I'll include a link at the end of this where you can check out all of the entries that we've had over the past couple of years. But this is really a great way for our members and really anybody who's interested in this sort of science art interface to express themselves and to sort of showcase sort of that artistic side of science and say, you know, this is scientific, obviously, but this is a way to talk about the work that we do in a way that isn't just a presentation or a slide deck, ironically, that I'm showing in a slide deck. Just to give people a sense, this contest is open for people around the world, and we've had submissions from countries around the world. I think we're up to about 150, but we'd really like to get that to everybody. So if anybody from, I don't know, Morocco is watching and wants to submit, you know, we'd love to add a little check mark to our map here of the submissions that we've received over the years. So just some basics, if you're interested in participating in the contest, all that you need to do is create your work of art. We ask for a description of sort of what the artwork is, because these are publicly available. We make them available on our website as well as on our social media pages. And then we also ask just for technical information, because we want this to be an opportunity for future or sort of burgeoning artists to be able to sort of replicate. We're really looking to expand who is involved and not just limit it to those who are microbiology students or researchers, but really anybody who has the interest and the access to the components that you would need. So just to point out, we do give cash prizes for the winners, and these are selected by a panel of judges who we recruit every year. So we have first, second, and third place prizes, and you're seeing the ones from 2020. We also have a People's Choice Award, which is voted on by our Facebook followers, where we post on our page. There's also substantial news coverage of the contest every year. We get coverage in international publications, National Geographic, Smithsonian Magazine, but from countries really all around the world. Just, you know, these are really pretty pictures and people like to look at them and write about them. So if you enter, you have a chance to not only win money, but, you know, fame and glory and all of that great stuff. So our contest just opened up this week, actually, and we'll be running through the end of October. And if you go to asm.org/augurarts, you can get all of the contest details. But for now, I'm going to stop talking and turn it over to Danielle, because you're probably interested in how to actually create Augur Arts.
All right, let me change. So I am using a virtual broadcaster. So I'm going to pin myself. So everyone should see me big now, so that I can do the demo and show you some slides. So like I said, you know, we are a biotech education company, and a lot of what we try to do is create experiences for students, you know, that really let them engage with science. And STEAM is a big area that educators are looking to expand in their classrooms. And so we're kind of at the intersection of art, science, medicine, and technology is this science art. And so the use of the arts as a way to convey scientific and medical education, medical information is nothing new. And so Jeff mentioned Alexander Fleming. He is a big inspiration to us as well at Edvotech. So if we're talking about a K-12 science curriculum, most of the time when you talk about Alexander Fleming, you are talking about his development of antibiotics. But he was known to paint with microbes that we find in nature that are brightly colored. And here, as we can see here in the slide, you know, there was this great photo, this great figure taken from a paper. It was actually about an experiment when they were analyzing unicorn poop. But I just love this picture because it shows the wide variety of colors that bacteria naturally express that we can produce from living cells. But one problem that Fleming did run into in creating these works of art was that many of these microbes preferred to grow in different media or at different speeds or even different temperatures. And there's a quote in an article, it's linked to on this slide. I don't know if we're sharing the slides anywhere afterwards, but it's from this article called Painting with Penicillin, Fleming's Germ Art. And what was said was that the paintings were technically very difficult. I'm reading. So the paintings were technically very difficult to make. Fleming had to find microbes with different pigments and then time his inoculation such that the different species all matured at the same time. these works existed only as long as it took for one species to grow into the others. When that happened, the lines between, say, a hat and a face were blurred. So too were the lines between art and science. And this is not a shock to me. There is a lot in trial and error and skill in both science and art. But most educators that we work with don't have the luxury of this time to really do the trial and error, but they want to do agar art because this is really cool for students. And so when we went to the drawing board to develop an experiment, we talked to our teachers. That's the first step that we take in developing new experiments. And their main concerns were cost, time, and reproducibility. Teachers have limited budgets. They want to make sure that the experiments and the exercises that they're doing work. And so they asked for simple solutions: inexpensive media, standard growth conditions, safe for classroom use. So many colorful microbes are not available for use in K-12 classrooms. And a high percentage of success. And they wanted these things to create their agar art. And so we decided to work with components that we had on hand that we already used in our experiments, like E. coli, which can produce white colonies, Serratia, which give red, Micrococcus that give yellow, and they all grow on LB media, but they do grow at different rates and their colors develop at different rates in our experience, which was giving us inconsistent results. And so we did go back to the drawing board in developing an experiment. And so we looked at what we had and we wanted to know what other microbes that we had that we were sending to teachers that could create color and allow us to paint with bacteria, but also be consistent and easy to use. And the answer came from our transformation experiments, specifically the plate that you see here on this slide, which is our rainbow transformation experiment. And in this case, you're transforming bacteria with plasmids that express chromogenic proteins under the control of a T7 promoter. And so this is great because the bacteria are all the same. It's all E. coli. They're all using the same promoter, which is inducible with IPTG, and they produce bright colors that are visible with the naked eye. And so by transforming multiple colors into the same strain of bacteria, we can make multiple colors of biopaint. And so what you see here is some of the colors that we can use in our experiments. And this means that all of your bacteria are going to grow at the same rate on the same media, which is going to simplify the process of creating bioart. They are going to, and so I like to think of this as a gateway to bioart. If you go to the ASM site and you see some of the things that people are creating, they're using many different colors, many different bacteria. These are going to be, 4 colors. It's a little more simple, but that doesn't mean that you can't make fantastic works of art. So this is one that was produced by one of the people in our lab. It's a wave with a sun, and these are all produced with E. coli expressing chromogenic proteins. And the yellow-green color here is actually GFP. It's an overexpression of GFP, and oftentimes we can see that as a yellow-green color on the plates. And so we can use it both as a fluorescent color and as a visible color. So this is what you want to get to, actually creating the art. So let me switch over to my hands camera so that you can see what I'm doing. So there's a couple ways that we recommend creating biopaints from transformed bacteria. While I'm getting my gloves on, safety first. Again, I'm using, these are all E. coli, so they are, they're K-12 derivatives, so they are safe, but we always want to practice safe science. So gloves, I always tell people you don't want to eat it, you don't want to drink it, you don't want to rub it in your eyes. But again, these are things that if you're teaching, if you're doing outreach, you're probably already using E. coli in some regards in your classroom. And so if we are creating the, bacteria from pre-transformed colonies, and I do have one plate here, which is just our transformation plate. Let me get to it. Oh, I'm giving away many of my results. Oh, good, I kept them off screen. Okay, so here you can see, here is a plate with some of our chromogenic bacteria. And you can see here that we have pink, we have purple, and we have blue on this plate. So these are pre-transformed. And if I were to make biopaint from this, what I would do is I would scrape the bacteria off of the plate using a sterile loop. I have some disposable ones here. You could use a reusable one if that's what you had. We're going to resuspend at least 5 colonies in 1.5 ml of LB media, which has ampicillin and IPTG in it, right? Because we want to make sure that we're not introducing. Another thing our teachers wanted to do was reduce contamination. So these plasmids all have AMP resistance, so our media is AMP resistant. So if your students are painting on here for a long time, most of the crud that's floating around in your classroom will not get onto the plate and disrupt your results. You're going to incubate your biopaint at 37 degrees for at least 30 minutes. And then you can use a loop, a toothpick, a paintbrush, a micropipette to then take these paint and apply them to the prepared plates. And so I'm going to demonstrate some of the methods using our Bacto beads. So one of the things that educators did ask for is that, this scenario is using biopaints from bacteria that you may have transformed yourself. We're going to talk about the one exciting innovation that we did put into this kit when we were developing it from our teacher feedback was that not all of our teachers are going to do transformation with their students. This is due to a lack of time, a lack of resources. Furthermore, we do work with outreach groups where they might only have a few hours of contact with the students. So we decided to create pre-transformed freeze-dried bacteria that can easily be reconstituted and used right as biopaint. So these are our Bacto beads. This is an innovation that Edvotech developed for educators. Basically, these are non-pathogenic, freeze-dried, pre-transformed E. coli that are easily cultured for use in the classroom. Each of these beads, so let me bring one out for you so that you can see it. So each of these beads is going to contain microorganisms, buffer, salt, and the nutrient broth necessary to sustain them in this freeze-dried state. And you can see here, it's a little pellet. If you're familiar with the ice cream of Dippin' Dots, they're about the same size and consistency as that. Because they are freeze-dried, but they are, each of them contains, millions of bacteria that are pre-transformed with our plasmid. And these are great because they're ready to go when you're ready to do the experiment. You don't have to worry about slants. You don't have to worry about plates. You don't have to worry about doing your transformation one day and then having the cells get kind of cruddy by the time you actually go to do the experiment. And most of these strains, when stored in the refrigerator with desiccant, are good for over a year. And so to prepare the biopaint from Bacto bead, and so here, these are our pink Bacto beads, but we also have purple, we have blue, and we also have GFP, which I don't have here with me today. So if we're going to use this to make our biopaint, what we do is pretty simple. So I have growth medium here that I've already supplemented with IPTG and LB. If you get the kit from us, it comes with the pre-measured amounts that you would use. And I am just going to take a pipette and I'm going to add 2.5 mils to the vial. All right. So I'm doing this at home. So I am using, these calibrated pipettes. You probably want to use something a little bit more high tech than this to measure. But again, these biopaints are pretty, they're pretty forgiving. As long as you get the bacteria in there, you get them resuspended. And then I like to incubate them for 30 minutes to give the bacteria a chance to kind of get healthy again after being lyophilized. But you can see, they're even pink here, because they were expressing a bit of the protein, before they were lyophilized in the first place. And so basically you would incubate this for 30 minutes and then start your painting. And so I'm going to do a little demonstration here. One thing I like to do is, especially when you're doing this with students, some of them might be a little nervous about creating work for the first time. They might not think they're artistic. They might not think they're creative. So this is actually a piece of clip art that I printed. I took a Petri plate and drew a circle around it. And so it's nice to give the students an opportunity to draw on paper first in case they're nervous about going right to the plate. They can plan out their artwork ahead of time. kind of get comfortable. If you want it to, we have the colors. You could even say, okay, I want to do this blue and I want to do this pink, and kind of plan out everything ahead of time. This is clip art. This is a little bug that I drew up for our International Microbe Day. So this is the one that I'm going to use. And we're going to start painting. And this process is it's fun. I find it relaxing. Every time I do one of these agar art demos, my lab loves it because they get to do some of the painting with the microbes. And it's as difficult or as easy as you want to make it. So I'm just going to do one color because I rehydrated the one color. And you can take the loop and just start tracing. And this is this is as simple as you can make it, for those who kind of are nervous or apprehensive about getting involved. And again, there's 4 colors, so you can explore with those colors, see what would happen if you had combinations of those colors. Do you get mixes? Do you get individual colonies? So I'm using the loop end. These, my particular disposable loops have the pointing ends too, so that you can easily trace and get a little bit more precision than the loop. Oh, I just stuck my finger. Good thing that these bacteria have ampicillin in them, because if there are any bugs on my hand, they will likely not grow on this plate. Of course, if you're in a classroom, you probably want to do this next to a flame or something, just to ensure that you are, keeping an updraft to keep any extra stuff off of the plate. Another technique that we can demo here is pipette. So I have this figure here. We do include a whole bunch of tips on how to paint with agar art. This is all freely available on our website under this kit. But, here's an example of the size dots that you can get when you're using pipettes to make some of your dots. So I got my pipette here, and I'm going to make some 5 microliter dots here to put, these little, let's call them plasmids or whatever, onto my little critter here. And that is, I have a pipette there. And that is as difficult as it gets, like if this is something that you want to try, you want to start, this is a piece of artwork that's done. I would let this sit here until all of especially the drops, until they soak into the media. So you probably want to let it sit here for 30 minutes before you're going to put it in your incubator. Let it go overnight to get the color to start. I actually recommend throwing the plates after the initial overnight incubation. I actually recommend throwing the plates in the refrigerator for about another 24 hours. At that point, I find that it helps the colors get really dark, really vivid, and then it will look great for your bio art. And so one last thing we found when making our works of art is that the actual plates that you're using, the quality of the plates is really important. So each Petri plate contains a solid LB. It is included with the kit. This has been in my refrigerator, so you can see that it's got condensation on it, and the label got a little messed up. But this is a solid LB agar. It's included with the kit. You microwave it. Once it's cooled down, you're going to add the IPTG. You're going to add the LB, just like with standard, LB, that agar that you would make in the lab. And then I like to let the plates dry on the bench for at least 24 to 36 hours to get really dry, get a lot of the condensation off of the surface of the media and off of the lid. Because if there is any condensation on the plates, your bioart designs are going to kind of smear and you don't get as crisp lines. And again, everything that you need with the kit is included. Pour the plates, let them sit for at least after you let them dry for about 36 hours, they can be stored in the refrigerator. And, then they're ready to go when you're ready to go. So after we kind of developed the experiment, We took this kit pre-COVID. We took it to the National Science Teaching Association regional meetings in 2019 to see how well our agar art system would work, because we wanted to test it in teachers' hands before we released it. And we had heard at this meeting that this was something that teachers really wanted to try. And what we found was the experiment was a huge success. So here you can see teachers at the meeting creating their bio art from our Black bead bio paints. We actually didn't have an incubator at this meeting. The teachers drew the designs on the plates on a Thursday. And so we found a warm spot in our booth and incubated the plates for 2 days. And so by Saturday, you could see we had really nice results. These are some plates on our True Blue 2, which is our transilluminator. And you can see the colors and you can see the glowing from the GFP bacteria, and it looks really great. And so let me show you some of the plates that our lab did. As I said, every time I do this workshop, our lab gets to do the results. I have a piece of paper. This is our True Blue 2 transilluminator. If you were doing GFP plates, you can use this to image them. I have a little note, True Blue. Oh wait, this is the way you can see it's a blue light transilluminator. You can see it makes my highlighter glow, but I also had that there to remind me to lift up that paper so I can start showing you some of the result from our labs. All right, so it's going to get really bright for a second. This is one of the works. We have a happy face. Here we have a sheep, and this is in pink. This plate broke. It got a little dry over the past couple of days, but this is someone, it looks like an ocean scene with an umbrella and maybe a beach towel. These were done by some of our lab staff. There might be some interns in there too. This is just a picture of the rainbow transformation plate. I love this because you can see all of the different colors, the green, the purple, the blue, and the pink. It looks a little bit like confetti. We called the initial kit rainbow transformation, but the other candidate was cupcake transformation because it kind of looks like sprinkles. Here is a flower, a fish tank. This is done with blue and pink. And so you can see, it is, and here is a little starry night scene. So we have a pink moon and then the colonies all around it in the different colors. And so really you're limited by, what you can paint. Here is a dog that's like pink and purple. So the beads were mixed and you can see that like the way the edges are, the bacteria grew in kind of an interesting way, that expresses both of the colors. And so these were just done in our lab. We have more examples on our website and also more examples. If you go to our YouTube page, we ran a whole workshop on how to do agar art with your students. And so just to kind of wrap up for now, basically incorporating art or design into your microbiology experiments can be as simple or as involved as you would like to make it. And so while I've been demoing this with our EdvoTech resources, these steps are going to be applicable to any transformed bacteria that you would use. So if you have, a chromogenic or fluorogenic promoter protein under an expressible promoter, give it a try and see how well it works for you. And so basically, I've shown you 2 ways you can do that. One way you can do it with pre-transformed bacteria in the context of bactobeads, and the other in context of pre-transformed bacteria that you might do yourself. And so with that, I think that we can take any questions, or if there's anything else you wanted to add, Jeff.
No, I think, like you said, we're happy to take questions in the couple minutes that we have left in our time slot. So Ben and Eleni, I don't know how those come through necessarily, but let us know if there's a way for people to get in touch.
Are there any questions in the chat or anyone interested in learning more? Or we just answered everything? So I know you said it's open, Jeff. Do you get to see the submissions ahead of time, or do you wait until you get them all in before you see what comes in?
Sometimes I take a sneak peek at what comes in, but usually there are so many that I'm just making sure that everybody's got their name sent right and everything. What I can do, I don't know how it will show up, is I'm just going to flip over quickly. So I talked about before that we post all of the finalists from our contest on our Facebook page. And I know Eleni's actually going to show the winners from the FEMS contest coming up in a few minutes. So this is just to showcase for folks from last year's contest. If you go to the ASM Facebook page, American Society for Microbiology, you can see these are all the finalists of people who submitted in 2020. And Danielle's lab team created those in a few days. I would say that people who are submitting to this contest do a lot of trial and error to get their submissions, because you can see that some are multi-plate like this one, some are multicolored. They get really intricate. And so it's pretty impressive every year to see what is created and sent into us. And I imagine takes a lot of work on the part of the artists themselves. So
but in the same regard, I would say some of the most expressive ones are done with one or two colors. So if a student I think planning on paper and then getting it on the plates is really important. There's one though that you've shown before where it's like a volcano, and that one is fantastic. It's like, it looks like there's scaffolding involved, and multiple different species of microbe that come together. And I just think it's really neat.
Here, I'll show the one that Danielle's talking about real quick. This one, right?
Yeah, that one is so cool.
Yeah, I don't know how somebody made that.
So Danielle, how do you get all the different colors in the
Yeah, so basically they are chromogenic proteins, so proteins that can produce color when expressed, and so they're under a T7 promoter. And so it is basically, there's no other gene in there, it's just the chromogenic or the fluorescent protein. And we just have it expressed under T7 promoter. So when you have it on a plate with IPTG, it turns on the switch and the colors are there. And so that's why, again, going back to the original Fleming works and others, they use microbes that have this endogenous color. And it's tricky to get them all to grow at the same rate in the same time. And so because all of our E. coli, once transformed, are going to grow at the same rate and produce color, it makes it easy because the only variable is color. So we have our blue biopaint, our pink biopaint, our purple biopaint, and a green biopaint. And then you just add the media, you paint them on, and they will express the chromogenic or fluorogenic protein because of the promoter system. So if you are an educator, let's say you're an AP educator, and you do GFP transformation, it's an inducible promoter. You can use this system to teach your AP lessons or your IB lessons because it is that genetic on and off switch.
Mm-hmm. Yeah, thank you for that. And I mean, this ties really well with our new education source that we have on the IMD website on making your own agar art from maybe easy to find materials. Yeah. So that is up on our website. So it will be very, very nice to see if some of the guys that are listening now, they can create some nice agar plates and then create art on them using that method. That would be really cool.
Yeah, I mean, we obviously make the reagents to send to educators who might not have an ultra low freezer or might not have the availability to do a transformation. So that's why we want to make it as simple as possible. As long as you have a warm place, you can just add the media and grow these cells up in your classroom. But again, if you are, even in a research lab, this can simplify things as well.
So I just want to ask some questions about the biobeads. So are there any colors that you currently can't get out of the system that you're trying to work a way towards?
Yeah, so basically we are limited by the plasmids we have. And so, we are currently, right now, the Bacta beads themselves, we do have them both pre-transformed and and untransformed. So our Bacto beads, we have E. coli, various strains that are good for transformation, but we also have Serratia, we have Micrococcus, we have Bacillus, a couple others that we use for experiments. The beads were actually developed to allow educators who don't have an ultra low freezer to be able to do microbiology experiments in their classroom. And then one day we were kind of just messing around and we said, what if we pre-transformed the bacteria and made them into Bacto beads as well? And it became another important reagent for some of the fermentation experiments that we do, where again, teachers might not be able to transform, but they want to do fermentation with their students. And so, you know, again, like this is a little, but you know, we're currently doing some research and development to have additional colors, perhaps for next year's kids. So, you know, keep an eye on this space.
So what color do you most want to get as part of your palette?
What color? I mean, I think that everybody has, I don't think it's one color. I think it's all the colors. So there is a beautiful picture going around for years of like a sunset scene with all these different fluorescent proteins. You know, you get a RFP, a GFP, a YFP, a CFP, and I think it's the combination of the colors that are the best for me. I will say that our production staff might not be happy with that because we do make all of these beads in-house, and you know, we might be making a little bit more work for them. But I think in terms of the chromogenic plasmids, I would love if my blue was a little darker.
Okay.
So we're working on that.
Yeah. But you do have a nice blue. You just want a particular kind of blue. Yeah.
It is beautiful. It's like a teal blue. I like imagine a darker blue that you could almost, I don't think there is any black chromogenic protein to really get those outlines. And I think sometimes with the agar art, it's nice to go back and have a sharp outline.
Okay, well, you know, this is all good fodder for your research and development teams to dig into.
If anyone in the chat has a color that they want to see, you know, put it there and we'll try and work on it.
Nice. Do you have pink?
We do have pink. We have a great pink. We have a pink. My favorite, I'll hold it up to the camera. It's actually in this purple. You can't really tell when I'm holding it up. It is super vivid.
Yeah, that is nice. I like the dog.
It's just fantastic. So, you know, here is a plate that has all of the colors that we are currently expressing. And you can see there's a pink, there's a purple, there's a blue. And the contrast actually gets better with time. If you leave them in the refrigerator for a couple days, those bacteria aren't growing, but they are making the proteins and they get darker and darker over time.
Wonderful. I think we've got just 15 minutes, right, for you to announce the winners of our microbe art contest. So we'll have to thank you, Danielle, for your wonderful demonstration and nifty camera skills as well. Because it's always good to see what you can push Zoom to do, and you've managed to get a bird's eye view of you doing two-handed experiments, which is great.
So I have been, shall I say, perfecting this system because we do run YouTube workshops for educators. We try to do them monthly on our YouTube channel, which is youtube.com/Edvotech Inc. And then we actually do one where it's an hour talking about agar art and microbiology. And so check it out.
Yeah, thank you of you both. I think we'll move forward to announcing the winners of our own microbe art contest. I just want to say thank you both. And if you do want to enter the competition, please do, because it's actually running now, Jeff. So when is the deadline?
October 22nd. And the
October
website
22nd.
asm.org/agarart.
So more than 1 month to investigate these nice colors and create a nice piece of agar art. So yeah, please do. Thanks of you both. And we'll move forward to sharing my own presentation. Thank you, guys.
Bye, guys.
Thank you.
So I'm going to see if I can share my screen. Okay. All right. Great. So yeah, our own third edition of the microbe art competition. It ran over a month, I think from mid-August to mid-September. We had just so many nice entries, and each year the selection becomes even more difficult. So it's nice to see all of your guys' creations. So yeah, this is just a short 10-minute presentation to announce really the winners of our competition. So I guess I'll go to the first one. Oh yeah, I prepared a few statistics just to keep the, adrenaline going for the winners. So yeah, the hashtag #MicrobeArt2021. Just a few data here to show you, how many impressions, over 1 million impressions over all the social media channels. We had over around 500 posts on social media, which is an even more is even more than last year. So, and then we can just see here in roughly which locations people have submitted. So around all the continents, which is great to see. And yeah, it's even nicer to see is that we have a good balance of male and female, which is
nice, yeah.
Which was nice to hear.
I think sometimes we get quite a heavy leaning towards more
female ones, yeah. So 50-50 this year. Okay, so I'll move forward to the winner of the best Eiger art, which is Stefania Rizelli, who submitted this beautiful art of bacilli in yeast isolated in dairy products. We particularly loved how colorful this art was, and I'm really eager to hear and see how this was created, because it's really fancy. It looks like different types of fungi for me growing outwards, or like mold and different colors. So yeah, congratulations, Stefania, and we're gonna be in contact with all the winners to supply some IMD t-shirts that we are wearing today. So we'll be in touch with you, Stefania, and all of the other winners. Then best cartoon, I really love this one, goes to John Chilton for the first online microbe E. coli for online, I guess. Yeah, I think it was a really loved cartoon across all social media, especially because E. coli is such a, it's a microorganism that if you work in a lab, you're definitely using E. coli.
Yeah,
so fundamental. So it's really nice. Moving forward to best illustration or painting goes to Tristan Young with his microbial landscape predation. And I just want to say, Tristan, do continue to do your art, because I've read your post and you are exploring this new avenue. So yes, please do continue making paintings, because they're really lovely. We love the colors. It was so unique in so many ways. So congratulations to you. Best photography goes to Ian Jones for his arrangement of diatoms in memory of Anthony van Leeuwenhoek, who did the first microscope. And we owe this day, International Microorganism Day, to his letter that was written to the Royal Society. Where he wrote about his first appearance of microbes. So it's a very, very nice arrangement and stack of different photographs, I assume. So yeah, congratulations, Ian. Okay, best physical piece. This one was interesting because it was the one of a kind, I guess, in this competition. We have really cool physical pieces this year, from crocheting, to glass structures of microbes. But yeah, this is a game called field, and it's a strategy board game having at its core bacterial behavior. So congratulations! I guess this game was made by a very international team, so it's not only one person that was involved in this project. So I want to congratulate you all. I'm not quite sure, but just I remember there was just 3 different institutions that did this game. But maybe we will be excited in getting in our hands this game and testing it. You know, that would be really fun. And I believe we have one last category, which is the grand prize, the best art overall, which was basically the creation that received the most unanimous votes. So the most votes out of all of us. I'm very sorry if I pronounce your name wrongly. It goes to Lieke van Gistenbeek for her lactococcus cell creations. I believe they are bacteria found in cheese. So yeah, really unique design. We love the vivid colors as well.
Yeah, we like the form as well, like the layout.
Yeah. I think, that should be the end of my presentation. I'll just stop sharing.
So do you know what the prizes will be this year?
We hope to be able to send you to all of you guys the IMD hoodies or t-shirts, right? Yes, which Joy is gonna display for you guys.
So we have. Yeah, so we have a wonderful classic white version of the IMD t-shirt, which Eleni is also wearing. And then we also have a stylish monochrome black version, which I think is like really funky as well. And you can see it's just like a nice black t-shirt here with the white IMD logo. And then we have these hoodies, which have the lovely IMD logo on the front, and then all 4 mascots on the back, which is great because we have, is this the new one here? Other side. Okay, so this is the new one, the microbe art contest, and we've managed to get it on the hoodie in time for IMD. So thanks to Eliza Wolfson as well for winning this competition and giving us Rizzo, the new mascot. And we now have this lovely set of IMD merchandise, which we'll be distributing to all the prize winners. And I guess we should also probably send a hoodie to Eliza, so she has a nice one with her new mascot on it. And yeah, well done to everyone who entered the microbe art contest 3rd year in a row. Do you have any? Yeah, what were the stats? We had millions of impressions of the microbe art contest hashtag, which was amazing to see. Still lots and lots of energy to put microbes and art together. And in fact, this session has proved beyond doubt that microbes and art blend into a wonderful combination, really. You know, we've had illustrations of women in microbiology, we've had microbe-based paints, we've had sculptures and installation pieces from Anna right at the beginning, you know, taking yeasts and bubbling in them in vials and eating away at marble to produce like beautiful colored sculpture. There's just so many things you can do with these living organisms to make beautiful, beautiful images, sculptures and pieces of art. So thank you to every scientist and artist who's come together here, because they often try and tell us that science and art are separate universes. But we don't believe this at all. We think they feed off each other and build off each other. So it's wonderful to have them together here for this lovely session. So we have a couple of minutes before we finish at the end. But Eleni has some more things to mention, so I'll just pass the microphone back to her.
Well, you know, I guess if you're still watching, I would highly encourage you to also watch another session that is linked with the science and art, which it's going to happen a bit late for guys in Europe, but for people in the US, that's a really good time. It's the Microbial Arts and Crafts workshops at 6 p.m. Eastern time, so USA time. But it's midnight CEST. But for anyone that cannot watch this session, then we are also going to provide the recordings to our YouTube channel at a later date. So you know, it's always going to be there for you to watch even if you miss it. But yeah, I think that kind of sums our session. We do have a vineyard tour or beer factory tour coming up. Maybe we're a bit ahead of time. Maybe we can have a small break until the time, or?
I think we can just chat about beer.
Yeah,
Oh,
chat about beer.
You could definitely do that.
So good to join us again, Ben. So I believe, if I'm correct, that you've arranged a live brewery tour in a few minutes, but not quite now. So we can just take a seat back and get ready for it. We've just had some coffees here in the IMD studio in The Hague at the Hague Humanity Hub. And would you mind telling us a bit about the brewery? What beer are we going to see today?
So that's a very good question. I've got no idea about actually what beers or what we're going to see there, but I know they've got a distillery as well. So it's actually not just beer brewing, it's distilling. And it's the first time we've done something like this live. And I think, it will be very easy to kind of pre-record this, but kind of for better or worse, I think we're going to try it live, and we want to get some interaction from people asking questions to Fergus, who is going to be showing us around his brewery.
Great. I mean, so we have Fergus. He's just popped into the waiting room. So shall I bring him on board? And then we can just talk a bit about the brewery name. So it's Adnams Brewery, I think. Yeah.
In the UK.
And we can ask Fergus when he arrives, but I have many questions about what kind of beers they make. So let's just check. We have Fergus Fitzgerald, our next speaker. And I think here we go.
Hi,
we
Fergus.
see you, Fergus.
Hello, can you hear me?
We can, loud and clear.
Excellent.
Oh, I can see the grey skies of England over his shoulder. I'll just wait a few minutes for it to stabilise. Yeah, okay.
Yeah.
We have you. Yeah.
Good. Okay. So I'm Fergus. I'm the production director at Adnams Brewery and Distillery in Southwold, which is on the east coast of England. I am going to be wandering around the brewery and distillery, and there's lots of metal knocking about, which isn't great for mobile signals. So if I do drop out or freeze, just bear with me and I'll be back pretty quickly. So I'm just in our distillery at the moment, which is here. And I'll talk a little bit once we get through about what ends up in the distillery. But as this is the most attractive view in Southwold, not with me in it, obviously, but I thought you might want to see where we are. So
You got a great lighthouse.
it's not bad, is it? There aren't many breweries with a lighthouse, so we're quite proud of that one. So this is the best view in Southwold until you get obviously into the really interesting things which we like to look at. So I'm going to wander down. Actually, what I'll do is I'll take you through the sort of start of the brewing process. So I guess most people know how beer is made in a sort of general sense, and most people are aware that it's turning sugar into alcohol. Strain on view since the 90s. We have changed it over time, so it's a mixed strain yeast. So we've got for breweries historically, because it was sort of a culture that grew up and sort of acclimatized to your normal brewery, and then it was just transferred from brewery to brewery and grown up. Our yeast was probably 4 or 5 different yeast strains when we had it in the sort of 1970s. The brewery had issues with the consistency of the beer, so they decided to clean the yeast up to turn it into two of the most dominant strains. So 90% of the sort of yeast was actually 2 strains. So they cleaned it up to leave just those 2 strains. And those are the 2 strains we still use today. So we do a lot of work in sort of managing yeast. And once I get through the brewing process, I'm going to hand you over to Connor, who's in our lab, and he's going to talk a little bit about what he does there to try and make sure we manage our yeast strain to keep those 2 yeast strains in balance. But going back to the brewing process. So in here, we've got some barley. So we use malted barley from this region. It's all grown and malted in this region, which is quite an easy thing for us to do, because where we are, it's surrounded by lots of barley, lots of wheat. So actually, there's lots of those malted barley quality or strains grown around here. And then it's also malted in this region. So we use different types of malted barley. Pale ale, which has got a relatively low color. We use some chocolate, which, as you might think, has got a slightly darker color to it. We use things called crystal malt, Munich, Vienna, which are all be more. The malting process turns that starch, or well, makes that starch more readily accessible to enzymes. So within those barley strains, there's an embryo, there's a starch supply. The malting process is really about getting that grain germination going. So it's making some enzymes, which will then eventually break that starch down to sugar, which is what we're going to feed the yeast actually ferment with. But the sort of process along the way involves drying it, and then they send it to us until we turn it into a mash. So I'm going to walk you through the brew house, and I'll point you at the vessels rather.
I think must be going.
So this is our
Fergus. I think we just lost you for a couple of seconds there. Maybe if you explained something, we missed it.
But I won't in the tanks. Taking that malted barley.
Fergus, can you hear me? We've lost the image, so we can't see what you're showing us. If you can hear me now. Did we lose Fergus altogether? Oh no, there he is. There he is. We're back. You're on mute. No worries, no worries. Great,
cool.
We're back.
Yeah, I think when you move through rooms, we lose you a bit. So if you just give us instructions when you're standing still, then we can follow the process.
Okay, I will do.
Nice.
Right, I'm just going to take you to a screen that sort of shows the brewing process, or a diagram that shows the brewing process a little bit clearly than my sort of waffling on. So this is our sort of description of the brewing process. I don't know if you can see that clearly, but essentially it's showing that malted grain is coming in, it's going through a mill, it's going into what we call a mash tun. So in the mash tun you have ground up grain and water. We control that mixture to a sort of temperature of somewhere between 64 and 69 degrees Celsius. And what that's doing is it's encouraging different enzymes to work at different rates. So what we really want to do is turn that sugar turn that starch into sugar. But we want to turn the sugar into a mixture of different types of sugar. So we want some complex sugars, we want some simple sugars. The reason we do that is because our yeast strain will use the really simple sugars. It'll use some of the complex sugars, but it'll leave some of the more complex sugars as unfermented sugar in the final beer. So that will give us some sweetness to that final beer. So we don't have to, as you might have to do in other sort of alcoholic drinks like cider, you don't have to sweeten back. You can just use the sugar that you've taken naturally from the grain. So our yeast strains, there are some yeast strains which will ferment some of those really complex sugars right out, so get rid of all that sweetness. But our yeast strain, our two yeast strains, they will leave a little bit of sweetness in there. So that's really where we set the final sweetness, and essentially the alcohol of the beer. So the more we turn those starches into really simple sugars, the more dry the beer, the more alcohol essentially it's going to make from that amount of sugar or amount of starch. If we try and have much higher temperatures, we tend to favor the enzymes that make much more complex sugars, and therefore we end up with a much sweeter beer. So once that mash has been sat in the vessel for about an hour, hour and a half, all the starch has been broken down to sugar by the enzymes. And again, this is enzymes that are just produced by the barley. We're not adding any extraneous enzymes. It's purely what they make. From there, that mash mixture, which sort of looks like watery Weetabix. I'm aware I'm talking to an international audience, so I'm not sure how translatable Weetabix is, but let's go with a normal cereal that's been watered a little bit. So it looks like that sort of consistency. And then from there, it goes into the lauter tun. In the lauter tun, essentially, it's a big sieve. It's got metal plates in the bottom with lots of little holes in it. This mixture of ground up grain and water is pumped on top of those plates, and then the liquid drains through. And that liquid we call wort, and that has all the flavor, all the sugar of the barley. From there, we move it to the kettle, and in the kettle, we boil it. We boil it for about an hour, and during that hour, we add some hops. So here are some, well, I was going to say fresh hops. They were picked about a week ago. They're so people know what hops look like. That's what they look like growing. when they're a bit greener, a bit more attractive looking. So we use hops to flavor our beer with, or flavor our work with. We use it for bitterness, we use it for aroma, and essentially they're a herb that we use to flavor beer with. We use lots of different varieties because all the varieties have different flavors. And then at the end of that boiling process, where we've extracted that bitterness from the hops, where we've extracted those aromas, we then cool it down to about 15, 16 degrees Celsius. And then from here, we put it under the road into our fermenting room. And that's when actually we start getting involved with microbes. Up until this point, all we've been really trying to do is create this sugary liquid to feed to the yeast. So it's really, really the next stage where we start to see the microbes. So I am going to have to start to move. And so if I lose you, bear with me. I won't be long before I get into the fermentation room.
Oh, we have faith you'll come back. Don't worry.
Cool. Yeah, exactly. Yeah.
So I'll walk through the brew house. So the brew house is what we call the bit of kit that essentially helps us extract that sugar from the grain and flavor with some liquid. And it takes about 8 hours per batch through this process. And at the end of that process, we have this sweet sugary liquid that we have flavored with some hops, but it doesn't taste like beer yet. It's very sweet. The pH is relatively high, so we're looking at a pH of about just over 5. All right, so we're now outside. So we have our fermentation room across the road. I don't know if we're still with me.
It's breaking up a bit. We can still see you though. Maybe it's gone now. It's frozen a little bit, but yeah. What are you thinking so far, Joe and Eleni? I mean, it's
yeah. I mean, I'm very much enjoying seeing the inside of the live brewery, and it's so huge as well. And
so this is our.
I think we're getting you back, Fergus, slowly but surely, but it's still a little bit stilted. I can't really tell what you're saying.
Different types of fermentation. Oh, I'm getting unstable.
We couldn't hear before, so the sound is coming back. So that's a positive sign, I think.
So I realize I have drunk Adnam's beer. So you found a brewery that I like in terms of the quality of the beer.
Nice. That's good to know. I don't know that I have, but I do. When you see the logo, it does look familiar, actually. Have you had it? You buy it from the supermarket or drink it in a pub?
I think I've had it in a pub. I think I've had it either in bottles or even on draft.
Yeah.
Do we know where the location?
Yeah, so it was in this. Where was the location again? Was it the southeast?
Yeah.
Coast.
Yeah, exactly.
Yeah. Okay, so I think we've lost Fergus for a second, although he's just come back. So here we go. He returns.
The suspense is absolutely killing me. I can't.
Yeah, here we go. We're back in the brewery. Now? Oh, you got to unmute. There we go.
Okay, I was just saying, tell it's live.
Yes. We can hear you now. I think we can see you. Gone back.
The stream's good now.
Yeah. So, see in there. This is one of our fermentation vessels. So the wort has gone in, which is all that sugary sweetness from the barley with the flavour of the hops as well. That liquid, then we check what we call the gravity, but essentially it's the density of the liquid, which we relate back to how much sugar is in there. Then we use our yeast, our house yeast, for almost everything we ferment, apart from 2 beers, where we buy in some other yeasts. Yeast goes in, and then obviously yeast does its fermentation, and you probably can't see quite well enough, but it is bubbling away a little bit down there. This fermentation is probably about 2-3 days old now. So typically we warm ferment. It's probably enough looking there. So we warm ferment for most of our beers, which means we ferment somewhere between sort of 16 and 23 degrees Celsius. So all these vessels up here, they've all got glycol jackets, so we can control the fermentation. Because our yeast will produce different flavors at different temperatures. Because it's a 2-strain yeast, it's quite flexible. So we can get quite estery flavors coming through. We can get lots of strawberry, lots of banana, lots of pear drop as well at those higher temperatures. So for those beers where we want lots of those fruit esters, we'll ferment a bit warmer to get those flavors. If we want a beer that's a bit cleaner, we'll typically ferment sort of colder, so 16 degrees Celsius with our yeast. It doesn't like to ferment very cold, so unlike lager strains or Saccharomyces cerevisiae or house strain, it likes to ferment a bit warmer, and it will slow down. It won't really ferment anything below 12. Anything less than that, it will sort of stop fermentation. So we sort of use those characteristics of our yeast, our house yeast, to sort of drive the flavor. So really, really fruity if we want it. Well, really high temperatures up to 23 if we want those fruit flavors, and 17, 16 if we want it drier, and less influence from the yeast. So we see every beer is its own recipe. It has its own flavor profile. So what we're looking to do is that every beer has something that is unique to it, and sometimes that's the malt character, sometimes that's the hops. For almost everything we use with our yeast, there is an influence of our yeast in there. It has some flavors in there. And one of our yeast strains produces a small amount of clove, which is unusual for most English yeast strains, but quite common in Germany and Belgium. They have a small amount of that clove characteristic. So we like that in some beers. We don't like it in others, so we play around with the temperature and the ingredients. There's Dan. It's live. So we play around with those temperatures and those characteristics to try and give us different flavors. Lots of people will use a single strain yeast. So a strain, usually lager strains, or sort of a very, very simple strain, which isn't really characterful. And we do use those strains for some beers, but we don't want our influence on those particular beers. But for the vast majority of what we do, we want our house character on it. We want our stamp on it. So I'm going to head back across the road. Hopefully I don't lose you this time. So bear with me.
We still got you.
I'm going to take you to our yeast room. So we do some public tours, and all this is on the public tour really, except when the CO2 alarm's going off, which they're not at the moment, which is good. But the place I'm going to take you to next is our yeast room, which I guess is pretty appropriate, but it's somewhere we don't take people normally. It's in the back of the brewery, sort of hidden away. But that's where we store our yeast and grow up our yeast for the next batch of fermentation. So you'll have to bear with me while I wander back.
You got a nice day though, beautiful blue sky.
It's not bad. There's the lighthouse again.
Ah, lovely.
So one thing I haven't mentioned so far about our yeast is we call it a top fermenting yeast. So breweries typically split. Sometimes people use top fermenting, but they bottom crop it. So essentially what cropping means is where you take the yeast off. So when we put the yeast in, as well as fermenting and making alcohol, it's also growing into more yeast. So typically we'll get 3 times as much yeast out as we put in. So with our yeast, because it's what we call a top fermented yeast, once it's finished fermenting the easy sugars, it likes to rise to the top of the vessel. And then we skim it off, which basically involves lowering a funnel down into the beer and vacuum that extra yeast off. And then from there, it goes back into our into our yeast room, where we store it at about 4 degrees Celsius until we want to pitch it into the next vessel. So we're essentially self-sufficient in yeast. Yeah. So this is our yeast room. So we've got some storage vessels here. So that's a storage vessel with some liquid yeast in it. So our yeast would be about 15% solid. The rest of it's made up of a mixture of beer and gas. Because obviously, as well as the fermentation, making sure it's also making lots of CO2. So that forms a lot of the slurry. So we'll store the yeast in here for maybe 2 days. We don't tend to like to store it much longer than that. And then within those 2 days, we'll usually have used it. So we'll weigh it into our weighing vessel. We'll have worked out the percentage solid in there. So we'll also do a viability test on it to see how much live yeast is in there. And that tells us why a very, very interesting calculation that no one seems to understand how it works. But that's how much we need to weigh to put into that weighing vessel, and then because it depends on the viability of the yeast itself. which is always changing how happy the yeast is, how healthy the yeast is. So we have a little centrifuge here, just helps us to spin down the slurry into its solid component, its gas component. And stain, so we use a methylene blue stain. You can use a violet stain, which is probably a little bit more accurate, but we use methylene blue because we find it's accurate for what we want to do. And that will tell us whether there's any dead yeast in there or yeast that isn't particularly healthy, because the cell walls of those yeast strains would have broken down enough to let the stain in. So we'll do a count under a cytometer, just looking at it to see what percentage of cells have absorbed the stain, and that tells us roughly what health the yeast is in. So,
nice. Fergus, I was going to ask you in there, like how many times can you reuse the yeast? Is it at some point you have to kind of start again, or is it just perpetual? Like you skim it off, you store it, and then you can put it back in as many times as you like?
So we run it probably for about 20 generations or so. So every cropping is one generation. It's complicated a bit because of this two-strain yeast we've got. One strain is what we call very flocculent, and one strain is not flocculent at all. And in combination, when they're roughly in balance, the flocculent one will pull out the non-flocculent one. Unfortunately, every generation, the non-flocculent one grows in percentage. So to try and keep on top of that, we used to wait till it got bad and then fix it. We decided we'd try and get ahead of it a little bit more. So we do a continuous culture of the flocculent one. So we're always growing. So we'll have, say, 7 hectoliters worth of the non-flocculent strain growing all the time. We'll pitch 3/4 of that into the next brew to keep the balance in place. And then we'll add some more fresh wort into that to grow that yeast up again. So we're always growing the strain that is dropping out all the time to keep us in balance. But we do then go back to the original strains, sort of between every, let's say 20 generations, we'll go back to the original strains again and grow that up from scratch. So we'll take it, we've got a yeast bank that holds our 2 yeast strains so they hold our yeast strains, they'll send it up to us, and they will grow it up from that up to a big enough size, which is probably something Connor might like to talk about. I'm just going to go and interrupt him, and he can talk about, I think that's probably a good place to cut in actually. So Connor will talk a little bit about how we take our yeast strain from those little vials up to commercial pitching.
Sounds good. Yeah.
Welcome, Connor.
Hi, Connor.
Yes, it's working. Thank you. Okay, so I guess I thought actually I will grab a slope for you to show you. Just into the lab. So if we were ever propping up, I've got laminar flow here, so we'll always do it in there. But these are the slopes. So we streak these up ourselves from the stored culture we get from Camden BRI. I don't know if you can. This is if you can see the yeast on there, but we'll take a small amount of zinc sulfate, which is 0.2 ppm, and we'll wash the slope off, and then that's washed off into a 250 ml conical, and then put on a shaker. It's into 100 ml, of wort as well. That shakes for 2 days, and then they get pitched into 1 litre of wort, just in slightly larger conical. And then that's just the simple part, but then we go up to the bigger vessel, which I have here. Hopefully you can see. So this is what we like to call R2D2.
It does look like a robot.
Yeah, it's a 25 litre. Well, it's actually, I think it was an old milk churn, wasn't it, Fergus?
Historically, it's a milk churn.
Yeah, it's a milk churn. It has been
adapted for beer use.
Yes. So from the shaker, which is over here, when we take off our propagations, they go into there, and then that goes down into the yeast room, I don't know if Fergus showed you.
We saw a fermentation vessel. Yeah.
And basically, that sits in for another couple of days. So obviously, the propagation has to work that way because if the cells just don't increase, they get to a certain point after a couple of days, and then for quorum sensing or whatever other reasons, we don't end up with any more cells. So that gets to about 25 litre stage. And then that goes into one of the bigger vessels you would have seen down there. We push in sterile oxygen and to push it into those big vessels. And then that goes up to the, how many litres is it, would you say, YPV?
So it's 4.5 barrels. Let's say 7 hectoliters.
So it's about 700 litres.
Wow, that's a lot of beer on the way.
Pitching with 700 litres of yeast.
Well, it was 700 litres. I pitch into 700 litres of work, then sits for a couple of days, and then we should end up with about, I think at the moment we're trending about 20 kilos of yeast when we centrifuge out. That then goes into top up because of our dual culture. So that tops up the balance basically, and then we pitch in probably what would you say we pitch in normally? Kilos.
Kilos, so we're about 150 kilos.
Yeah, so 150 kilos or around there.
Of just raw yeast.
Each fermentation. Yep.
Yeah.
Of each fermentation. So.
And you've got
we'll probably end up getting about 3 times that amount off it when we skim it off as well.
And the thing you said, you had top fermenting yeast, so it floats at the top of the fermentation chamber.
Yes, so that's
in that area, yeah.
That's where we do what we call skimming. So then at the right time, it's skimmed off, and then it's funneled back into those vessels. So.
And is that the same?
Use that for the next brew.
Okay, is that the same kind of yeast? So I know there's top fermenting and bottom fermenting yeasts.
Yeah.
Is top fermenting, which is the one that, so lager is one and ale is the other, I remember being told at some point, or was it a bit more complicated than that?
Yeah, it's a bit more complicated than that, but traditionally bottom fermenting is lager and top fermenting is ale, but really you can do either.
Okay, nowadays it's a bit more complicated. Okay, but so this would be, your yeast is perhaps more like a traditional ale strain than a traditional lager strain?
Yeah,
or again,
definitely.
Yeah, okay.
And I'd say ours is actually more familiar. I would guess it's more like a Belgian ale yeast.
Yeah, okay. I mean, we're here in the Netherlands, so we get a lot of those Belgian beers.
The profile we have on it, it's definitely more. It's not very traditional British, I would say, but that's why we like it.
Yeah, absolutely. And so I mean, nowadays you can go into things like ester profiles. Does that really inform the way that you make, the flavors of the beers that you're brewing? Is it very central to how you're thinking about the outcome of the beers?
It definitely is for us. For certain beers, we really want certain esters. So, like I said, on new on Cobalt, we're really relying on our yeast singing, basically, because there's not much else that goes into it. So, the idea is that you can really taste what our yeast tastes like in that beer, and it's really banana-y, and it might have a bit of clove on it as well, which is, again, it's more like a Belgian ale strain. You wouldn't usually create a lager with that either, but we do.
Okay, so you're mixing up a little bit. And what's the kind of highest strength in terms of alcohol percentage that you get to in your brewery? And does it start to kill your yeasts off, or do you not get that high? Because here they do about 12% sometimes when you get a triple or a quadruple beer.
Yeah, so I mean, we do both barrels, which are barrel-aged beer that is bottled broadside, but then with a bit of extra, some cherries in it and other stuff, but it's barrel aged for, I don't know, how long are we barrel aging that now for? Depends.
A year, but it's also
a year, but then it's blended, so it's, but that can get up, some of those casks, some of them will get up to 11, maybe, I don't know, the highest one we've had is probably around 11. Some others are only do 8, but I think our yeast tends to sort of tails off at about 11 at the moment.
Okay, gets a bit unhappy, a bit too drunk, and has to slow down.
Yeah, it really does struggle at that height, but up to 8 or 9, it will go very quickly.
Yeah,
Especially
okay.
one of our, because it's dual strain, class 1 strain, is it's really fermentable. I think attenuation is over 90% easily, and the other one's a bit lower. So when the balance goes out, the fermentation actually changes. So our final gravities will change depending on the balance of the yeast.
And do you have to? Yeah, I was going to say, do you have to be quite protective of your yeast strains? Are they your top secret strains, and do you have to make sure no other breweries get hold of them? Are you proud of special strains you've developed? Anything secret or
I wouldn't say ours is really a secret, because it's also a bit of a pain to work with, so no one else would want to work with it. So we don't have to worry about that. But obviously we do have it stored in multiple places, so obviously we don't want to lose it. That's the main thing for us. I don't think it's too dissimilar to probably some other strains, but the fact that we have this dual strain, that's what's really unique about it.
So what makes it so difficult to work with? Is it just the conditions, or does it have some weird characteristics that cause you difficulty?
Yeah, because it's two strains, and one of them is more flocculent than the other, and also one seems to skim better. It means as we go on, so as we go through more generations, it will tend towards class 1 rather than class 3. Class 1, the class is just means class 1 means it's non-flocculent basically, and class 3 is fairly flocculent.
So what does this flocculent word mean for someone who's uninitiated?
Basically it means that the cells clump together, so they'll produce on the cell surface membrane, and then they'll clump together. Obviously, and there'll be co-flocculation with the two classes as well, even though class 1 is non-flocculent. But that's the main reason why it's really difficult to work with, and because they're quite different, it will also cause issues in the brew house. So we'll have the fermentation changes, so the PG will go down. One's better at clearing diacetyl as well, class 1 is, but then we can't get the yeast counts down, which is really important for cask racking. So we're always trying to keep it in balance, even though it's constantly going out. That's why we do those top-ups every 2 weeks. We're constantly having to top it up just to try and keep it in balance, because when it is in balance, that's when it's at its best.
Okay, so you are dedicated to this strain of yeast, and you've never thought about replacing it with something a bit easier? Is it the flavours you get from it?
I think we have thought about it, definitely. But we're used to it.
Yeah, fair enough.
It just works.
Yeah, it's like your own child, really. You just get used to it.
I mean, if we could find a strain that gave us everything we wanted, then maybe, but I don't know.
Well, maybe it's the price.
Some other strains at other times, and special brews will use some dried yeast of other things just to change it up a bit.
Okay, and so how many different strains have you said you used 2 in this kind of fermentation process, but how many different strains do you keep track of across the brewery?
Really only those 2. The others will all take in as dried yeast, so they'll just be for a special brew. So they'll be one-offs.
So you got 2 main yeasts for your main brew, and then a couple of special yeasts for the one-off batch brewing. Okay.
It's interesting. Like when you take in another yeast, like how do you choose another yeast to kind of put in? Is it because you know it gives you very specific properties for a new type of beer, or do you have to kind of really understand all of the different kind of esters and aromatic compounds it's producing to add to the flavor? Is it a very complex process to kind of bring another yeast into the mix?
Well, luckily we have some quite experienced brewers here, so it's mainly the profile. So certain yeast we'd use for certain styles. So if Fergus wants for us to brew a specific style, then we'll know that there's a short list of yeasts that we might use from whoever we can get them from. So that was a good one we did recently. So, and that created quite a low pH as well. So that's it. It's just basic. It's generally on styles, and we'll pick them. And we've used what else we use? I guess we use some saison yeast.
So we use the saison, we use the Lauren yeast, Quebec yeast. We have used Kolsch yeast, we use wheat beer.
Yeah, Kolsch and wheat beer as well. Those yeasts, but really, we'll just check the profiles, and then we go for it. When it's a special brew, there's not so we don't have to worry so much about it. It's sort of a test in its own, and because we know we use trusted yeast strains that other people use, so we know that they're going to do the job that we want them to do.
Excellent. So, where are we in the beer brewing process now? Then, do you have any next steps for us?
That might be back to Fergus if we're on to the next brewing step.
Okay.
Oh. Right.
But yeah, give our thanks to Connor.
Thank you, Connor. Thanks.
Thank you.
All right. So I've kind of talked about whether you want to know about some of the other microbiology work he does. Because obviously yeast is very important to us, but we do have other spoilage bacteria, other things that we're interested in testing. So if you wanted to look at that, if you have time, then Connor can talk to that as well.
Oh yeah, I mean, yeah, definitely interested in that.
Yeah, I'm going to pass you back to Connor then. And you can finish his bit. Yeah. There you go. Do you want any plates or anything?
No, it's all right. Yeah. All right. Okay. Yeah. So, yeah, I guess not in line, but it's all in the same range for me at least. Anyway. So we plate up. I mean, the beer at almost every stage now, from yeast right through to the final pack. What we're mainly looking for is Acetobacter, lactic acid bacteria, or any wild yeasts. So wild yeast, which we might also be interested in both barrels. So like Brettanomyces, for example, we don't want that in the majority of our beers, well, basically all of them, apart from we might want a couple barrels of both barrels to have some Brettanom so they can produce some more interesting aromas, so they can produce some really fruity esters. Also the barnyardy and sometimes a bit funky, but when you're doing something for blending, that can be really handy. But generally, they would be spoilage, or termed as spoilage microorganisms. The others are also, can also be used. So Acetobacter, for example, would also be used in both barrels. So Acetobacters, it will produce vinegar basically from beer. But if you don't have too much, you'll just get a nice acidity. So I think probably the best example is probably Rodenbach, is quite strong in acetic acid. It also has lactic acid bacteria, so that will yeah.
So if you get some kind of spoils, like because the kind of acetic acid, lactic acid bacteria, like I think of my home brew now, it's like destroys it, and you can never like brew in the same bucket again because you've got this bacterial contamination. But are you saying like because you're plating it up and you can see it, is there any way of kind of controlling it as you're brewing to adjust the flavor profile so it actually doesn't ruin a batch, or is it, you know?
Generally, we keep a close eye on things, but for most it doesn't affect it. So I'll see if there is a certain amount of Acetobacter, for example, creeping into maybe cask beer, and if it gets too high, then we might have issues. But generally, we won't. And especially for keg, you don't really have to worry about Acetobacter. Even if it ended up in there, then it's a very low dissolved oxygen environment, so it's not going to survive. It's not going to spoil because it needs that oxygen to produce acetic acid. Same with lactic acid bacteria. Really, like if it's in low amounts, and it also depends what lactic acid bacteria you have, because it turns out we had one and it doesn't spoil very well. We found it, but we even tried to force the beers to make it spoil, and it wouldn't. So it was there. It's sort of pretty much cleared up now anyway, because we've had to change. We changed cleaning regimes because of that, and many other things around that. So it's a bit more complicated than just, oh, we've got Acetobacter, that's bad, or we've got lactic acid bacteria, that's bad, because sometimes, even though they're technically a beer spoiler, they might not actually be spoiling your beer.
I didn't know you had a homebrew, Ben, but I'm not surprised.
This is my kind of like ulterior motive behind kind of coming on this tour. I want to kind of get some tips and
you're here to glean insights for your own beer.
Yeah, I need to get like a little robot, I think. I don't have one of those, so maybe I should get one of those. And also 20 kilos of yeast. I use a lot less than that. So.
Okay. And is yours top or bottom fermenting?
I use different ones. Normally every batch I kind of get a different yeast, so it depends what style I make. But yeah, I think the last one was a top fermenting one as well.
Okay. I know how much is in a keg or a barrel for you in comparison to? So you don't have 700 kilograms. You have perhaps a few liters.
So I think the most common one is like 20 liters, but I actually make 10 because I had a few bad experiences in university where I made such a mess of the beer, and like throwing 20 liters of beer away is quite painful. So 10 liters is it's not very much, but it's enough to kind of test it out and play around with different things.
Very nice. And what's the outcome? What kind of beer do you get at the end? Is it anything like Adnams, or is it more like a lager, or what are you aiming for at the moment?
Sometimes I haven't done it for a while, actually. So I'm definitely inspired now to kind of do some more, but it just, I've got this little book and it's kind of got lots of recipes. So normally I'll try and do something a little bit unusual, something I haven't tried before. Although the last one I think was like a Saison or something like that. But yeah, either something like kind of different or something that I think would just be a nice beer that you can kind of produce regularly. But I haven't been able to do that yet. There's always something different. I guess I don't control my conditions well enough.
And you don't have a laminar flow hood?
No, I don't.
Oh, alas. I noticed you've got some nice rare pipettes behind you. Now, is this for testing your yeasts?
Well, yeah, that's just generally for plating up. So
yeah, okay.
So yeah, I guess with plating our yeast, so checking our yeast culture specifically, I would check that for wild yeast. So what I use is YMCU, so it's yeast of mild agar, but with copper sulfide added, which our yeast does not like. So our yeast won't grow on that, but wild yeast like Brettanomyces should. So generally, I will just do spread plates for that. I do those now almost every day. We would take samples every day just to check that we're not, because that's the most important part, is that if your yeast is contaminated, then you're going to contaminate everything else down the line. So you've got to make sure that's good before you start trying to concentrate on everything else.
And what kind of contaminants do you get? Is it just other molds or other fungi, or can it be bacteria, or like you're never sure?
Yeah, you can get bacteria in your yeast, generally anaerobic, just because of the conditions they're stored in. But so generally it would be lactic acid bacteria, which we do occasional testing for. It's hard. There's a lot of the microbiology in brewing is very difficult because beer's fizzy and it's full of yeast or solid parts. So certain things don't work quite as they should. So we have to have some sort of adaptive methods. We can't sometimes plate as much as I'd like. So I can only do 0.1 mil on a spread plate for YMCU. I'd like to do more. I'd like to be able to be more sensitive than that, but because the yeast slurry is so thick, that's just the way it is. There's not really much else we can do apart from trying to force something and then just being a detection test.
Yeah. Wow. So yeah, I'm just looking at the time. Are we doing anything else in the lab, or do we have to kind of get a move on? I'm not sure.
I think now you'll move on to Fergus. Yeah.
Thank you, Connor. Thanks very much for that. It was really interesting.
Right. I'm just going to take you now to show you the rest of the brewing. So we've sort of seen where we make the works. We've seen the initial fermenters. So I'm now going to show you where we finish off the beer and then where we package it. So having had a very stable connection for quite a few minutes, I'm probably going to ruin.
Right on cue.
We're
ready for it. We'll re-establish the stream.
Good.
We have a vineyard tour in Australia coming up. I wonder how the connection will be at that point.
Oh, the connection here will definitely be worse. Absolutely. No
okay.
Right. So I guess one of the things I didn't say at the start. So Adnams has been here since 1872, but the brewery has been on this site since 1660, and it passed through a series of hands until the Adnams brothers bought it. But Southwold itself is quite a sort of tourist town now. So we have a very limited amount of space. Basically, everything's built up around us. So we sort of squeezed everything in that we possibly could. So these are our conical, well, we call them dual-purpose fermenters. So they're dual-purpose vessels. So they can be used for fermentation. The fermenters I showed you earlier were flat-bottomed because we're not trying to take any yeast off the bottom of those. Our yeast likes to rise to the top, so we skim that off the top. There will be some yeast that drops to the bottom, but it drops at the sort of base level, and we have a standpipe in there which allows us to draw the beer off above that. These vessels have got a cone on the bottom, and if we're fermenting in there, typically you'd ferment with a lager strain yeast in there, or a bottom fermenting yeast, so that the yeast drops into the cone, and then we have chilling in the cone to sort of make sure we maintain that yeast and keep it from going off, and then we can take the yeast off. Conical fermenters are much easier to work with, much easier to clean, much easier to automate, but our yeast doesn't like them. So our yeast determines what we do, and I think you probably asked Connor, have we looked at moving away from our yeast strain? And we absolutely have, but there's a bit of romance to it, I guess. It's a bit of heritage. It's been with Adams longer than anyone else here has been, and it is part of the workforce. It is part of the character of most of the beers that we make. And we have dabbled in the past about whether we could get a single strain yeast, because actually that would be much easier to work with. You make one change, the yeast reacts in one way, whereas you've got a dual strain, you make one change and you get at least two different reactions, which you then have to try and balance. So one single strain yeasts are much easier to work with, but they're not as flexible as our yeast strain is in terms of the range of flavors we can get from it. But the primary reason is we like it. We like the uncle in the corner who's a bit moany, but actually he's got some good stories, and he adds some color to the party when the family get together. So we like that. So we have resigned ourselves that our yeast will always be here, and we will always have to find ways to work with it. So what we mostly use these vessels for, although we do ferment some lager in them, most of the time we use them as conditioning vessels, which means beer that's been in the open fermenters where we saw earlier. Once the yeast has dropped out of that, once we skim the extra yeast off, we'll send the beer from there into one of these vessels. We'll then chill it down, and that helps more of the proteins to come out. So that helps it get ready for filtration. And then from there, it'll go through the filter. It'll go into a bright beer tank, which is a tank that holds beer that's been filtered. And then from there, it'll go to kegging, or it'll go away for bottling or canning. But actually, most of what Adams has done in its history is really around cask beer. And cask beer is beer that hasn't been filtered. So instead of coming into one of these vessels, once the yeast has dropped out enough in the open fermenters, and we want to get down to a place where the yeast count in there is about 1 million cells per mil. Once it's in that level, then we'll bring it back here and we'll put it into a buffer tank, and then from there we'll put it into casks. And so we won't filter it out. I guess it's as close to drinking beer straight out of a fermenter as you can possibly get. And that's what these look like. So these casks here, these come back from our pubs. We'll clean them, we'll put it through a washer, and then we'll put this beer in that still has live yeast in it. And we want there to be live yeast in it because the yeast is then going to generate, well, we call it conditioning, but essentially what it's going to do is do a small amount more fermentation. It's going to create a very small amount of alcohol, but mostly it's creating some more CO2 to carbonate the beer while it's in the cask. And that's why these tubs are shaped the way they are. So they've got a belly on them, basically. So when it goes into the pub, it sits on its side like that. So the belly is at bottom. The tap goes in there. And then you leave it settle. So you put it up, you let it vent, you let the excess CO2 that it's built up over, say, maybe 2 weeks since you first filled it. You let the excess CO2 go. That allows the yeast to sediment properly. Then you hard peg it, which means you make sure you retain all the extra CO2 that's going to be built up from there on in. Our yeast does make some sulfur as well, so there is a bit of sulfur dioxide. So some of that initial venting is about letting some of that extra sulfur dioxide go. So it does generate a bit of natural preservative. We let it settle then for usually 2 days, and then the bright beer, after the yeast has dropped into the bottom of into the belly of the cask, that bright beer is then drawn off, and that's what we serve in the pub. So I think England is probably one of the very few countries in the world who are serving beer in that way. And cask beer is what we call it, but that's sort of been the primary bit of what Adams has done over its sort of 149-year history. So this is then the final bit of the processing. So that's a filter. The centrifuge in the Dan there, that's a Dan. And these are our filling heads. So along here, you can see there. So this is our filling. So we're not filling at the moment, but essentially casks will come up along this conveyor, come up here, and the brewing team, the racking team will take the casks, lower them down here, and then the head will drop in, and that head is basically feeding beer from that very first tank that we saw. That's been pushed into a buffer tank and then fed into here. Because it still has yeast in it, if you serve it that way, unless you can let it settle for maybe a month, you will have some yeast still in suspension. That will make it look cloudy. And historically, cask beer has always been served bright. So the way to get it bright is to add some finings. Historically, that finings has been a fish protein that attracts to the yeast. The yeast then clump together, become heavy, and drop down. You can use some other vegetarian finings now, but that isinglass finings is by far the one that is still used the most because it's the most repeatable. Because for a fining, what you want is something that you want this charge which will make the yeast stick together, and then when the cask gets rolled up to the warehouse, it breaks those bonds, and you need a finings that reattaches those bonds again, and then it gets taken from the warehouse and put it on a dray, and those bonds get broken again. So you need sort of finings that repeatedly has that with the yeast, and isinglass is still the one that sort of probably primarily works the most. I think that is the tool. I think Connor did also talk a little bit about both barrels, which is our sort of barrel aged beer. What Connor didn't mention is we treat it like a Solera sort of system. So when we put a distillery in 2010, so we had some whiskey barrels that we brought in to sort of age some whiskey in. We thought we'd age some beer in them as well. So we filled one barrel with our bottle broadside with some cherries, and then we left it for about a year. We didn't add any extra yeast to it, but what we tried to do was get some wild yeasts growing from the cherries that we added, and we also wanted a bit of wild culture in there, something that we didn't really know what it was going to be. So we got that a little bit from the wood, because obviously the microbes can live within the sort of porosity of the wood, really. So that one barrel, that was back in 2010 we filled it. But that then one barrel, we used to take little bits out. We would take it to beer festivals. Whenever we took a bit out, we'd always top it up with some fresh beer. And over probably 5 or 6 years, we constantly did that, but we still only had the one barrel, and we just kept topping it up, leaving it, topping it up, leaving it. And then about 4 years ago, we decided actually this beer was too good just to take to beer festivals. So then we took the beer that was in there, which we called the mother beer, and then we spread that across 10 whiskey barrels. We'd washed the whiskey barrels out a bit because obviously the alcohol will kill too much of the sort of wild yeast and bacteria there. So we wanted a little bit of whiskey flavor in there. But then those 10 barrels were then left for a year. And then that was our first bottling of that. And again, we never empty those barrels. All those barrels are always left with something in them, unless they go off. So if we haven't been very careful about making sure there's not too much oxygen in there when it's fermenting, or other things, other wild yeast get in that we're not particularly keen on. If they're particularly phenolic, then we don't really want those in there. We want it to be more acid-led, more fruit-led. But the barrels we keep, we will always leave some in there. So we always constantly top it up, and then age it, and top it up and age it. And that's how that beer sort of keeps getting made now.
Oh. How big are the barrels?
So the wine, they're either wine or whiskey barrels. So they're usually about 220 litres each. So they're not big. It's a very small batch beer for us, but it's an interesting beer. It keeps Connor interested. So he likes looking to see what wild yeast he can find under there, and then growing them up to see whether they give us that fruit characteristic. So particularly Brettanomyces, you can get some Brettanomyces which initially are very farmyardy, give you lots of horse manure flavors, which is not an overly attractive flavor for most people. But actually over time, some of those funky flavors can turn into some wonderful fruit flavors as well. So you can end up with pineapple and mango and peach. And usually for beer, we're getting those flavors from the hops, but actually with the right yeasts and the right conditions, you can get lots of fruit characteristics from those wild yeasts as well.
Amazing. Useful little organisms.
Yeah.
And that is why we're here, obviously, today, is to celebrate International Microorganism Day, which is technically tomorrow for us, but I've just realized in Japan, it's now nearing 1 a.m. on the 17th. So in Japan, if you're watching, it has just begun International Microorganism Day. And we will catch up with them in a few hours, 6 more hours to go for us. Yeah.
Nice. Yeah.
You're going to be visiting a sake brewery later on as well?
Yeah, so our Japanese ambassador, Ken Ushida, has filmed, I think this one's a pre-recorded tour, but he's gone to a sake brewery.
Oh, pre-recorded chickens.
Yeah, I know.
What are you doing?
But he has made us a lovely video of the sake brewery. And it is a brewery, because I always thought sake was maybe distilled because it gets quite strong. But no, it is brewed up to about 20%, which is amazing. Yeah. It's a very strong brewed drink. But we'll find out more about that tomorrow at, when is the time? For the UK, that will be at 8 a.m., 9 a.m. for us in Europe. And then we also have a vineyard tour, which Ben will be doing with a vineyard in Australia at 6:30 a.m., 5:30 a.m. if you want to wake up in the UK. But 1:30 midnight in America and 10 a.m. in India and 1:30 in the afternoon in Japan. So we're hitting all of a good number of alcoholic beverages across the course of today and tomorrow.
Yeah.
But best to start with beer, I think, as the fundamental.
It's a good time for beer now. I don't think I'll be up for wine or sake tomorrow morning. I think.
Yeah, sake for breakfast and wine at 4 a.m. Yeah,
that seems reasonable.
Yeah,
6 o'clock. That's perfect time for beer. But yeah, I mean,
I was just I had a question a little bit about the barrels there, and I was wondering because they must, like, the flavor profiles must be very very complex. Does it change over time, like with the taking out and topping up, and kind of, I suppose, the beer kind of maturing and maybe the barrel changing as well?
It definitely does. Obviously, even if you ignore the sort of microbiology of it, just the tannins of the wood decrease over time. I think one of the interesting things about wood, you get this micro aeration, so it's never fully anaerobic. There's always a little bit of oxygen coming in, so that does change how some of the microbes would normally work. So it's absolutely different, sort of storing something in a wooden barrel as opposed to a stainless steel one. It is, you will see differences that you would never see in stainless steel.
Anyway. So apologies. Oh, here we go. Here's some beer.
Very nice.
I thought I might as well finish off with a beer.
Yeah, I got to see the final product. I was going to say, apologies if I missed this, but what does your distillery produce again? Is it whiskey or like a?
So it's primarily gin.
Gin, yeah.
But we make gin from scratch. So most people who make gin, certainly in the UK, they buy in neutral grain spirit that's been essentially distilled already and taken up to a purity over sort of 96, 97%. And then people make gin from that. So we make it, so it goes through the same process, the same brew house, the same fermentation room. We use our own house yeast. We get that to about 7%. Then we send it back to the distillery, who then turn that into vodka. And then we take that vodka and we put it into the gin still with the botanicals, and hey presto, gin.
Cool.
We do some whiskey as well.
Sorry, because we talked a lot about the flavor profiles of the yeast and how it affects the beer. Does it also affect the gin?
Yes, I think obviously in much smaller ways, because you're particularly with gin, you're taking it up to over 96% purity when you're making the vodka element. So you've only got a very small amount of stuff that isn't ethanol in there. But you do find that our gin or whiskey has a different profile to gins or whiskies that are made much simpler. And there is a trade-off, I guess, because I think Conor mentioned our yeast strain is not very alcohol tolerant, and you'll all be aware that ethanol may be considered toxic in some environments, but it starts to kill off the yeast once it gets too high. So you can find distilling yeast that will get up to 15-20% ethanol, whereas our yeast, particularly on the washes that we make for the distillery, will top out about 7-8%. So there is a compromise that you don't quite get the efficiency you do with some of those other distilling strains.
And how is your whiskey? You said you made a small amount of whiskey. I mean, is it just a few batches for staff members, or do you sell it on?
It's a small amount. It's a few. It's more than a small amount, I guess. I think the sales team would quibble at with the term small. In comparison to everything else, it's a very small bit of what we do. But we started making whiskey in 2010 in the UK, and I think the rest of Europe. It's got to be aged for 3 years before you can even call it whiskey. So it's got to be aged in oak for 3 years. So we have been selling whiskey for a few years now, but it's sort of building up, and English distilleries making whiskey is a pretty new thing as well. It was frowned upon. It was, and people scoffed a little bit when English distilleries started making whiskey, because that was meant to be Scotland. Only the Scotch or Irish could make whiskey, but I think everyone accepts that's no longer true, and actually good whiskey can come from anywhere.
And is it called Adnams whiskey, or do you have a different name for that brand?
So we call the distillery Copperhouse Distillery, because that was where the old brew house for the brewery used to be. But we put a new brew house in 2007, and so that space became redundant. And then our chairman, Jonathan Adams, decided we should put a distillery in. And then he started growing some rye. So now we use his rye to make a rye whiskey. So yeah, it's it's all quite local and quite an intimate relationship.
Yeah, that's nice, with the local land and the local produce.
And weirdly enough, the place that Jonathan grows the rye is a place called Raiden, which is just the next village outside of South, where the brewery is based. And Raiden historically means rye on the hill, which we only all found out after we started making rye whiskey.
So it's back to its original usage almost. Yeah. So the beer you're just finishing up with, is that a normal pint of Adnams, or is that a special batch?
This is our New England IPA.
Nice.
So this does use our yeast now. We did play around with using some other yeast strains, but actually the New England strains that other people use are typically all English strains originally. So I think the principal part about it is you get lots of interaction with the hops. So you do get changes. So the hops can use some of the flavors the yeast can use some of the flavors that the hop produces, and do this thing that we call biotransformation, which is quite popular, I think, amongst breweries at the moment. The idea that you can actually put a hop in, and actually the yeast will change those hops into different flavor compounds. So you might start off with being quite citrus, but that biotransformation will turn it into quite pineapple-y, quite mango sort of flavors.
Amazing.
Awesome. I was going to say, we have to thank you for your wonderful brewery tour, because we have some speakers coming in for our next session as we come to the end of the hour. But it's been amazing to see a live tour of a brewery, seeing all of the distillery.
You'll never do it again.
But we have it recorded, so anyone else who missed it can watch it again, and we can send you the recording as well. It will be edited, so we'll cut out those bits where we're in transition. But it's been so good to see the inside of the brewery, all the different casks and fermentation tanks, and the final product as well. So thanks so much, Fergus, and send our congratulations and thanks to Connor for helping you complete this tour. It was a bit of an experimental idea, but I think it went pretty well, to be honest. So thank you.
Yeah, thank you very much.
It's been a pleasure. Always happy to support the microorganisms.
Yeah, absolutely. Yes. And I hope you all celebrate tomorrow, on Friday, International Microorganism Day, and get down to the pub and spread the word to as many people as you can, while giving out its beautiful products to everyone around you. Right. We'll let you sign off. Thanks so much. Have a wonderful evening. Right. I will just admit into the Zoom room our next two guests, and let's see if they can join us. So we have Kenneth Timmis and Joanna Varan just joining us for the next session. So this begins our education session. So for the next 2 hours, we're going to be talking about education with microbes and approaches to education in primary, secondary, and higher education, and also public engagement. Thank you so much, Kenneth, for joining us. I know you're pressed for time and a very, very busy man. So I just will give you a short introduction and let you get on with your talk to avoid delaying you in any way. So Kenneth Timmis is a professor at the Technical University of, I'm going to butcher this name, Braunschweig University in Germany, and joins us from that town and is going to be giving us a beautiful talk about improving microbiology literacy in society. So if we just check you can share your screen, we'll set you off on the talk. Joanna is joining me as my co-host for this hour, and so you'll see more of her later as we discuss different aspects of education. So let me just check. Can you share your screen, Kenneth? Yes. Okay, brilliant. Yeah, we can see that just perfectly. And I'll hop out of the way to stop distracting people. And we'll see you at the other end of your talk. Thank you so much for joining us.
Is it okay?
Yeah, it is. We can hear and see you perfectly.
Okay, thank you very much. Okay, it is a pleasure to have been invited to talk about the International Microbiology Literacy Initiative on this very special day of the International Microorganism Day. Why doesn't? So this initiative was launched with an editorial in Environmental Microbiology in 2019 entitled "The Urgent Need for Microbiology Literacy in Society." So what I'd like to do today is to discuss a little bit why society needs to become microbiology literate, how society may become microbiology literate, the International Microbiology Literacy Initiative school curriculum and resources, and then a few concluding remarks. So why does society need to become microbiology literate? So I've listed a number of things that I believe are important, and but there will be others, of course. So the first is planetary importance. So microbes are pervasive in their activities and in fact affect the planet in many different ways. Just a couple of examples: they generate a quarter of the oxygen which is respired in the biosphere. They recycle most of the waste that the biosphere generates, and they're responsible for 2/3 of oceanic carbon fixation, which is obviously very important for greenhouse gas fluxes and global warming. So microbes are the life support system of the biosphere, and we ignore them at our peril. The second reason is crisis importance, to minimize preventable crises and to mitigate unpreventable crises. So microbes are at the center of multiple intertwined crises we face. And I've just listed a few here. There are obviously others, but these are really important: antimicrobial resistance, resurgence of all diseases, pandemics of new diseases, starvation and food security, the soil crisis. We are losing fertile soil globally at a high rate at the moment. The clean air crisis in urban environments, clean water crisis in many parts of the world, global warming we're familiar with, waste crisis, pollution crisis, eutrophication crisis, and sustainability crisis. So in order that policy makers and decision takers respond to crises, they must understand the causes and remedies, and they must become microbiology literate. So just a couple of examples of crisis importance. So vaccine hesitancy. So these diseases are coming back, increasing in frequency. Measles, whooping cough, diphtheria, diseases that were virtually eradicated, and they're coming back because of vaccine hesitancy, because of parental concern about vaccine risks. And these concerns are exacerbated by misinformation and conspiracy theories. And as a result, herd immunity is decreasing and infection rates are rising. Well, knowledge is power. Knowledge counteracts information, prejudice, and conspiracy theories. The second example I want to give is the mitigation of unavoidable crises, namely pandemic preparedness and the COVID-19 pandemic management. So COVID-19, or something like it, has been predicted for many years, and there's been an increasing probability of it happening with increasing interactions of humans with wildlife. Many countries were fully aware of this, and they had prepared strategies to deal with it. Surprisingly, therefore, most countries were totally unprepared for the pandemic. And as we all know, the major problems consisted of supply of personal protective equipment to frontline medical professionals, which meant that they often caught the disease, and we had a reduction in capacity in our medical systems. And most importantly, the key issue of transmission was poorly understood by many. And this resulted in disparate instructions being issued by various governments, causing confusion in the population. And of course, this was all compounded by misinformation campaigns. I don't know what the current numbers are, but obviously we have a very large number of cases of COVID, and we have a very large number of fatalities. So if politicians had understood the seriousness of the predicted pandemic, and they had listened to the experts, we could have been much better prepared and managed transmission in a much better way. The third example I want to give is socioeconomic importance. So microbe-based processes and technologies contribute substantially to societal well-being. Examples given here: health. So microbes are sources of new therapies, vaccines, and diagnostics. They're sources of new chemicals and materials. They enable the production of energy from renewables and wastes. They are sources of food. They enable fermented foods to be prepared, and they are sources of flavorings. They're crucial to agriculture and crop yields because they are very important to plant health, and they can promote plant growth and suppress disease. They enable environmentally friendly recovery of natural resources, a process called biomining. And obviously they're central to waste management because they're important biodegraders of wastes, remediation of contaminated environments, and they enable recycling of resources through bioconversions. And they represent an expanding commercial sector in the sector and in the activity in the sector of biotechnology. So microbial technology is high tech, it's innovative, it's problem solving, it's rapidly growing, importantly, and it's a significant sustainable driver of creation of enterprises and employment. It represents the core of the new bioeconomy. Microbiology literacy is important for sustainability, because microbial processes are currently crucial in efforts to attain the Sustainable Development Goals. And they will assume increasing importance as innovation drives development of new technologies in very different sectors. I've listed the Sustainable Development Goals here and highlighted in red those which are impacted most importantly by microbial activities. But in fact, all of the Sustainable Development Goals are impacted to some extent by microbial activity. So we've already dealt with hunger and health, clean water, energy, and employment. But we also have sustainable consumption of natural resources, climate change we've seen, the protection of marine resources, and the protection of terrestrial ecosystems. And then microbes have a very significant importance to individuals and to us personally, because they're part of us. So we and all other organisms on the planet are covered in microbes. These are our microbiomes, and these provide us with essential services and regulate to a significant extent organism well-being. So microbiomes are the life support systems of individuals. And again, we ignore them at our peril. And to update Descartes, who formulated the famous phrase, "I think, therefore I am," we have to update it to "I think, therefore we are." So to put a little meat on the bones of the issue of personal microbiology and the implications for evidence-based everyday decisions, a few examples. So whether to elect for a cesarean, an aseptic, or a natural delivery birth. Here the issue is the transfer, microbiome transfer between mother and baby, which is crucial for development of a healthy gut microflora, which will orchestrate the development of the immune system. On the same line, whether to breastfeed or formula feed the infants, because human milk oligosaccharides again orchestrate the structure of the microbiome and its immune training. Whether to use powerful disinfectants and how frequently, because they reduce the microbial diversity of the home, and the diversity of the home also regulates the microbial diversity of us. Whether to be vaccinated against an infection, that means avoiding the infection, or to take the risk that we have it and then to be to have the morbidity, inconvenience, and possibly worse, and the need to treat the disease. So whether to prevent or to treat the disease. This is an important issue also concerning vaccine hesitancy. Whether to use phosphorus-containing household cleaning products, because phosphorus is a diminishing resource, and ultimately it gets into surface and groundwaters and causes eutrophication where it ends up. Whether to use germicidal soaps, which perturb the skin microbiome and its important barrier function. Whether to acquire a companion dog. The positive effect here is that a dog will increase microbial diversity of the home and therefore increase the diversity of our microbiomes. But there are other issues, as we'll see in a moment. The choice of the food to eat, whether we eat beef and lamb, and if so, how frequently, because they carry a significant carbon and environmental footprint. The provenance of our food, the shelf life of the food, the associations with known risks, how to store our food and prepare it, and how much to ventilate. Just think of the SARS transmission issue, or humidify or dehumidify our homes. These are everyday decisions that we're confronted with, and where a knowledge of microbiology will help us make evidence-based decisions. So microbiology literacy is essential to transit from ignorance-based decisions and chance-based outcomes to knowledge-based stewardship of our lives, the biosphere and the planet. And the aim of the International Microbiology Literacy Initiative is to provide this knowledge and thereby become an enabler of responsible stewardship. Well, I've talked quite a bit about literacy. So what is literacy? Well, here is a definition from the UNESCO. It's the ability to identify, understand, interpret, create, communicate, and compute using printed and written materials associated with varying contexts. Literacy involves a continuum of learning. So learning is the key word here. So learning can occur throughout life, but tends to be concentrated in formal education and during childhood, early adulthood. In essence, its product, in other words, the product of learning is literacy, and it is the qualification of adulthood with all its challenges and responsibilities. So literacy is best acquired in school through incorporation of the subject into the curriculum. Qualification of adulthood, what does that mean? To understand our world, humanity, our needs, risks, and so on. To assess and develop informed, objective policies at the individual level, community, family, national, international level. To make rational and accountable decisions which are understandable by everyone. To take productive, accountable actions. To assume our stakeholder responsibilities. And to participate fully in the family, the community, and the wider society. This is the qualification of adulthood. So for teaching, we need resources. Well, in fact, there are many excellent microbiology resources out there, both online ASM, FEMS, Microbiological Society, the Spanish Society for Microbiology, eBug, and many, many more. And physical resources like exhibits by the University of Oxford and Harvard. But the formal incorporation of microbiology into school teaching requires the creation of a comprehensive microbiology school curriculum for different age groups and settings. So the goals of the International Microbiology Literacy Initiative are the following. To provide appropriate curricula for different age levels in diverse societal and cultural settings worldwide. To reveal the major planetary biosphere human processes and problems impacted or underpinned by microbial ecophysiological activities. To inform how these activities affect our well-being and that of other members of the biosphere. So in other words, how microbes affect us. But on the other hand, to reveal how microbial activities are influenced by our actions and the ensuing consequences, how microbes are impacted by us. To indicate how we may steer or exploit microbial activities for personal, human, planetary benefit, and to contribute towards attainment of the Sustainable Development Goals. And finally, to provide a perspective of our place in the wider world and how we are microbially connected in the global village and with the rest of the biosphere. So now I'd like to come to the IMLI curriculum. So it's important to note that the curriculum is child experience centric, child interest centric, child excitement centric. And it consists of the following: class lessons, the topic frameworks, and I'll go into this in a moment. Multimedia teaching aids. So the big handicap we have as microbiologists in terms of communicating our science is that microbes are invisible. So for teaching, we need to visualize. And we do this through teaching aids of various descriptions. Class experiments. So microbiology is so hands-on, and kids love hands-on activities. Home assignments to transport microbiology into the home. Class excursions to immerse children in the local microbiology, where they can experience real microbiology in a local setting, and to get them out of the classroom. And there are a number of other things in the pipeline. Most importantly, all of the teaching resources that are being created will be freely available internationally. So let me come to the topic frameworks, which are the heart and soul of the strategy. These are basically the class lessons, but they're not prepared class lessons. The teacher has to prepare them. So what they consist, that they are generic outlines of knowledge, they're generic frameworks of knowledge, which are adaptable to all age groups in all settings. They're written in non-specialist language for teachers. So the teachers are the target of the topic frameworks. The teachers can select the topic framework or select information within the topic framework that they consider to be interesting or important for their class, and they transmit and interpret it for different age groups and teaching needs. The topic frameworks are standalone, so different from typical curricula where you have to follow a set sequence, and if you miss out some parts of it, you are handicapped. In this case, the topic frameworks are standalone, so teachers can select and make their own collections, their own groupings of topics which they consider to be appropriate for their class and their purposes, their teaching aims. So they can mix and match them. And actually they can take bits from one topic framework and take bits from another one and mix and match them, and thereby create new topic frameworks. The topic framework places the topic treated not only in its human and microbiological context, but also in the context of wider issues, the grand challenges, sustainable development goals, and societal problems. And they identify relevant decision issues impacted by the microbiology presented.
The topics are grouped in sections, some of which are listed here, but not all. So our personal microbiology, where microbes play a role in our well-being, so body odor, toothache, acne, things which are important to children. Our food, our plants, our animals, our well-being, health issues, our planet, our water, how microbes help us, the topic of biotechnology, how microbes influence civilization and culture. So what microbes have done in the past to create society that we recognize today. And finally, some background information on how we study them. So let me come to the topic framework. So the topic framework consists of a title page, which has a title, and a child-centric question, plus a relevant image to enable the teacher to catch pupil attention and establish child relevance and interest. There's a storyline, which is a short description of the TF content to enable teachers to rapidly browse the selection of TFs and to make a choice of the ones that they want to use. Then you have the body of the topic framework, which is basically consists of brief descriptions of microbial activities that underlie, control, or influence the issue discussed. Then we have a section on relevance for sustainable development goals, a section on potential implications for decisions at the personal level, community, national, international level. We have some pupil participation exercises, and then at the end we have what's called the evidence base, some references, future reading, and teaching aids. And finally, a glossary. Let me show you some one example of a topic framework. So this one is on pet dogs. So this is obviously not classic microbiology. This is a child-centric topic. And the question and the image, Maisie has just been given a gorgeous little puppy for her birthday. Can we have one? And I think many families will have been confronted with this question, and many children will have posed this question. So it's relevant. So what do we have in this topic framework? Well, companion dogs obviously bring in microbial diversity into the home. They increase exposure to microbial diversity. And as we know, microbial diversity is very important for development of a healthy immune system. So this is one of the positive aspects. Of course, there are a lot of non-microbiological positive aspects to ownership of a pet dog, which I won't go into. But like us, pet dogs can become infected. Some infections are zoonotics, so we can get infected from the animal. And that means that we have to take our pet to the vet and have it treated. Of course, we can prevent disease, some diseases, by vaccination. So infections come up in this topic framework, vaccines and vaccination come up. And then our pet has to be fed, and a very considerable fraction of agriculture is devoted to the production of dog food. And this consists of both animal meat and grain. And if we think about this, one of the consequences is that this is diversion of food production away from humans to animals. So this impacts on the issue of food security and on the issue of starvation. In addition to that, all of the agricultural activities have an environmental footprint with regard to greenhouse gas production and also eutrophication. And finally, dogs pollute the environment. So these are issues that are microbiological, that are relevant to ownership of a pet dog, and that relate to discussions in the family about whether to acquire one or not. Now we come to the relevance for sustainable development goals and grand challenges, and I've mentioned already this particular one: end hunger, achieve food security, and there are others that are discussed in this topic framework. And then we come to potential implications for decisions. So if we decide we will have a dog in the family, the question is, should it be a large one or a small one? Because the environmental and energetic footprint will be approximately proportional to the size. So if we're discussing the environmental aspect, the size of the dog will be relevant to that. Community policies, so I've mentioned pollution, in this particular case, pollution of public spaces and local water bodies with feces. Nitrogen, phosphorus will contribute to eutrophication and so on. And then we have national policies relating to dog ownership, greenhouse gas production, and global warming related to the production of dog food. And then we come to pupil participation, so a class discussion of issues associated with dog ownership, pupil stakeholder awareness, so dog ownership has a positive and negative, has positive and negative consequences for the SDGs. Which of these are most important to you, either personally or as a class? Can you think of anything that might be done to reduce negative consequences? Can you think of anything you might personally do to reduce the environmental footprint of your dog? And then we finish with the evidence base and the glossary. So all of these issues that are brought up in the topic framework are dealt with in sufficient detail to be able to explain the issues and enable people to discuss them when they're taking decisions. But of course, they may raise interest in the teacher or in the child, the children, to have more information. And so we have obviously topic frameworks that are dedicated to some of these things, like the early microbiome, like infections, like vaccines and vaccination, antibiotics, so the treatment of the pet dog, antibiotics and antibiotic resistance in the environment, climate change, eutrophication. And then we have, of course, a lot of other topics which are unrelated to pet dogs. So dirt is good for you, so microbial diversity is good for you. Foodborne microbial disease, some more information about microbiomes, diagnostics, indigo, moisture damage and mold in our home. You see, all of these are child-centric. Volatiles and communication between microbes and between microbes and higher organisms. Food preservation. Recovery of resources from wastewater. Red and green tides. Fruit flies. Microbial control of animal behavior. So those are some examples of the TF. So what is the current status of the initiative? So we have something like 200 plus commitments for topic frameworks from authors. More than 150 of these have been submitted already. More than 80 have been completed. So I'm anticipating that the core of the curriculum will be completed this year, but there will be others which are added later. The multimedia teaching aids, this is in progress. Class experiments, this is in planning. We hope to complete that by 2023. Suggestions for class excursions, that was already completed in 2020. And we have a small-scale trialing of topic frameworks in schools during the coming school year. So the initiative is well underway. It involves a highly diverse group of players from many countries who provide very diverse outputs. It's attracting a lot of interest also from teacher relatives and friends who will carry out the initial trials and provide feedback to us. I should emphasize that the TFs will not only serve for school curricula, but also for adult education and self-learning, and importantly for university outreach. And while we're on the topic of universities, I think that TFs and their concept may also provide new ideas, both in terms of content and approach for university, undergraduate and postgraduate teaching. But I should also emphasize that the aim of the IMLI is not to create microbiologists, but rather to create excitement and interest in microbes and their activities, and thereby adults equipped with the knowledge they need to make responsible decisions. Well, there are many things to do. Authoring of more topic frameworks, creation of the MTAs, creation of experimental protocols, translations of a few key documents, recruitment of additional teachers for trialing, engagement, most importantly, engagement of education ministries, authorities and agencies, and establishment of regional centers to promote and coordinate the IMLI. So if the IMLI excites or interests you and you'd like to contribute, do get involved. Here is my email address. And if you do get involved, you'll be joining many other well-known microbiologist friends worldwide, including all of these people and many more. And finally, I just want to say that attainment of literacy will depend on societal acceptance. And societal acceptance of microbiology literacy will depend on persuasion, realization of its practical value, and collective excitement about new discoveries. And this persuasion must come from the experts, you and me, the microbiologists. We are excited by microbiology. And excitement is infectious. We need to be transmitters of a global microbiology excitement infection. So on this special day, the International Microorganism Day, we must commit to the task of communicating to others the fact that microbes rock. Thank you.
Thank you, Kenneth, for that wonderful talk. Detailed and wise and incredibly coherently delivered as well. I believe you have to go before we can ask you too many questions. But I just wanted to say, what are you going to be doing for International Microorganism Day tomorrow in celebration?
I am going to give a second talk for the Portuguese Microbiology Society. The Portuguese Microbiology Society initiated the International Microorganism Day, and I'm very proud that they invited me to make a presentation for them on that day. So this is the first thing I will do, and the second thing I will do is jump in the car because I have to go to the UK for the first time in 2 years to visit family and friends. COVID has been a dreadful thing for most people, and I'm incredibly lucky that I haven't been seriously affected, but many people have been affected by the same issue of not being able to see family and loved ones and friends. So this is what I'm going to catch up on tomorrow.
Wonderful. And yeah, I hope you have a good day and celebrate microbes with excitement, infectious excitement. I guess that brings us on to the next stage of this education session. And we'll say goodbye to you, Kenneth. Have a wonderful evening.
Thank you.
Which brings, my co-host Joe into the studio with myself, Joe, and Delaney. If we just unmute you and check we can hear you.
Yes, good afternoon.
Yeah, good afternoon, good evening. How's it going in the UK?
It's great, thank you. I've been really enjoying the day. It's been fantastic. And that was really interesting, that curriculum as well, and the way that it sort of extended right across from children through to adults. I think it fits in well with what we're trying to do today.
Absolutely. And I guess it's a call to arms for everyone in the microbiology community to try and be as excited and persuasive about the wonders of microorganisms as possible. And I guess for the next 10-15 minutes or so, I'm going to talk us through some education resources we've put together for International Microorganism Day, aimed for school kids, aimed for young adults and children. So if I just start sharing my screen, we can start to work through them. How is that? Can you see the education resource page?
Yes, I can.
Yes. Okay, great. So, I mean, last year we produced two wonderful education resources, the Winogradsky column and the sourdough bread education pack. They contain wonderful infographics that show you how to produce sourdough bread and Winogradsky columns. But we're very pleased today to announce that we've produced two more. And I think you had a hand in a couple of these, didn't you, Joe? With
yes,
Ben Liberton.
I worked with Ben. I think he asked me to kind of read them like I didn't know what to do, so that I was able to follow his instructions.
Yeah, wicked. And they were clear from your perspective. Yeah, okay. So I'll just dive into the first one of these, which is making yogurt. So, I mean, everyone at some point in their lives might have had yogurt or bought it from the supermarket, but you can actually quite easily make it from home. So in this education pack, you'll find an infographic for use by the children in the class, and then some teaching resources for the teacher to have a bit more of in-depth knowledge and a few little tasks for the children. And as we work down the infographic, we can see that mostly you only need things that you would find at home. I guess the most complicated thing here is probably a thermometer, but you can buy this in most good kitchenware stores, usually for testing the inside of meat, but you can use it here to make yogurt. So we have, a litre of whole milk. You need to use a few different types of yogurt to start off your culture. You've got 2 clean tablespoons, some clean lidded jars, perhaps a Kilner jar or something similar, marker pen, small pan, thermometer, clean measuring jug. Then the infographic here demonstrates very clearly what you need to do. You use your milk, and then we have to sort of pasteurize it in the first step, which means heating it up to nearly but not quite boiling to kill off any harmful microbes. And then we let it cool down to a more tepid 40 degrees. So now you have your milk prepared. You can then add the yogurt starter. So Joe, can you explain roughly to me, why do we need to use a yogurt starter? What's it doing in this process?
Well, you've eliminated a lot of the microorganisms already in the milk. So you're adding the right sort of microorganisms from the hopefully live yogurt into the milk so that they can then replicate within the milk.
Exactly. So we're kind of inoculating the mostly sterile yogurt with the right microbes to make yogurt. And we know they're the right microbes because they are from previously made yogurt. So in this setup, you have a few different yogurts to try out, and you hope that the yogurt you make will be similar to the one you started with. So in this case, you have 2 yogurts. You take a little tablespoon and add it to each of your Kilner jars, and then you take your 2 Kilner jars, and then you mix in the milk that you've pasteurized, and then you stir this all together with the spoon, and then you place the lids on top. So in this case, they have let's have a look. 2 batches of yogurt 1, 2 batches of yogurt 2, and then they have just plain milk to see what that does. I guess that is your control if you're thinking about experimental design. Then you simply leave it in a warm place. So the 2 ways that they've got is you could either put it in a box containing warm water, or you could perhaps put it in a warm space, maybe a really, really low oven on top of a radiator, a warm cupboard near your boiler, that kind of thing. And it says the yogurt will form between 6 and 48 hours. So within the space of a day or 2, you should have brewed yourself some nice yogurt. But we've got a test if the yogurt's ready. So there's a simple test that you do here. You take your spoonful of yogurt that you've made, you pop it on your plate, and, well, if it's thick like yogurt, it's ready to eat. And if it still looks like kind of a runny milk or something a little bit more like a weird cream, then it isn't quite ready. And you put it back into your incubating space. Now, we use different yogurts. So the last step, I think it's the last step, almost the last step, is you take your yogurt containers from your warm place and you put them in the fridge for a couple of hours to make them nice and cold, which is how we expect yogurt to come. And then you taste it. And hopefully you'll have something that tastes like a delicious yogurt that is similar to the one that we used at the very beginning. If it isn't quite ready, again, like you say, you put it back into the warm container, and once cooled, you should be able to taste them and enjoy your homemade yogurt. Storing your yogurt, wow, we keep them in the fridge like lots of different foods, and you should be able to keep these yogurts for maybe 2 to 3 days. So don't eat them beyond that point because they might spoil and become too acidic and become a little bit more like a weird cheese. And that is basically as simple as it is to make yogurt. If you want to get a little bit more complicated, you can test different varieties of yogurt. You could even try different milks. You might actually be vegan and decide you don't want to use dairy milk. So you could use skimmed soy milk or oat milk, or maybe even almond milk, although I haven't tried that one. Have you tried any of these milks in yogurt production, Joanna?
No, I haven't.
No, okay.
No,
just
no.
a straight dairy yogurt. And the other thing you can do is, instead of cooling it straight away, you can try and heat the yogurt for some time, maybe 5 to 20 minutes, to see if that changes the consistency of your yogurt. And you could also try adding different other sources of bacteria. So maybe probiotic drinks, there's a huge range, and you could use these instead of the yogurt. For teachers, we have some teacher notes, which I can open up here, which just give you a bit more of background knowledge, give you some information about cleanliness so no one gets ill, a bit more detail on temperature, and just fills you in with a bit more of the kind of like detailed knowledge you might need to run this as a classroom experiment. Let me just end the full screen and hop back 1 page. And the infographic here is easily downloadable. It runs really well on a smartphone screen because it's so long, so you can just scroll down it with your classmates. That's the first education pack. The second one is making agar plates. So this one's perhaps a little bit more complicated and a bit more like using science in a, I guess you'd say, laboratory condition or laboratory format. So again, beautiful infographic we've put together here. Were you just asked to read this one through, Joe, or did you have any more inputs on this one of the education?
Yeah, I worked with Ben on both of them. Yeah.
Okay, wicked. So yeah, this one again uses loads of household ingredients. I guess the most difficult thing to find is actually the pack of agar. But apparently you can get these from health stores or Asian supermarkets because they're sometimes used in styles of cooking that are probably beyond my skill. But you can buy these agar packets, and then other than that, everything else is fairly straightforward. Small containers, heat-proof bowl, cotton swabs, waterproof markers, kitchen towels, kitchen scales, tablespoons, teaspoons, a litre of boiled water, some beef bouillon cubes, adhesive tape, aluminium foil, spatula, a fridge, thermometer, oven gloves, and then some nice things to keep yourself clean and disinfected, plastic gloves, paper towels, and disinfectants. So a few more pieces of equipment needed for this one, but you do get, perhaps I'd say, a more tangible understanding of the microbes at the very end. So, oh yeah, you also need to have a sealable bowl that is, at least 100 milliliters, has a sealable lid and is wide and shallow. So some kind of Tupperware style bowl will be what you need here. And to make the agar mix, well, you take 1 liter of recently boiled water, you add it to the bowl, a heat-proof bowl, and you add 5 beef bouillon cubes until they dissolve completely. So it's like making a stock to begin with. But then once that's all dissolved, you add the agar until that's dissolved, so about 15 grams. And you allow it to cool to about 50 degrees, and that's what you use your thermometer for. So once you got to this stage, you need to pour the agar. And this is something that scientists used to have to do in laboratories themselves. They used to have to make their own agar and pour their own petri dishes and agar plates. But luckily, you just buy them nowadays. But for the purposes of understanding what science used to be like, you have to pour your own agar. You use your spoon, I think, here to just basically ladle out a little bit of agar into these small containers. Try and make sure it's all gently and smoothly poured and you don't have like stuff running up the sides. And so it's just a nice even layer. Also try and make sure that you put lids on these containers so other bits and pieces and micro dust don't get on top and contaminate your petri dish. And don't seal them too tightly because it's a hot mixture. It will cool, it will contract the air, and you might crack it. So you need to have some kind of air able to get in and out. But once you have poured your agar carefully, you can then put them into the fridge. Leave them 30 minutes and check by touching the outside they're approaching room temperature. And once it's cooled to room temperature, you can seal the plates fully and the agar should be completely set. Then you turn the containers upside down and put them in the fridge. I think now the reason they're upside down is so that you don't get water falling on the surface. Is that right? So it just condenses inside and then falls onto the bottom of the lid and doesn't contaminate again with liquid your agar. So you leave them in the fridge overnight and then they're ready the next day. So this is the exciting bit now. That was the kind of lengthy cooking process. But now you have these agar plates. You can go around and start sampling areas for bacteria and see if we can grow them. So you might try some dirt in your garden, your sink plug hole, your fingertips. I remember when I was working in a microbiology lab, I had loads of wristbands from festivals that I'd gone to over the summer. I had about 7 or 8 attached to my wrist. And my supervisor took my wrist and he took the bands and he stuck them in an agar plate and put them in the incubator overnight to show me how disgusting these bands were that I'd had on for about 7 months. And of course, the next day, the agar plate was full of the most revolting looking bacteria and fungi and molds that you'd ever seen. So if you have some festival wristbands, they're full of bacteria. So you can stick those on the agar plate as well. The suggestion it has here is to use a sink plug hole and take your cotton swab and run it around the inside and then dab it onto the agar plates, as you see here. Label them with the location of the swab and the day that you've taken the swab. Now label it with your name, date. If it's a class, you might want everyone to put their name on it so people don't forget which is theirs. So repeat it for all the areas you want. Build up a nice collection of agar plates that you've inoculated with the cotton swab from different locations, and now you get to grow them. So you need to leave them in a warm place for about 2 days. So this could be a radiator near a boiler in your house, that kind of thing, or on a bright sunny windowsill. And then these tiny numbers of microorganisms should sit on the agar plate and grow and grow and grow until they have formed colonies we can see with our naked eye. And that's the exciting bit. So when you take them out of the warm space, remember, don't touch them, because there's going to be like just billions of bacteria and billions of molds cells on the surface of this plate. So do not touch them. Make sure your class do not touch them. That's the main thing. But because you've got a clear lid, hopefully you can see these beautiful colonies, and you can judge the shape, the size, the color, and try and guess what microbes you have. So you'll have an agar plate. Perhaps you've even decided to put it into some kind of fun design to enter an agar art contest. But once you have this, you can start to have a look at your colonies and see, what you might have. So it says here, sometimes microorganisms will grow as individual colonies that are most often round. You may see colonies in an agar plate or just a few. If you have a sample of lots of microorganisms, the colonies can join together to form a lawn on your plate, and all outcomes are good. But you can start to compare the different agar plates and see which location is more or less full of microorganisms. So that's the second teaching resource. Again, we have some teacher notes, which perhaps give you a little bit more information about what the Petri dish is, and some things to consider about heating, cooling, and cleanliness. And a little bit about why we use beef bouillon, and some other things about how to deal with spills, and then crucially disposal. So how do you get rid of all this stuff without contaminating anyone's bins too much? Also, there's, I think at the bottom, some special student instructions. If I click here, we'll take it to, just a nice set of instructions for students that comes in a nice PDF form with a few extra questions as well to kind of challenge your students as to what's the microbiology behind this process. So that's my very brief run through of these two new education sources that join our old two education sources to give you a nice little quartet of useful tasks and useful experimental protocols to use with your class. So I hope Kenneth, if he's watching this, is happy with our efforts here to produce some nice teaching materials to improve microbiology literacy. I believe we have Beatrix joining us. Beatrix Farnhart from the University of Lincoln, who is also the section editor for education and professional development, I think, for our journal, FEMS Microbiology Letters, and will join us for a little bit of a structured discussion on how the pandemic has changed teaching and education in universities. Now, Joanna Varan at the Manchester Metropolitan University is also another education expert, a member of the FEMS education group. So we have two of the best people here to discuss what's the pandemic done to teaching? Big question.
Well, I think I'll start. Obviously, when the pandemic happened in universities, we were kind of halfway through the academic year. That was in the 2019-2020 session. Then we had the 2021 academic session, which was almost entirely, to some extent or another, during the whole pandemic. And now we are just beginning the 21-22 academic session. And I'm sure that Beatrix, I don't know if your students have arrived yet, but I think we've got a lot to learn from what we did instantly when the pandemic first arrived, how we managed in between, and how we're going to carry on. So I thought I'd kind of ask Beatrix a couple of questions based around those three, before, during, and after, I suppose, sessions. That's hoping it's after anyway. When universities were first affected by the pandemic, it was kind of said that universities were closed down, but of course they weren't. Education and teaching had to continue. And my first reaction was, oh God, lectures, labs, tutorials. But actually, Beatrix, I think probably you would think there's a lot more to it than that. It wasn't just that was going to be affected, was it?
Yes. Hello, everyone. To start with, by the way, I hope you can hear me. I've managed to unmute myself, having practiced this for months now. Hello, everyone. And it was, of course, quite surprising when everyone had to close campus. But as you said, it didn't mean we closed our universities, far from it. probably been busier than before. And if we think about when it happened, not only in terms of within the academic year, but where we were technologically, if we had the pandemic like this 20 years ago, things would have been ever so much more difficult. In that context, we were quite lucky because we had changed education already for quite some time in ways that then helped us to actually deliver successfully, not only for a few weeks, but over an entire academic year then. And of course, technologically as well. Universities have virtual learning environments for quite some time already. People at home have broadband or Wi-Fi internet connection. So that was quite a different kettle of fish than if it had been 20 years ago. And very, very quickly, we had to make sure that we could deliver what we had planned just the next week or probably the next day. We didn't know how long it would take. And as you said, it wasn't just lectures. It's just everything. It's tutorials, it's pastoral support, it's labs, of course, it's field work, depending on what everyone is doing. Of course, our colleagues in arts know dancing and all these things. So the first impulse was just getting things going for the next week and so, but as we realized it was taking longer, we then had to properly plan strategically and go from there. Don't know how much you want me to go into detail already right now.
Well, I suppose, as you said, you had the kind of immediate aspects of lectures, tutorials and practicals, but then you had, I mean, perhaps not even so far away, there was assessment as well.
Yes, and so now I start already sort of talking a little bit broader. We've for some time gone away from what probably many people think about as a classical lecture, where you're basically talking for quite some long time at students and then they go away and absorb all the knowledge that you've given and then apply it, and then they come back and you're giving more. But we've gone away from that. We've flipped classroom, for instance, so we've given students before the material that would cover knowledge, and then when we see them, very often we would help students apply it, because education isn't just about the knowledge, as we know. It's about the skills as well. In applying this knowledge, it's the other transferable skills, is interpersonal skills, it's the discipline-specific skills like in the practical. And again, assessments had changed as well to reflect the changed ways of learning, because we make sure that always lines up nicely. So we've gone into the direction of often more open book as well, which means students can access the material, because we want the students to show us how they apply what they've learned and the skills they've developed. We've already started having a lot of virtual material or online material, basically. We often use already in labs videos of the techniques rather than going around showing it to everyone at their benches to make sure that everyone could see very well. We could draw on that. And of course, a lot of that was also provided by learned societies. So we could very readily access those sources that we either had made ourselves or that we knew existed, and transform all these things that had happened over time then quite readily into this virtual environment. And even if we weren't able to, say, record how to streak a plate that one made at home, we could direct the students to that. So it was about curating the material in the beginning, making sure that students have access to that, but actually, cared more about our students. Okay, how can they survive in their little rooms somewhere, being isolated? So that actually took quite center stage, making sure that all that is, and that of course the assessments would be happening, and they then became in many places where people were not on campus open book, because of course you can access your material at home. So we then had to think about again how we write these assessments, but it's not University by Google, other search engines exist. So that was getting us quite quickly, because of all that background that we had and helping each other. Our networks kicked in quite quickly, helping each other, really, really important. to make it happen at the end of that academic year. And then we spent the summer realizing that we had to plan a lot more and could draw readily on a lot more. By then, of course, people had upskilled. The institutions would, of course, help staff training quite readily with technology and in terms of pedagogic support. And yes, that trying to be, in a nutshell, talking about something really complex and big.
Yes, I know. It's massive then. So we had the labs, the lectures, the practicals, so the tutorials, the assessment. We had student well-being, and we had staff upskilling and student upskilling, distance learning, and pastoral care.
Very different levels as well. So the need of students who were about to graduate and then go into the world of employment, we wanted to make sure that of course is an easy transition and no one feels like they couldn't complete their education. Even though that wasn't the case, but maybe students would worry, or the current student cohorts, that no one thinks that anyone hasn't developed all the necessary skills or hasn't learned as much as other student cohorts, and we can now help students to be very confident in this and convey that wherever they go. And of course, when students come back now at some point, hopefully not closing down again, helping the students transition back. So all the students at different levels have different needs where they come from. And of course, our incoming cohorts wouldn't have been in class in schools. So then the transition into university is a very different thing. Or the students going in the second year, they wouldn't have really been much on campus and in a normal world. So it's all these things that we're now already been preparing for quite some time to make happen when they come back.
Yeah, I mean, I wonder if our audience on this program is other academics who all this will be sort of resonating with. But just to, after sort of outlining all of those challenges, then before we go on to the next bit, I read an article in the New Statesman included a statement: people all over the world can learn and at the close to zero marginal cost. Because it doesn't cost anything anymore because everything's online. So that there is that one kind of view of it, that it's really, everything's really easy now. And then there is the other, that certainly I've found that in the UK that applications to actually go to university have gone up, because I think students want to come back. So I think, I think that academics, the universities have done very, very well in this time to actually to make themselves more valued. So, I mean, what I wanted to ask you, Beatrix, as well, as part of your work as the editor of the professional development aspect of the FEMS Microbiology Letters Journal, have you seen through the pandemic academics wanting to tell other people about what they've done during the pandemic? Because it's quite difficult, isn't it, sometimes to, if you've tried something and it's worked or it hasn't worked, how do you share that, and how do you get comments back?
Excuse me for coughing, it's not COVID, anyone. Again, that is a very complex question. How much time do we have? So in the beginning, some colleagues were actually quite quick off the mark, starting research questions, before, after, and surveying students and academics in various ways to actually focus their research on the response. That's one thing. The other is sharing materials to help each other was probably more readily done through networks in a more informal way, rather than sitting down, writing a paper, sending it in peer review, then your revisions, because that, of course, you want to make sure that it's at an extra polished level when doing this. So there was a bit of a lag, never mind. But of course, rewriting your provision, learning how to use Zoom or any of the other things, looking after your students, probably even longer periods in the day when you want to make sure that they are well. You would maybe also homeschool your children while your pets were climbing over you, all these things. So that, of course, was an impact on workload. So we wouldn't immediately see manuscripts mushrooming, but there were, of course, opportunities where they were then taken by colleagues depending on their situation. But of course, meanwhile, a lot of papers in various journals, including ours, have filtered through. And in fact, we are looking forward to launching a thematic issue next month on the beginning of October. I think it's the 5th, the International Teacher Day. And there we can draw on some papers that we had before. As I said, we've been going in the right direction for quite some time. And very exciting papers that were written by colleagues who were reporting on what innovative deliveries they were able to develop and then successfully implement during the pandemic, which help us going forward on that trajectory that we have in terms of being more flexible and blended and more personalized. And so that will be not just, oh, this was what we done in the pandemic and, you know, put it in the drawer and everyone has seen what we have been doing, but they will be instrumental in terms of professional development for educators and taking education into the next steps, because one of the silver linings of the pandemic was that it basically speeded on developments that we sometimes been taking a bit longer, because it takes quite some time to implement change if people are very, very busy. So say active learning, authentic learning, some of the good blended aspects, not, you know, leaving someone in their little room unsupervised, which of course can be very demotivating and causing anxiety. So all the good things that we could now push a little bit forward more quicker will help us a lot in going into the right direction where we all should be going.
I just had one question, actually, if that's okay, before we move on to the next section, which was, did you find that different kinds of students excelled in the kind of online system compared to when you were in person? And, you know, did you find it helped some people who perhaps would have been too shy to ask questions and this kind of thing when you were teaching online mostly?
Absolutely, yeah. There were quite a few students who found that much more helpful because they might be shy. Also, of course, students who usually would have quite a long commute or might have some other accessibility issues or caring responsibilities found it very, very helpful to make use of this sort of flexibility that it afforded. So it has good and bad sides. And when we look forward, we have to make sure that we keep the good parts and lose the ones that were not so helpful. Say, you know, impact on mental health. We know that people have been struggling with the whole not uncertainty is the word I was looking for, because you didn't know, is it next week, is it next month? And of course, now we still don't know how long we might be affected by it in practical terms in education. So again, that helped students also become far more resilient and adaptable than in a normal time. So while there might have been a lag in developing practical skills, which we will help everyone to catch up very soon, and anyone who goes in the employment world now, employers will know that there might be some gap in practical skills, and help everyone to catch this up. In other ways, we have developed other skills much more thoroughly than we usually would be able to do on campus. I mean, digital use and being resilient, independent, self-managing, all these things that are absolutely crucial for employment going forward. So hence we can tell the students and support them in taking all the confidence, having gone through this, come through this with an excellent education as well, in a different way, and then requiring different activities to transition into whatever comes after that. And even, you know, doing practical work at home, not only with making yogurt, I felt like I wanted to open my fridge when you were talking, and making your own agar plates. But we, you know, just changed the research question, for instance, even doing dissertations at home, you know, rather than develop looking at growth in the lab of a pathogen, you transform it and, you know, have your yeast at home, which is fine. So these things, we were very, very inventive. And I say we, not of course just only my colleagues, but everyone in their setting, in their universities around the globe, helping each other, making all these things happen. So aside from the in-person community building and some of the sort of more hardcore pathogen related practical skills, we could deliver everything and help the students develop all these skills and learn the core knowledge as well, as we all assessed at the end of the last academic year. And the students did fantastic, and we were very proud to see this.
That's fantastic. You mentioned authentic learning. I wonder if you could explain what this is.
Basically, what is real life, real world problem scenarios. I still hate it when people sometimes say, oh, this is just, academic, because of course that's the perception, and that's probably a historical way that we were dealing with things that were not real world and not applicable, and of course for a very long time when I was a younger educator, not now, getting old, I was initially surprised by questions that students would say, so how is this relevant to me? And I was thinking, well, that's obvious, isn't it? But it's not necessarily obvious. So we learned very quickly, I did, and that we need to make things more relevant. And of course, with the talk earlier, it's important, microbiology literacy, but also what is the worth, the value of any education if we're not dealing with the problems that our students should be solving when they leave us, because that's what we prepare them for work in the real world, solving all the problems that we've created in the past and our predecessors, and that we depend on. So why would be creating an artificial environment doing something that's not relevant? So again, we have gone a long way, and more using real world examples, real world problems that are everywhere. You don't have to look very far.
Yeah. And you're talking about the courses and the learning, but some of our courses in microbiology, so for example, biomedical science and hospital-based work, how did the organising bodies or the official accrediting bodies react to the pandemic and the degree?
Yeah, that's a very, very important question. So when I said, future employers might help students transition into those environments where they have missed something, of course, there are some professions that require a specific skill set that is regulated and that's accredited in terms of the programs that prepare the students for those work environments. Generally, we would usually be in contact with those accrediting bodies whenever we make changes, even in a normal year. And so, of course, with making all these changes, we had to have a very, very swift and helpful interaction. And again, when I say we, I'm not just talking about our programs, but everyone in the same situation. And many, many thanks to all the accrediting bodies for being extremely quick in their response and very, very helpful. And so we made sure that everything that was done was still within the required context. And also depending on the national regulations, I can now only give an example of my own because I'm not au fait enough with what other countries necessarily did in detail. But some of the programs that were allowed on campus, even if everything else was closed, were those very programs that were accredited, feeding into, even, training nurses and our biomedical students and anyone in this context. So again, making sure that that was possible. But then also after that, in summer, many universities offered catch-up opportunities for lab skills. And again, in conversation with the accreditors, they would be aware that that happened. And then anyone who is still with us or any other university, one would make sure that that's caught up particularly well as well in the next academic years. And please don't look anything down again in terms of the virus. So again, there was great care taken and a very close eye that did happen because it's people's lives. And that happened everywhere.
Thank you. So, I think that sort of finishes the second part of our chat. How did we cope? What did we do? And I think some lessons coming out from that is to share, to ask people, to exchange ideas with whatever networks you have and whatever networks you can find, and that hopefully people in the audience who are not in universities can see how much work went on, but also other people within universities. Hopefully this conversation will resonate with their experiences.
Something I haven't mentioned yet, sorry to disrupt you. We wouldn't have come anywhere without talking to the students. So they have been extremely instrumental in making sure that we adjusted things as we went along. And only we could make it work in this way. So again, engaging students in their in the development of their education is important in normal days, and particularly in this context as well.
Okay, thank you. And so, what do you think you've learned, and are you positive going forward? Not just you, Beatrix.
Right. Am I half glass full glass half full or empty person. Now you caught me out. Right, so I think we have all learned a huge amount, and it's now another learning curve in making sure that when we transition back, that we apply what we've learned in the best possible way. So as I said earlier, keep the really, really helpful things for students and for staff. And again, we are in conversation with everyone to maintain some elements of flexibility, particularly the authentic context, the active context, helping students to thrive even if we are coming back, because again, this will cause other issues for some of the students who might be, worried coming back or meeting suddenly loads of people again, not even infected ones, but just being in a crowd again. And again, I imagine helping each other in our networks through learner societies and in everyone's institution, working together going forward. But what we've learned, that we can deliver blended activities, something that then helps going forward to be more personalized, so that students at any time in their education, we are now of course talking about the traditional higher education, but of course how education at some point will be is that you of course never stop learning, and so you might do some top up here and there, or you mix and match maybe already your program as you are in your main education. So having learned how one can blend things together and run robust and reliable assessments, even if they are not on campus, so all these things we've learned now will help to make that happen as of lifelong and life-wide and technology supported, going along, looking at your phone and suddenly you've learned something that doesn't require you to sit somewhere with other people in a room at any age. So we're looking forward to that. And I think having gone through this, we've learned that we can do those things where we before may have been a bit more hesitant. Oh no, that's not possible. If you are forced into it, you realize it is, and then you can see what you take from there. Of course, ideally we wouldn't have been forced this way.
Thank you very much, Beatrix. Joe, have you got any other questions that you want to ask? We've got another couple of minutes, have we?
Oh, well, yeah, we've got about, let's have a look, 18 minutes actually until, well, you've got some, you want to talk about the Bad Bugs Book Club in 3.
Yeah.
So I had one more question for you, Beatrix, and that is, term is beginning right now. Are the kids and the students coming into campus, or are you doing a hybrid system, or is it mostly online, or are you really uncertain as to how the winter is going to go?
So yes to all. So a very complex again, and I of course I'm in my institution, but of course I observe what's going on in the sector, and again different countries will differ because they have different local risk rates and different weather and so on. But we are preparing mostly for in-person in terms of the value that offers, not by sitting in a lecture, which of course isn't necessarily as valuable as we said before, talking at someone, but by having the opportunity to interact in person in applying the knowledge and the developing the skills, rather than doing that online and the whole community building. Of course, we are prepared for going back into more online if that was necessary. So we've got all that under our belt. But I think, and again, depending on the institutional size as well in the sector, so some will have some more online elements, particularly where they have larger groups, and some will be able to make more use of smaller group contexts where it's safer. But I think particularly also driven by public perception that online wasn't valuable, which I hope now everyone heard that isn't necessarily the case. It depends what you're doing online. You can be doing something really awful in person and it's totally without value. So it really depends what you're doing. So I think part of that is that there is a lot of face-to-face as well, so that there is no perception that students are not prepared very well. not perceived by the students or anyone who would then receive students later on, which is important to not, give impression that something isn't valuable, even if it is. So hopefully looking forward to being in person and hoping that won't change anytime soon.
Yeah, well, best of luck
Thank you.
to you and your students, and thank you for joining us today. And enjoy International Microorganism Day as it begins tomorrow.
I will. Thank you. Thank you all.
Thank you very much.
I think you've got my slides, Joe.
Let me just double check, because you emailed them to me, didn't you?
I did.
I'm just going to go grab the keyboard for 2 seconds.
Okay. I'll just introduce it anyway. So continuing through with this education theme, listening to Kenneth Timmis's discussions about microbiology literacy, and then the discussion with Beatrix about university education, I kind of wanted to combine the two of them by telling you a little bit about the Bad Bugs Book Club, which is a public engagement activity which I set up in 2009. So should be some slides.
Yeah, just coming through now. And just give me a heads up when you want the slide.
Okay, thank you very much. Yes, so this is the Bad Bugs Book Club, the website. It comes out from my university, Manchester Metropolitan University. But if you search Bad Bugs Book Club, you'll find the website. So the aim of the Bad Bugs Book Club I set up, as I said, in 2009, was to encourage scientists and non-scientists to talk about novels or fiction where infectious disease formed part of the plot. My idea was that it was particularly aimed at non-scientists and to encourage dialogue really between scientists and non-scientists, but in a way where everybody was coming to a meeting having the same experience behind them. In other words, everyone has read the book, everyone has got different opinions about the book. The scientists will know some more microbiology, the non-scientists will have different perspectives on the book. So everyone has got something to say. The idea is, again, to engage in discussion, particularly about infectious disease, but we have actually also ended up talking quite a lot about literature, which has been a really nice cross-disciplinary extension of my original intention. So this is the homepage of the website. The next slide is a part of the website itself. In fact, the website is pretty dull, really. If you just go down through the middle of the website, you just rack through, you get pictures of the covers of all the books that we've read with white space on the side. So I've just put in a few photographs of some of our book club meetings and a picture of me 2 years ago with some of the books that we had read. So you can see the pile would be a lot higher now. For each book that we've read, there is a downloadable meeting report, which tells you what we talked about and what our opinions of the book was. But there is also a reading guide. So these are questions which are designed really to encourage people to talk about the microbiology, particularly in these novels. We don't always need all of the questions in the reading guide, but they are there really to kind of guide the reading or to make sure that people have something to say. And since 2009, we have read more than 70 novels. It's quite incredible that there are that many. And the book club itself has got about 8 members. So, and of those 8 members, about 4 are the original. We have people who stay with the book club for quite a long time. Some come and go, but a size of 8 is good for having face-to-face discussions. So that I would recommend if you were going to set up a book club would be a good sort of size for discussion. So the next slide. The book club really is aimed as a public engagement activity. So 8 people obviously isn't very many. So often we have book club meetings within larger events from Manchester Science Festival or World AIDS Day or other activities. We also combine the book club meetings with guided walks or film screenings. We've had community quilt making for World AIDS Day. We've had a music event when we were reading books about malaria. So there's a whole range of different activities. And as I said before, if anybody wants to set up their own book club, obviously all the information is there on the website. One of the things that has been really exciting for me, though, is having read all of these books, you start to get a bit familiar with some of the themes that occur in some of the books. I started reading about Dracula and Twilight, particularly as vampirism as a sort of model of some sort of infection that's transmissible, and then started to go on to zombies, and zombies as a model of infectious disease provided quite a rich scene for mining for a whole range of different microbiology education activities. I also want to tell you something that I have observed about influenza infection, AMR infection, and something about coronavirus. And just recently I had a paper published in the FEMS Microbial Letters, which says a bit more about the book club and its activities. The next slide. So zombies as a model for infectious disease, they're probably a bit too successful because they tend to be sort of 100% transmissible, so the world would end really. So in many of the books, there has to be some sort of struggle, and then they are beaten, or they have to modify themselves so that they can live in a world with humanity. So there are plenty of themes that you can pick out reading about zombie novels and zombie apocalypse, which is actually quite nicely translatable into infectious disease and infectious disease control. And a colleague of mine, Xavier Aldana Reyes from the English department at my university, worked together on a paper, Emerging Infectious Literatures and the Zombie Condition, which we published in the CDC Emerging Infectious Diseases Journal, which I was so proud of. And you can see the other paper I published with some of my mathematician colleagues, where we produced a computer-based model, a simulation to show how zombie epidemics can spread depending on the different parameters you input to that program. So I never knew going into a book club that I would end up learning all of this. So it's not just learning for some sort of audience, but also for yourself when you're taking part in something like this. Next slide. These are just some more books. On the left-hand side are some books where we've had, for Manchester Children's Book Festival, a whole series of young adult books. Code Orange is about smallpox, Fever 1793 is about yellow fever, Twilight's vampires, but there is mention of flu, and The Enemy is another zombie-type movie. So we have used those books, Warm Bodies, also zombies. We've used those books to engage with young adults about infectious disease control. And then for younger adults, we had the tale of the 13-week-old moldy cheese and pickled egg sandwich, which we simulated and discussed with the author, who's there looking at me, looking down a microscope with me, about a boy who ate it. The public engagement activity was in part to advise not to eat 13-week-old cheese and pickled egg sandwiches, as well as other aspects of hygienic sandwich making. The next slide. If you look at certain diseases that are popular in fiction, you wouldn't be surprised that plague was one of them. There are many, many books about plague, let alone the more famous ones. And the sort of scenarios of plague fiction are often set, say, in the time of the Black Death, such as Minette Walter's series, or during the time of the Great Plague of London, such as Daniel Defoe's and Forever Amber, which is a bit of a, I think you call them bodice ripping yarn. And then there are also quite a few more modern spins where there is a release of a virulent plague bacillus. So plague is really conducive to fiction, not surprisingly, because it's been with us right through history and causes very dramatic symptoms and pandemics. The next click. The book that we're reading tonight, we have a book club meeting tonight, is Hamlet by Maggie O'Farrell, which is a fictitious account of the death of Shakespeare's son Hamlet. In this book, it is from plague. So we're going to be discussing that very, very soon, in fact, at the book club meeting. Next slide. There are very few books of fiction about antimicrobial resistance. And when you compare those with novels, say, about plague or HIV, and I wonder if it's because it's quite hard to spin a story around AMR, which at the moment tends to be more stories about individuals. So The Infectious Futures is a series of short stories projected into the future. Fierce Radiance is about the discovery of antibiotics at the end of the Second World War. Deep Zone is about the discovery of antimicrobials in extreme environments. Robin Cook's Contagion is bioterrorism. But last year we read Eve Smith's The Waiting Rooms, which is a fantastic book about a world in which antibiotics cease to work. It's a near future book and resonates very much also with the coronavirus pandemic in terms of isolation, the experience of the elderly in care homes, and the changes in behaviour. And The Perfect Predator is a real life novel about phage therapy. So there are some AMR novels starting to turn up now. So the next slide. Influenza is again another disease which has many, many novels. And again, interestingly, some of them are based on the 1918 pandemic. So The Last Town on Earth, The Pull of the Stars, which one we read very recently. But then there are also pandemic novels where there's an influenza pandemic that has killed everybody. And the nature of the story and the nature of the plot line is related to essentially the virulence of the influenza virus. So if you have something which is highly infectious and highly virulent, you end up with an apocalyptic type novel. Station 11, Lovely Way to Burn, and The Stand are examples of those. And then there are others. So The Health of Strangers, which we discussed at the FEMS Congress 2 years ago, is about the management of a pandemic afterwards, as is immunity. So in those cases, you're living with influenza and living with the aftermath of it. And I think you can probably see from what I'm saying, there's plenty you can extrapolate. But the next slide is what I wanted to come towards the end of the talk about, is the coronavirus pandemic obviously had a massive impact on my book club. The 8 of us couldn't meet anymore face-to-face, so we went online and met on Zoom. And actually, this has been transformative in terms of the book club, because we now have international members. We still have relatively small numbers so that we can have a discussion. There's about 12 of us, but we have members from America and mainland Europe, and also not just in Manchester, of course. We have others from the UK. And every book that we've read during the pandemic, which are these ones on the slide, have enabled us to explore what's been going on with the pandemic at the particular time we've read it, which is something that obviously we didn't know was going to happen. So Not Forgetting the Whale is about community spirit in a flu pandemic in a little village in Cornwall, and how people work together and support one another. John le Carré's Constant Gardener is about clinical trials. So we were able to explore the vaccine trials at the time. The Pull of the Stars is about influenza and how people, again, were living during 1918 and how they were coping in hospitals, particularly during that time. So we learn about coronavirus through reading novels of fiction about other books. I just wanted to mention at the end, our last meeting was Michael Rosen's Many Different Kinds of Love, which is his experience of having COVID, and it's a really, really moving and beautiful prose narrative of his extreme illness and lengthy recovery. And Ali Smith's Summer is a narrative just about other things that happened during the summer of coronavirus. She obviously wasn't anticipating the coronavirus pandemic when she wrote these near to present time novels, but had to cope with it. And the number of hits on the website shot up, the number of engagements with the book club shot up as well. So in all of my attempts to try and promote the book club as much as I could, coronavirus is what actually helped that to happen. So my final slide is just to say that the Bad Bugs Book Club started off as sort of cross-disciplinary interactive engagement to get contribution from other partners, scientists and non-scientists. Everybody who comes are already motivated, they enjoy reading, so discussion is always easy. But the corona pandemic allowed us to extend our audience, extend the meetings, extend the membership. We had authors joining our discussions online as well, which was great. Social media helped us to encourage others to join in our discussions. I haven't mentioned the book club as potential work for students, but I did use it in class, and I also used it in student project work. So it is potential for that as well. I've also used it for school children. We have a schools Bad Bugs book club down in Cornwall where they regularly have read the books, again, all through the pandemic. I've tried also to evidence what I've done by publishing in peer-reviewed journals, just so that there's still evidence that the book club exists as well as the website. And I just wanted to finish by, I never knew how much I would learn as well by being part of this book club. I never would have thought when I set it up that that would happen. Anyone who wants to, just have a look on the website and email me if there's any other questions that you have. So thank you very much.
Thanks, Jo. I mean, the number of novels there are just incredible that feature something about microbes in some way. And I guess they're usually written, you know, in a very accessible way for tons of different people. So it's a wonderful way of encouraging conversations about microbiology. So thank you for sharing this fantastic initiative. I believe you've got to shoot off now to the Bad Bugs book club itself.
So
And
thank you very much.
we've got another speaker. So I won't keep you any longer. Thanks so much, and enjoy International Microorganism Day tomorrow and celebrate as best you can.
Bye.
See you soon. I'll just admit now our final speaker for the education session, which is John Warhol of the Warhol Institute, and he is just joining us in the live stream. Hello, John. I think you need to unmute yourself, and then we can talk. Let's send you a request.
Can you hear me now?
I can. Thanks for joining us.
Thank God.
I would say this is a wonderful little story because I think we bumped into you on Twitter talking about education practices, and then managed to invite you to join us to give a talk about public engagement in microbiology. And now here you are with us in person on, well, not quite International Microorganism Day, unless you're in Japan or India or nearly, but on the doorstep of the day. So I believe you have a presentation for us about ways to engage the public with microorganisms.
Oh, I do, sir, and I hope you all enjoy it. Let me click the share screen button if it's appropriate to do that.
Absolutely.
Where is it? There we go. Share. How's that look? Is it up? Hi, is it working?
All working. You're free to talk for the remainder of the half hour.
All right, wonderful. Well, thank you very much for having me here, to say the least. Oh. I am here to talk to you about getting engaged and being educated. Thank you so much for inviting me. It's a pleasure to be speaking before an international audience. At this point in my life, there's only one thing I really want to do, and it's not pipetting. All I want to do is change the world. I want to change it from a place where when people hear the word microbe, they no longer go, I don't know, I don't care, but instead say, wow, that is cool. Tell me more. But doing this is a little bit like being Daniel in the lion's den, or it's being like Hamlet, who suffered the slings and arrows of outrageous fortune, because it involves engaging the public. And engaging the public always sets up an us versus them or a me versus them situation. And this is truly outside of the comfort zone of most people. But engaging the public is really not too bad as long as you remember one rule of engagement, and that's this: microbes have stories. You need to tell those stories. And one more time for emphasis, microbes have stories. you must be able to tell those stories. Why? It's a simple question. We all know microbes are undoubtedly cool, the coolest thing on the planet, but there's not much to see, let's face it. So microbiologists need to tell what's cool about an organism and relate it to the larger world so people can understand. Having said all that, now it's story time. Here's our first story. It is simply called The Most Amazing Thing in the History of the Known Universe. And this happened only 2 years ago. We should all know about it. 2 years ago, New Jersey here in the US declared, we have a new official symbol. It's a bacterium. And that bacterium was Streptomyces griseus. The New Jersey Assembly and the New Jersey Senate and the governor all voted unanimously to make Strep griseus our official state microbe. Are you surprised? Is everybody out there surprised? Most people in New Jersey don't even know this. The history of official microbes is short and sweet. Oregon was the first. In 2013, they picked Saccharomyces cerevisiae in honor of their $2.5 billion a year craft brew industry. New Jersey in 2019, Streptomyces griseus in honor of its production of streptomycin. And Illinois, the state of Illinois, only 2 weeks ago became the third place in the universe to have an official microbe. Theirs is Penicillium rubens, which used to be known as Penicillium chrysogenum, the producer of penicillin. Why should anybody care about state microbes? To me, it's an important step in global microbial literacy. Because every place, for me, every place needs an official microbe, simply because every place has microbes and every place has official symbols. Official symbols are things that represent the character, achievements, aspirations, values of a place. As such, they could be anything. Here's some on the screen: sheep, spiders, dogs, cats, pheasants, even Kool-Aid. Official symbols can be anything, even microbes. For example, if you're fortunate to live in Roquefort-sur-Suzanne, France, it behooves you to approach your legislators and say, we'd like to have an official microbe. We think it should be Penicillium roqueforti. This is the organism that gives Roquefort cheese its characteristic blue flavor. This should be easy. The whole town of Roquefort has 700 people. I'm sure they all know each other. That's the same size as my high school graduating class. Petition your legislators, put yourself on the map as the 4th place in the world with an official microbe. Another good choice, or another alternate choice, Korea, is a wonderful organism called Vicella coriensis. It's essential in the fermentation of kimchi. It's a really cool organism because it works at below 0 or below 32, depending on where you live. It's one of 36 different organisms that are involved in kimchi fermentation. Now, to my mind, it would be great if both North and South Korea agreed on this. You can imagine that 2 nations have been at war for decades, and they both agree, we're going to have an official microbe. It's going to be Vicella coriensis, and the 2 of them are happy and get together, just like they did here in New Jersey, for a brief moment when all the Republicans and all the Democrats said, we're going to go with Strep griseus. And if you're from a place where beer, wine, or bread are important to your culture or your economy or your history, go with Saccharomyces cerevisiae. It's a wonderful organism. So now that I've convinced everybody that they have to go out and get their official microbe, how do you do it? First step, discuss among yourselves. This is not a one-person job. Pick something that makes sense. It has to make sense geographically or any other way you want to do it. It doesn't have to be discovered where you are. It just has to be important to where you are. And once you get it, stick with it. Don't change it halfway through. That results in disastrous outcomes. You can ask me about that later, but stick with it. Once you know what you're going to do, communicate it to infinity and beyond, in the words of Buzz Lightyear. Use every available method to tell everybody: Instagram, Twitter, Facebook, get a hold of your legislators and tell people. It's really important when you do this, it's really important to get the press on your side at the beginning of your cycle, because if you don't, it can be brutal and set you back weeks or months. And it's also very important to get students involved. Legislators love students. If we have time later, ask me about the New Jersey bog turtle, another one of our official symbols, and I'll tell you why students are important. You can get an official microbe because democracy is a participatory endeavor, and participation is basically easy. Legislators have readily available contact information. They have websites, they have internet, they have email. They're out there for you to contact. You just have to do it. And it's easy to do because microbes have stories and we can all tell those stories. When you tell your stories about your favorite microbe, whether it's for your state organism or anything else you're doing, your stories have to be to the point. They have to be brief, they have to be interesting, and they have to be compelling. And I have to drink. Having said that, the story within the story is for Strep griseus. Strep griseus started with a 2-minute elevator pitch that I was able to give to a lot of legislators at various times here in the US. The 2-minute elevator pitch is based on the premise that you're stuck in an elevator with somebody really important and you got to tell them everything. So what do you tell somebody who knows nothing about your favorite organism? The Strep griseus story goes something like this. Mr. Senator, New Jersey absolutely, positively needs to have its own official state microbe. And here's why. New Jersey is home to the microorganism known as Streptomyces griseus. Strep griseus produces the antibiotic streptomycin. Streptomycin is the first antibiotic after penicillin, the first American antibiotic, and the first antibiotic to kill tuberculosis. Streptomycin changed the world, like I want to do. Streptomycin producing strain of Strep griseus was discovered in New Jersey, in New Brunswick, in dirt in 1943 by Albert Schatz. Dr. Schatz was supervised by Dr. Selman Waxman, who won a Nobel Prize for his work on streptomycin, Strep griseus, and the systematic study of soil microbes. Strep griseus and streptomycin gave birth to the American antibiotic industry, an industry that employs thousands of people, starting with drug discovery and ending with use in patients. Recognizing Strep griseus as the state microbe honors the contribution of all those people, but it does more than that. It creates the opportunity to teach basic science to people, to have them understand that microbes are essential to all life on Earth, and most of them are beneficial. And here's a copy of the proposal that this all came down to. If you go to do a project like this, you don't need a thesis. You don't need, if people still use them, a phone book. 1 page, bullet points, hit the highlights. Fortunately, I was at a political meeting and I was able to put this in the hands of legislators who were able to make the process happen. Remember, democracy is a participatory endeavor. As scientists, as educated people, you should get involved. Attend fundraisers or volunteer. There is no substitute for face-to-face contact with your legislator. And a really good way to do that is to invite them to your lab. Explain what it is that you're doing. You should be able to make a pitch and make people understand what you're doing, even if they're not microbiologists. But why do this at all? Again, it's not for a pat on the back. It's not to make you feel good directly. You do this because it creates the opportunity to teach basic science to people and have them learn that microbes are beneficial. Because if, like the US, microbiology is not part of your normal science curriculum in K-12 schools, It will give students a chance to learn some science when they're doing civics and social studies and they're learning about their official symbols. Having a state microbe or an official microbe of a county or a city or a region is a first step to creating broader understanding of the microbial world. Speaking of the world, the second story is called Around the World. And this is fun. And it goes like this. I have been around the world, and I have come to understand deeply and painfully that nobody knows nothing about microbes. And I said, my God, what a shame that is. So I searched for an answer. I searched far and wide, hither and yon, until at last I said, Eureka, I found it. It's the periodic table. The periodic table is the single most iconic element. It's the single most iconic thing in all of science. It hangs in every classroom from about the third grade on. People see this, they know it's science, they know it's chemistry, they know it's elements. They might not know anything else about it, but they've seen it. And I looked at that and I said, oh, I could use that. I can use that to make connections between something that people know, periodic table, and something they don't know, microbes. So I grabbed the periodic table and I shook it really, really hard, and all the elements fell out. And I replaced them with microbes. And I got something that looks like this. It's the periodic table of microbes. To do this, I went through 15,000 named organisms. I'll say that again. 15,000 named organisms. And I pulled out the ones that were cool, significant, or interesting, things that had stories to tell. And more importantly, they were organisms whose name could be reasonably abbreviated by a chemical symbol, like C for carbon became C for Clostridium on the table of microbes. And so that it's not just a pretty wall hanging, I came up with a legend, so that when you look at the table and you look at an organism, you can immediately know what it is. Like H for Haemophilus, it says negative RP in the corner, negative, gram-negative, R, rod, P, pathogen. The other side of the screen, leuconostoc, positive CE. It's a gram-positive coccus, and E is for edible, so it's used in food production. This all made perfect sense to me, and it makes a beautiful wall poster, but as we all know, microbes have stories, and unfortunately, all those stories were just in my head. So in order to give people access to that, I did the companion book. It's called The Small Guide to Small Things. It's called The Small Guide because it describes 118 microbes in 300 words or less. Brief. That's an abstract. It's an abstract per organism. In addition to the 300-word summaries, it contains an introduction, which is something that will orient every reader to the world of microbes. It's the absolute minimum you need to know, or Joe Q public needs to know, in order to understand what microbes do. If somebody reads this, they will be able to have an intelligent conversation with their physician, with their nurse, with their next door neighbor who's a microbiology professor. To help orient everybody to what it does, I came up with lists, like the top 3 microbes in a variety of categories, like the hottest, the coldest, the deepest, the loneliest. I like the loneliest. It's Modestobacter. It lives in Antarctica on an ice shelf inside of a rock. It has no friends. And then lists of people, places, things, and animals that are all associated with microbes to help make the link between the bigger world and the microbial world. And I validated it. During the course of production, I validated it with blind reviews. I don't really know a lot of people because I'm basically a hermit. I sent it to the National Science Teachers Association. I had 36 people, 36 science teachers review it blind. I didn't know any of them. They looked at it and I was getting reviews back like, this was so good. I blew off my prep class, this prep period this morning before school, and I just read it from start to finish. Elio Schachter saw it after it was all done, and he said, John, we need this. He loved it. Doug Evely helped me do the editing on it, and he was positively giddy at points at the book. And most important for me is James Burke. James Burke is the world's greatest historian of science and technology. He had 12 BBC TV series. I sent him the draft cold. I don't know the guy. I thank God that he picked it up and looked at it, and he read it, and he had this to say. This is a wonderful book. I rarely enthuse, but this is outstanding work, immensely enjoyable, and full of really bad jokes. And that's part of my process in there. It's a fun, enjoyable, engaging way to introduce people to microbes. Or if you're a microbiologist, there will be stuff in here that you haven't seen because there's just so many of them. What I like to tell people is that it costs less than a burger and a Coke. It's more fun, and it lasts a lot longer. My third story about microbes is called Fashions Lead to Engagement. Excuse me. Fashions lead to engagement. We're all here to learn how to engage people. Well, we know microbes are not much to look at, but there are ways around that. For example, when you're working on your state microbe, and I know you all will, or you're working on other projects, put it on a polo shirt. Put it on the front and back of a polo shirt so that people can see you coming and going. I wear this shirt and others like it all the time. It creates engagement if I'm in the supermarket buying food. It creates engagement if I'm out on the street. And see, people will look at it and say, hey, what is that? And you'll get other people who will look and say nothing, but it creates the opportunity for you to say, hey, I see you're looking at my shirt. Let me tell you how wonderful microbes are. And if you're really, really, really lucky, your microbe becomes a Jeopardy question. You can put that on a polo shirt too. On April 5th, 2021, the state microbe became an official Jeopardy question. Thrilled the heck out of me. Lower end of the budget, if you're having a project, go to anybutton.com on the internet and get your favorite project put on a button. It's a lot cheaper than a shirt. It's portable. People collect them. And these are like a dime each. And you could do them in any kind of quantity and size you want. Extremely cost-effective engagement tool. But what if you don't have a state microbe and you need a preformed graphic element? What do you think you could use? Well, I said, hey, I have this table. Let me put it on a tie. So I did. And I have a friend who's a virologist and specializes in norovirus. And she said, hey, could you give me a norovirus tie? And I said, sure. And that's there. And on the other side of the screen, we see some guy wearing that tie on a TV interview. It stands out. It's a really wonderful tie. But here's a quick story. People watching this are all scientists. Here's the science story. I wore this to a science meeting. None of the scientists in the room said anything. Nothing. But a dozen of the facilities people in the building said, wow, that's a nice tie. So this is the person checking your ID, the person getting your coffee, the person, just people there. There's a dozen different engagement opportunities based on just wearing a tie. And I have the same experience when I go out and I talk to legislators. They'll look at it and say, nice tie, or they'll just look at it and I'm like, oh, well, let me explain my tie to you. And it creates a chance to talk to people about your science. And microbiology is important. And okay, if you don't wear ties, you could wear scarves. I put it on a scarf. Story behind the story of this is, during the first stage of our pandemic, our Dr. Birx was always on TV every night. She wore fashion scarves like you wouldn't believe. And I said, it would be really good if she had one of these because it's educational, it's not just fashionable. And I tried to find her address to send her one. And I discovered that the government thoroughly and absolutely scrubbed all contact information for her. So if any of you know Dr. Birx or her people, have them get in touch with me and I will get her a Table of Viruses scarf. And then if you're not a scarf or a tie person, you could always put your favorite organism on a t-shirt. Okay, my last and fourth story is called 200 Miles of Fishing Line. You all are probably saying, what does 200 miles of fishing line have to do with anything? Well, we all know microbes are undoubtedly cool. We know there's not much to look at. And we know you need to tell stories and be able to relate your organism to the larger world. And we also know Strep griseus is the official state microbe of New Jersey. So put them all together, what do you get? Well, during the run-up for the state microbe activities, my good friend Doug Everly said, hey, John, I think there's a lot of this stuff in the dirt. Let me do some calculations. And he did. And it turns out that there's anywhere between 75 and 280 miles of bacteria, of actinomyces and streptomyces per square foot of Earth. That's a lot, but you got to conceptualize that. First of all, a square foot is something the size of a Domino's pizza box. It's a medium-sized Domino's pizza, not even the large, and it's an inch deep. And 200 miles, what's 200 miles? It's the distance from New York City to Washington, DC. But to me, that's still a bit abstract. It's a 4-hour ride. And I'm like, how can I get this across to students that is 200 miles of anything in a Domino's pizza box? And I thought, and I said, well, it's a filamentous organism. What's the closest thing I can come up to with filaments? And the answer was fishing line. So I took it upon myself to find 200 miles of fishing line so that I could do a demo for people. And I went to every bait shop within 20 miles of my house. And I ran in the front door and I said, Mr. Tackle Shop guy, I need 200 miles of fishing line. And he said, why? And I said, state bacteria, on and on. And he said, I don't have that much. Try the guy down the street. So I repeated that about a dozen times until finally the last guy said, well, why don't you just call the factory and order some? So I did. I called the fishing line factory. And the guy there said, well, yeah, I can send you 200 miles of fishing line, but you're going to need a shopping cart to carry it around. And I said, well, that's really more than I need. It's not convenient for bringing it to a school or anything. And he said, how about 20 miles? I said, yeah, 20 miles would work. So he sent me 20 miles of fishing line. And here it is. This is a stack. Each one is 2 miles of fishing line. There's 10 in there. And it gives me 20 miles of actinomyces or streptomyces. 20 miles of bacteria is roughly the amount that's inside of an area or under an area the size of my iPhone case. That's a lot. That's a lot of really, really small organisms. And if you do a demo like this and you really want to have fun, stick a pencil in the end of one of the spools, take the loose end and you give it to a kid to run with, and he could run around the school 10, 20, 30 times and you don't even make a dent in it. But what's 20 miles or what's 200 miles? And I started thinking, and I have this internal conflict between microbiologists and astrophysicists because they're always using really big numbers and they have really, really great television shows and we don't. So I just started playing with the numbers and I said, well, if there's 200 miles in every square foot, how much would there be on planet Earth? So I multiplied 200 miles by all the available surface area of the US, but only an inch deep. And if you had sufficient time and really small forceps and a lot of grad students and postdocs and everybody else helping you, and you unraveled all the organisms and laid them end to end, you would get a string of bacteria 8 quadrillion miles long. That's 1,360 light years long, which coincidentally is the length of our galaxy from side to side. I just find that amazing. I find that amazing. It's like we have an infinite amount. We have an infinity of smallness beneath our feet. And I think people, it's like you tell them that. It should make them sit up and take notice. Okay, my time's up. I have rambled on for 25 minutes and I thank you for listening. Now it's time for the summary. Really quickly, microbes have stories. Tell them. Tell them with passion. Probably talk a little slower than I have here, but tell them with passion. And why should you do this? You should do this because ignorance is always bad and knowledge is always good. And by doing this, you put yourself on the side of good. I want to thank you all for listening to me. And here is my contact information in case anybody wants to get a hold of me. Thank you all.
Thank you, John, very much for that inspiring talk. You have
inspirational.
Yeah, you've inspired us. We have a podcast, and we were called Microbes in Us, which you can find on Spotify and all good podcast platforms. I think we'd definitely like to invite you to it, to talk with us, but we'd also love to be able to, we're going to order a copy of your book, and I think we're going to do an episode talking about the different stories in your book. And we'll tell some stories about microbes. I think all the advice you've given here is really straightforward, simple, and great advice.
Thank you.
And we'd love to collaborate after IMD on your geographical compendium of microbes. And also, I mean, we'll do our best to push efforts to get a Netherlands microbe as best we can, but we'll see what we can do. But yeah, thank you so much for that fantastic talk. Honestly, we'd love to chat for longer, but we have to move into the next session, which is a live microscopy session, which you can watch and join in. But we'll try and get you back for the podcast. We'd love to chat to you more.
I would love to talk. My saying is, anytime, anywhere, anybody wants me to talk about bacteria, I'm there.
Brilliant. Well, we'll speak very soon, and thank you again for your wonderful talk.
Thank you so much.
We'll see you soon. Good luck on International Microorganism Day tomorrow. I hope you celebrate it well and go around and tell every fish shop owner that it is International Microorganism Day and they should be celebrating.
Honestly, God, I don't make any of that up.
I believe it, and that's fantastic. So yeah, we're now going to move into the microscopy session. So we'll say goodbye to you, John.
Bye. Thank you all very much.
But see you soon for sure, and enjoy your evening.
Goodbye. Thank you.
Goodbye. Now we move into the live microscopy section, and I will.
Am I still there?
Yeah, a little bit, but if you wanna, we can mute you, and then you can just watch. Hi, Hunter, you're with us. You're muted, so if you could unmute. We just lost James. He will be with us back, I hope, in a second.
Hey guys, how are you?
Yeah, we're good. We're now we're into the 9th hour of the stream.
Oh, that's easy. Still early.
Yeah, still early. Just 3 times this to go.
Absolutely.
I think we've got James with us as well. So for this session, Eleni is going to be operating the microscope.
Right,
are you able to hop into the Zoom call on your own account and share the screen when you have a moment? And we'll just have it going in the background. And you can put slides up. For the first 15 minutes, if we can get James online, I have some interview questions for him, similar to the ones I asked you, Hunter, last year, because of course you were here last year for our microscopy session.
I was, yes. Lots of fun.
Yeah, and are you in the lab this time, or where are you joining us from?
I'm joining you from my home lab today. It's a lot more quiet and less work to be done. So, yeah.
Yeah, okay. And, well, we're joined by a bigger and more powerful microscope this year than last year.
Yeah, beautiful. Front and center of the screen. I love it. Huh?
Yeah, it's been here as our mascot and our prop all day. We also have IMD hoodies this year, which contain all 4 mascots.
Wow.
So here we go.
Hello, hi.
Hi, James.
I'm having some technical difficulties with my headphones.
Don't worry, I can relate. But we're with you now. So thanks for joining the microscopy session. So yeah, Eleni is going to be our microscope operator, and we'll start getting some slides up in the Zoom call. We've got 15 minutes, James, to ask you some questions, and then we'll just have a discussion, and you guys can share videos that you think are cool. So the reason both of you here is you're both giants of microscopy Instagram, is how I like to put it. You have over 100,000, approaching 200,000 followers each on your various accounts, at Microbial Ecology for Hunter and at Jam and Germs for James. And you're both also quite big on YouTube and bringing microscopy and microbes to, thousands of people. Here we go. Here's our little microscope. Here's Jam and Germs' account. And, I just think it's 2 great examples of a really, really fascinating way to touch into loads of people's content streams with really beautiful images. So I'll start with some questions for you, James. What got you started with @jam and germs? What got you going with the account?
Well, honestly, it was like an accident. Let's put it in that way. So I was taking a college course, and then we had to just do some microscopy to just assess the wastewater treatment plant, like the health of the wastewater. And then I saw my first microbe under the microscope, and then it just blew my mind. And then it was 4 years ago, I think, and since then I did nothing. It was just microscopy, and like 2 hours a week lab course wasn't enough for me, so I got a microscope, and I wanted to share my videos with my professors. So I started on YouTube first, and then one of my friends, she was saying like I should get an Instagram account and just start there. And they made a joke about the James's and germs, and I never thought it's gonna be something big. So I started my account, and it got a lot of attention, and yeah, it's been like almost 4 years, and I keep doing that. So, and also like I have probably hundreds of thousands of microbes in my home, but I also have 2 cats, and it's their zooming time. So I'm so sorry in advance. You might see like cats jumping in.
Oh yeah, jumping in. I mean,
yeah.
I had a cat for a while last year, and it was just always coming across the Zoom call interface.
Yeah,
yeah.
I built a cage for my like overly expensive microscope, so and they're just zooming on top of it right now.
Amazing. So yeah, you said that this account started growing really fast, or at least it got very big. Do you know who your main audience are? Is there kind of a demographic spread you can see it tilts towards, or is it just completely diverse?
So I think it's because my account is mainly in English, in English. So I get I think around like 30-something% of my Instagram audience comes from the US, and I have like 7-8%. I'm sorry about the
we can hear them. No worries. Yeah.
So I have like 7-8%, I think, from the UK. So we can say like half of the audience is coming from the English-speaking countries.
Mm-hmm. Yeah. Okay. And then the rest are just from all over the world. Do you think?
Yeah. Because on Instagram, I think you can see the top 5.
Yeah, I think something like that.
Yeah.
Okay. And you mentioned that you started off, labs were not enough, and so you got your own microscope. So I wanted to know, what equipment did you begin with, and what equipment do you use now?
So I started with the cheapest microscope on eBay. It was like I think $160. I ordered it, and it was let's call it non-branded, but it's like such a generic microscope. So if like anybody wants to start microscopy, they can just go and look for compound microscopes. And because also like we produce some microscopes with our YouTube channel. So we have like pretty standard objectives and stuff. So every budget microscope, they use the same optics. But the thing is with microscopy is it's like an art. You have to practice, you have to know how to prepare your samples, and it changes from microbes to microbe, and you have to know, you have to experience all these things, all these techniques to get some good image with any microscope, So it's like I'm really grateful that I started with such a bad microscope, actually, because that taught me a lot how to.
And you had to get good with it. Yeah. Okay.
Yeah. And then I got so now I think I completed my light microscope adventures. I have a Zeiss microscope at the moment, so I'm using DIC contrast, and I have fluorescence light module for, like giving things under the UV light or monochromatic light like blue, green, and red.
And are they just in your home?
Yeah. Just
Nice.
Yeah, my cats were zooming on it.
Yeah, okay.
I
But so
built something around it to prevent the dust going inside of it.
Yeah.
And yeah, so yeah.
And do you just have a microscope room now in your house? Or is it?
No, not really. It's just like in my living room.
Okay.
Yeah.
Nice.
I'm just my stay here in Poland is temporary, so I'm gonna be in the US, I think, in like 3 months.
Yeah.
And then I'm gonna get probably a room.
Okay. But so you're in Holland at the moment?
Yeah.
Oh, well, we're in Holland. We could have done this in person almost.
Poland.
Oh, Poland. Sorry, I misheard you. That would have been just too good. Okay. So, yeah, I mean, one question I had for you next was what microbes do people seem to enjoy the most of the ones you've posted?
I think people like to see something dynamic. So it depends how you're just presenting your microbe, let's say. So people they love water bears, for example, but they also love stentors. So we started we made some stentor videos and they gone viral, and now everybody knows about stentors. It's like these are like giant microscopic.
You have one behind you, James, right?
Oh yeah.
Oh, nice.
Not real size, obviously.
Yeah. Okay. So that will help people wanting to know the answer to my next question, which is, what advice do you have for people wanting to share microbe videos online?
Just be original. Just follow your passion and just do whatever makes you happy, and that will be appreciated. So I don't think of my this audience. I don't think of it in a way that it just it comes with your passion. So what you love and people appreciate it. So I'm not seeking more followers, but I'm just seeking to do the thing that I love the most in life, and it's just a byproduct, the followers or anything that comes with it.
So the authentic passion and joy happens to bring people to your account, but yeah, okay, that's good advice. And yeah, this is my final question before we go into a bit more of a free-form discussion. What other microbe communication initiatives should we know about?
I'm not sure about this thing because I think there's nothing else than some social media. Or, am I getting the question correctly? Or
well, I mean, you have there are other communicators that you might have want to point towards. I think also you mentioned that you begun a new YouTube channel more recently. The Microcosm. Yeah, or didn't you?
It's been 2 years. Yeah,
2 years now. Yeah,
we made over 100 episodes, 10 minutes.
Yeah,
long 100 episodes. So it's not that new, but we are just it's wonderful. So people can go check it out, and I would recommend some scientists. I can recommend. I'm sure Hunter knows her because it was her PhD supervisor. She is also my PhD supervisor, so it's we share that one with Hunter, and so people can go check books, from amazing scientists, and I think I think that's it.
Okay, nice.
I'm not nothing really there are some accounts, but no one is doing this in a big way, so we are starting that now.
One of the things that I think someone said last year was space gets so many great films and videos and TV shows made about it, and and microbiology doesn't quite have the same blockbuster presence with long form film media yet. So it's kind of, how can we find ways to do that, I suppose, and get microbes on the TV more in a way that's positive and not just you know, zombies and plague.
Yeah. I have a couple of projects going on, so it's like
Okay.
You can probably we will get a series on a big network. So hopefully we're gonna, you know, reach to more people, and because it's such an alien environment, you know, like as you said, like we know we have so much publicity about space, but we don't know anything about microbes.
Yeah.
So like when people think about microbes, they think about like just blobs not moving, you know, staying and getting you sick. And so I think like we are changing that.
Yeah, and I think making films like the kind that you guys do is a hugely important part of trying to engage and bring people into microscopy and microbiology. Are we able to join the camera, the microscope camera? Cool, because I wanted to in this next stage. We're going to have some live images up on the screen, and B, I wondered if you guys had some videos of your own to share. Any of your favorite videos, either from your accounts or sitting on your computer.
Go ahead, James.
I think you should go ahead now. I was speaking for 15 minutes, so.
No, I already did this last year. They're sick of hearing from me. So let's see what you got, man.
Okay, perfect. So yeah, I prepared some videos, but should we start with this?
So what do we have here, Eleni? We'll just run through this one.
Penicillium.
Okay, so this is a slide of penicillium and stained, I guess, with some nice stains. And I guess are these the hyphae we can see running through here?
Mm-hmm.
Yeah. Okay. Nice. Does it have a video version? This one. You took some videos before, but I can't remember how we did that one.
Yeah, but this is a big slide.
Yeah. Okay. There's not much dynamic stuff to see, but yeah, we have a few like beautiful colored slides like this.
Right.
And I wondered, you mentioned you had some what's the word, fluorescence microscopy that you were attempting. So how did you use that with could you use it with any dynamic organisms, or is it only with static things?
Like I can use it with living organisms. Also, like I don't do photography, so it's like everything has to be something living. And I can actually we can show some fluorescent light like images. From here.
That one was very nice though. I like that slide.
So yeah, okay, I can present. Can I share my screen?
You should be able to, yeah. If you click the share screen, I think it's on the right. Yeah, it is.
I'm not sure what you're seeing right now.
Yeah, we can see a tardigrade surrounded by maybe paramecium or something.
So let's start with fluorescent light microscopy. So we were recently just making a episode about phototoxicity, which is something, like with fluorescent light microscopy, it's a problem because you kill microbes if you expose them to UV light, and there are some microorganisms, they die quite beautifully. So we just I think this Monday we shared a video of this. So oops, this is a short video. I didn't realize that. Sorry. So the red things around here, they're like chlorophyll-based autofluorescents. Yeah, we have some moss leaves and some fungi hyphea here.
So what's the blue moving organism?
This is a microorganism called homolozoan. And I'm sorry, these microorganisms are like Lord Voldemort. You don't pronounce their name.
Yeah.
It's like absolutely necessary. So I'm butchering the names. So I'm sorry for that. So I have some other footage of it. So it's a large single cell organisms in a group called the ciliates, and it shows blue autofluorescence. I don't know why, but they're like probably some proteins. They're emitting this blue light under the UV.
So that's its own fluorescence rather than it's not been stained.
No, this is these are all autofluorescents. So they're dying, and when they die, just they explode like little galaxies and stuff.
Wow. It is like a whole universe. I mean, I'm not sure if you saw the last talk by John Warhol, but he was saying the microbes have stories and we have to tell the stories. And I think, telling the stories of these organisms is something you both do really well in your microscopy. Is this the one that's behind you?
This one?
Or is this a different microorganism?
No, this is a stentor behind me. It's I can also pull out some videos of them. So this stentor is a large single cell organism. They're quite common, but they're also some of my favorite microbes to view under the microscope. So let me, oops. So these are some. Can you still see the video?
Yeah, we can. Some amazing colors here.
Okay, perfect. So these are some stentor roselli, and they are viewed under the polarized light. And I have some other stentor species. This is stentor amethystinus, and it has like this purple natural pigment, and it also has some algae living inside. It's in the symbiotic relationship. So the algae using the sunlight and producing sugar and sharing some of the sugar with the host stentor. And in exchange, the stentor is just protecting them from predators, because stentor is one of the largest organisms in the pond. So you want to be, friend with them, so they can protect you. And so we have, I'm sorry, I'm just pulling some videos from our Microcosmos archives.
Oh, no problem.
Yeah. So yeah, this is the one behind me.
Yeah. Oh, cool.
Yeah. They're really beautiful.
Is it playing or is it frozen for us?
Can you?
Oh, it might be frozen for us.
Oh, okay. Sorry, I was.
Ah, here we go. Yeah, now it's doing it.
Okay, sorry.
Oh, amazing. Yeah.
Yeah.
Wow. So is it covered in cilia then?
Yes.
Moving it about, yeah.
Mm-hmm.
So you can see the, all the different symbiotic algae within it as well, the chlorella, the little green cells. This one's really cool because it has the circular Maniliflora macronucleus. So it looks kind of like a bead of rosaries. Those little clear dots are the nuclear apparatus. So it's really cool to see this just from a cell bio standpoint. It's such a massive creature, and you can see its internal organelles. And those rows, those lines are the actual rows of cilia, which project out of it. You can see them really nicely at the top there, coming out of the mouth. Beautiful, beautiful footage, James. Well done.
Wow. Yeah, it moves just so gracefully.
Yeah. And they, as I said, they can be quite big, and they can like look at the size of this, for example. Yeah. And this is like I think this is 2 millimeters in size when it extends fully. So we have some other ciliates. These are Frontonia. Hunter wrote a paper about Frontonia. I think, which one was that? Frontonia
Frontonia vesiculosa, yeah, the largest one of the Frontonias. Have you found that yet, James, in Europe?
Not yet. But I'm,
right, well, I'm waiting.
For that. Yeah.
You give me a call, and Eva will be very happy as well.
Yeah.
some healthy cross-Atlantic rivalry, I guess.
No, really, it'd be great if you could find it, because no one's found it before in Europe. It's only known from these really warm environments. So I found it here in Florida. It's known in Africa and South America. So if James or somebody else, on YouTube or Instagram can find it and post a video, it'd be really cool record, the first for Europe. So that'd be really exciting.
Yeah.
Hopefully he will.
But do we think it exists somewhere, it's just not been found, or could it just be it only exists on the American continent?
Yeah, I mean, the stuff that we publish is it can thrive wherever it finds its preferred ecological niche. So, theoretically, it should be able to thrive in parts of Europe where it is warmer. It likes warm, shallow waters with a lot of, stuff to eat, which Europe obviously has. It just as Europe has a bit, cooler waters. But I think in a time period, in the summertime, when you get warm waters, these things should thrive there. So only a matter of time till somebody finds it.
Okay. So you need a warm country with lakes or shallow lakes.
Yeah, shallow, eutrophic freshwater ponds or water bodies would be really ideal. Some kind of a swamp with water around 25 to 32 C would be perfect for the Frontoni vesiculosa. And it's about 1 millimeter in size. So if you see it, you're going to know. It's a very obvious feature.
So it's a very visible microorganism.
Yeah, that's one of the things I think doing microscopy, because at some point you don't need a microscope to just identify some cells. For example, like if I see a paramecium, I can say like, okay, this is a paramecium just by looking at the tube or something, because how they swim, how they move. So it's like if I see Frontonia, I would be like, okay, this is interesting. What is this? So I'm just keeping my eyes open. But it's like in Poland, I think we it's quite cold, but still, because as Hunter said, like it's the theory is like everything is everywhere, but the environment selects. So Frontonia, this species of Frontonia should be everywhere, all around the world, but it prefers to live in a certain environment. So if you find the environment, if you sample enough, environment in this organism's range, you will find it eventually.
So with this lovely blue background you've got, are you just using a blue underlight, or?
No, this is because this is a DIC footage, but with DIC you need DIC illumination based on polarized light. So you need to set 2 sets of filter, like with a correct angle to get the DIC footage. But this blue footage is when you set the DIC in a wrong way. So it just like, this is not a correct thing. So if you talk to not a microscopist maybe, but like an engineer, they would tell you, okay, this is a wrong way to set it, but it looks beautiful.
And so, okay. So you wouldn't do research like this, but for making nice images, it adds so much color. Yeah. Can you set it in such a wrong way that you get different kinds of color?
Yes, exactly.
Yeah.
You can do that. I'm not sure if I can find any footage right now of that, but yes, you can do that.
That's one of the cool things with Instagram. Some of the other accounts I follow, which do similar stuff to James and I, they'll publish on Instagram some crazy colored videos, and you think, what the hell are you guys doing? But it looks so amazing, and it's such a unique technique. You almost want to, have them write it in the description so you can kind of emulate it, because it's really cool stuff.
Nice. So we've got about 13 minutes left. Should we see if we can find anything in that soil sample?
Yeah. Do you want to?
Yeah. Yeah. So we basically have just some soil here in water, and we're just going to see if we can find anything alive in it while we have a few more minutes. So I'll pop this over here and we'll see what we can do. But in the meantime, feel free to show us some more lovely videos, because this will take a couple minutes to set up.
Oh yeah?
James, do you have any sporosum semivirescens you can share?
Yeah, sure. Like when I found it, I recorded.
I'll tell the audience the story. So sporosum semivirescens is a really cool ciliate. It's well over 1 millimeter in size. It's full of symbiotic Corella algae. And I found it years ago with Dr. Genevieve Esteban, my PhD supervisor, who's now James's as well. We found it in England. We flew to Sweden to find it there and showed that this ciliate is looking the same and also has the same morphological signatures So it was really neat, but it's very rarely recorded. So a really cool thing with social media, James was literally live online doing an outreach thing for Journey of the Microcosms, and he found this ciliate for the very first time in his samples. So it's really cool and like a great example of how, social media can actually produce some real science, because that was, a new record for his country, and somebody could be finding this in a totally different continent no one's before seen it in. So it was a neat bit of kind of collaboration there. And here's just a fabulous shot that James is showing now of this very ciliate from his samples.
Yeah, it's a big one.
And really cool too to have a real discovery made live on YouTube. So that's just such a fabulous piece of science there James was involved in for this particular species.
Yeah, I was blown away because I didn't expect to find it, and it was like just a live stream, and
your reaction is so good.
Green was like swimming, and it took me like 5 seconds to realize.
Yeah, anything that big, right?
Yeah,
one thing it could be.
And
this one has the maniliform macronucleus as well, similar to the stencil we saw earlier. So it has those clear bead-like circles. That's the nuclear apparatus, which is really cool.
I'm trying to locate some lower magnification footage.
This is a species I've been trying to find here in America, in Florida, but it just seems like it's too hot in Florida for this guy. So I haven't found it yet, sadly.
Another person who does something on social media is my microscopic work, and he also found that recently, I think, in Denmark.
Yeah, in Denmark. Yeah, I saw that video. That was great.
And there's also one person, David Seymour, I think. He found it in Australia.
Oh, really?
Yeah, and at the moment, I think he's in contact with Professor G. So like they're trying to find some old footage that he recorded, like old photographs and stuff. And I think he found it in 1996. So it's like, yeah, people are finding it, and it's like because of the social media.
Back to those old sites. Let's take a look. Yeah. Obviously, the whole continent of Asia they haven't recorded on, or the continent of Africa also species. So it'd be great if anybody watching is in those areas. There's a lot of really cool and really obvious units that are out there to be found. It'd be really nice contribution to biogeography.
You can share the screen as well, I think.
And here I think it was building because this is a ciliate that builds like a little house for itself called lorica, and I think it's trying to build a lorica here because this is like this is a pretty common behavior with every lorica building ciliates, like for example with Stentor-Roselli, and they just stay stationary and they go back and forth, and they are just collecting like particles around them and binding them with some sort of mucilage. I think they release, like maybe it's not, it doesn't call it mucilage, but with some secretions, they are just collecting the particles around them, and then they're just building this long lorica. But mine was under slide, so it didn't build the lorica in the end. I wasn't able to like record the whole thing. But yeah, it's a gorgeous ciliate.
Beautiful, yeah. Great footage too.
And it's, I think this one was around 2 millimeters long. This is like 200x magnification. So like the field of view of around 1 millimeter. So yeah.
And they're only like 50 microns or so, 40 microns wide. So it's really a neat creature.
Yeah.
Okay, so what we have here will not be as beautiful, but it will be live.
I'm gonna stop sharing.
And there are some things moving around and wriggling in there, so we'll try and see if we can find something. Okay. Ooh, there's something alive.
Something alive.
That's most likely a cold-blooded. It's like I have a book next to me trying to identify one.
Yeah, that's the Feuchner book, right?
Yeah.
Yeah. A series of like 6 he wrote.
Yeah, it's like
underneath the
800
lump.
page just on one group of ciliates,
Yeah.
and it's like
exactly.
Yeah, they're
Yeah, so this Colpoda, they're very common in soils, and they make resting cysts. So
Yeah.
once you add water or add different foods, they'll come back to life. So pretty much immediately you're going to see Colpoda in these kind of samples. So that's probably our best guess.
I mean, so we took this soil from the plant pot in this room. So
So
that
there's life
seems-
everywhere. Yeah, we're surrounded
Yeah.
by microbes. Yeah.
there it is. So it does bob up and down a bit, making it hard to focus.
Yeah, ciliates are not super easy to work with, that's for sure. The bigger ones are a bit easier sometimes, but not always.
Can we get in closer?
No. Ooh.
Very suspenseful, isn't it?
Yeah, it is. I mean, the thing is, I'm sure you guys are aware that this actually takes a lot of time and patience, not 7 minutes. But maybe we'll find something worth looking at. I'm going to go back up to, I think this is 100 times or so. One of the difficult things is I'm not so used to using...
That doesn't work. 'Cause it requires-
Oh, that's the oil one. Yeah, I went the wrong way, didn't I? I want this one. Bring it in. Let's see if it's... Oh, there's something tiny there.
Yeah, I'll let James identify that one.
Yeah, which one's that one, James?
It'll be in that orange book you have for sure.
Yeah. There we go. It is quite suspenseful in the sense, you know, looking for something alive and seeing what we find.
And the cool thing with soil is too, there might be a lot of cysts which aren't yet, active. So if you were to feed it something or change the temperature, you might have a huge bloom of some species which is cryptically present, which is kind of interesting.
Yeah. I mean, we only added water to it like this afternoon, so
maybe
stuff.
a couple of days will be a lot more, and that's another confounding problem, with identifying all these species.
Yeah. Well, we'll keep an eye on it then. We'll keep the soil sample around.
Hours, right?
Yeah.
Yeah,
Check it live.
just add a grain of oatmeal or something, and it's like
Yeah, okay.
It's just gonna be like really cloudy around like the food that you add, and then they're gonna be like literally tens of thousands of ciliates.
Yeah.
Oh, that'd be cool. Okay. Well, we'll do this and then hopefully find something fun in the next few days.
Maybe I can show some videos that I took the other day.
Yeah. Do you have them, are they on this laptop?
Yeah.
Yeah. Okay. We've got 3 more minutes before we have our scheduled break. So we'll just show a few final videos. Yeah, go for it. But at least we did find something alive. I think next time we should breed like a sample for several weeks on a radiator with a little bit of sugar and then see what we find in there.
Absolutely.
So yeah, this is an upgrade from the microscope we had last year, although we still need to get used to using it and making great videos with it, but we hope to start using it a lot more at FEMS to make nice content.
Right.
Yeah.
Practice
So yeah. Oh, nice.
Nice.
So
Ooh.
this was a Daphnia.
Yeah, play it again. So what was it, Lenny? You can tell us.
Oh yeah, well, this was from the Delft canals, and it looks like a turtle, but I found out that it was actually a Daphnia. Yeah, so, well, I put a cover slip on it, and then I killed it, and then I could take a nice picture of it. But yeah, I quite found that if I add the cover slips, they die. Well, if they're big enough, they get killed.
Need a little well slide, a little well in it.
Okay, we'll buy some of those then. So did it split?
Yeah, with the well slides, the issue is like you cannot get good focus, so you need to like I use like flat slides, but I put
So this is how it looked like when I killed it. Close up.
Yeah.
Great image.
This was the same, but I just added some color to it, how it looks a little bit smushed up. Maybe we can look at this video.
It's called, I think, Cymocephalus. I'm not sure if that means.
So yeah, this is like the whole body, but it was fascinating because you could see it breathing at some stage.
Oh, did they have like a gill or something?
Yeah.
Or something, I don't know. Does that video play? Or did you just show it?
Let me just see how it. Bees are not playing. Okay.
Our final video.
It just happens that they swim quite fast, so it's just really hard to chase them under the microscope when it's like.
So skittish.
Yeah, I think it's at the end that I got it. Yay. It's just difficult when they fly, and then it's difficult to, get a close-up.
Yeah, okay.
We know, trust us.
Do you have any tips?
I like to make a bunch of slides and let them wait for a little while, because as the water might evaporate or as the light might start to bother them, they'll start to slow down. So there's that sweet spot before they die or they get smushed, but they're just slow enough. So that's what I do for ciliates especially.
Okay.
And the more you watch these things, the more you get to know the motion. So it's a bit like a video game. You can follow.
So this is my best.
Lovely.
This is my best shot, I think.
Very artistic. You see the eye spot there.
I was like, get in the middle.
Yeah, that's
You can see the heartbeat in there on the left. Yeah.
There.
Like you can see the muscles controlling the eye.
So that circular part is its heart, and then
Middle left.
it has like internal legs that it uses for respiration and stuff like that. Yeah. This was like my second try on this microscope.
You did great.
Yeah.
Yeah.
Well done.
You had fun doing it. That's all that matters.
But yeah, thanks. I found it quite hard, like, to get the technique ready, because I tried like not putting a cover slip, but then putting a cover slip killed some of them.
I'm sure
It
you
just
get it in time.
yeah.
There's plenty of Daphne out there, so plenty of time to figure it out.
Ooh, we're hooked under. Yeah, I was going to say, unfortunately, we've come to the end of the microscope session because we had to have a short break and then restart the streams because Facebook puts an 8-hour limit on streams, which is very irritating. But thank you both guys for joining us today and showing us your videos and chatting to us about microscopy, and hope for inspiring some other people to go and do their own microscopy and put it online.
Thanks
And
so much for
yeah.
your time. Appreciate you guys having us.
Yeah, it's been great fun. Tomorrow is International Microorganism Day for everyone here. I think it is already International Microorganism Day in Japan and India and China. But I hope you celebrate tomorrow as best you can on a nice Friday evening and tell everyone you can that it's International Microorganism Day. And yeah, we'll bid you farewell. And thank you for joining us on the live stream.
Thank you so much.
Take care, guys. Best of luck.
Bye.
No worries. We'll see you soon. Right, so we're going to have a small break. And then we will see you on the other side of that break in roughly 20 minutes or so. But yeah, see you soon.